So good morning, everyone. Let me begin by congratulating and thanking the organizers of this joint symposium honoring the legacy of Joan Guinovart. This remarkable initiative, bringing together IUBMB, FEBS, and the IRB, it is a fitting tribute to an extraordinary scientist and extraordinary citizen. I would therefore like to thank the symposium organizers, the past president of IUBMB, Alexandra Newton, the FEBS treasurer-elect and chair of the Integration and Networking Committee in FEBS, Jerka Jumik, and the director of the IRB, Francesc Posas, for their initiative and dedication in making this meeting possible. And of course, the current IUBMB president, Dario Alessi, so well known at the IRB for his continued support and friendship. I would also like to thank the invited speakers for kindly accepting our invitation and all the registered participants who are joining us today, either here in person or online. Well, on January 1st, 2025, with deep sadness, we learned of the passing of Professor Joan Guinovart, a distinguished scientist, a visionary leader, and a cherished colleague. Joan was a professor at the University of Barcelona and at the Institute for Research in Biomedicine, the well-known IRB, where we are just meeting today. His groundbreaking contributions to glycogen metabolism, together with his role as founding director of the IRB, left an indelible mark on the molecular life sciences community in Europe and around the world. Today, colleagues, collaborators, and friends here gather to honor his memory, recognize his leading role in glycogen metabolism research, and celebrate his remarkable legacy. As the current FEBS Secretary General, and above all, as a close friend, it is a great honor for me to speak in his memory at this symposium. I used to meet Joan in the 1980s at the annual congresses of the Spanish Society for Biochemistry and Molecular Biology, SEBBM. He, Joan, as a brilliant young professor from the new wave, and I as a novice apprentice. Our closer contact began in 1995 when he served on the committee through which I obtained my professorship at the University of Seville in Spain. Just a couple of years later, shortly after Joan has assumed the presidency of SEBBM, we met again at the course in Jaca, northern Spain. With his irresistible powers of persuasion, he entrusted me with organizing the SEBBM Congress in Seville in 1998. That same year, he also invited me to accompany him as a vice president of the SEBBM during the second half of his term. This marked the beginning of a long shared journey in our lives. He gradually opened the doors for me to the circles of the International Union for Biochemistry and Molecular Biology, IUBMB, and the European and Pan-American Federations of Biochemistry and Molecular Biology, FEBS and IUBMB, where I learned firsthand the arts of scientific and diplomatic negotiation. For almost 30 years, we traveled the world together, always with Rosa, his wife, at his side, building a deep friendship. His vision for collaboration between FEBS and IUBMB led to the first joint IUBMB FEBS Congress in Birmingham in 2000, paving the way for regular joint meetings and a lasting partnership. At Joan's initiative as FEBS Congress Counselor, and with the support of Vicente Rubio as President of the Spanish Society, Irene Díaz-Moreno and I myself prepared in 2006 the bid to organize the joint IUBMB FEBS Congress in Seville in 2012. Together, we formed the core management team for the event, with Joan as the keystone in this role as chair of the scientific program. The Congress turned out to be a great success, attracting 2,500 participants from 73 countries. Indeed, the Congress is scheduled to be organized jointly again by IUBMB and FEBS in Spain in 2030, following the express invitation of both organizations. As president of the Spanish Society for Biochemistry and Molecular Biology, Joan played a pivotal role in its development and strengthened its international presence. But one of Joan's most notable achievements was the Confederation of Spanish Scientific Societies, COSSE, created in 2005 to promote the dialogue and collaboration between scientists and politicians. Reflecting Joan's interest in serving as a bridge between the two were the dinners he organized near the Spanish Congress of Deputies, the parliament, bringing together presidents of the scientific societies and members of the parliament. As the first president of COSSE, Joan led the organization with a clear and pragmatic vision, demonstrating that cooperation and mutual understanding are essential to addressing the challenges of the 21st century. Within FEBS, Joan served as a Congress counsel for 9 years, where he introduced a standardized model for FEBS Congresses. As a member of the FEBS Executive Committee, he was instrumental in laying the groundwork for the establishment of the European Research Council, the well-known ERC. He served on the FEBS Society and Society Committee, advocating for stronger connections between science and society. In recognition of his many contributions, Joan received the FEBS Diplôme d'Honneur in 2007 and the Israel Pech Award in 2024, honoring his outstanding dedication to the molecular biosciences community and his efforts to strengthen FEBS societies through collaboration. But Joan was not only president of the Spanish Society and COSSE and a member of the FEBS Executive Committee. He also was the second Spaniard after Severo Ochoa to serve as president of IUBMB. Across all 4 institutions, he made clear his commitment to promoting science beyond borders, strengthened ties between countries under the banner of science, and worked tirelessly to consolidate Spain's place on the global stage of knowledge. One of the last times we met was actually in July 2024, a few months before he died, at the FEBS Congress in Milan, when I had the pleasure to present him with the first Israel Pech Award. The illness had left him weak, but he remained full of spirits and enthusiasm. There, I told him about the meeting with IUBMB and all the other continental federations worldwide, from which emerged the Milan Declaration on the crucial role of science in addressing the global challenges facing humanity. He loved the idea. He was perfectly aligned with the guiding principle of his life. Beyond his academic achievements, Joan was a true humanist. For him, the scientist is not an isolated figure in a lab, but a member of society, responsible for contributing to the community's well-being. It is a pity he did not live to see the Milan Declaration made public at the end of January last year, a few days before he died. Beyond his scientific achievements, which we are going to remember here today, Joan will likewise be remembered for his gentle personality, his vibrant spirit, and his readiness to share a joke that could brighten any room. He was a generous and supportive mentor, always willing to share his experience, offer advice, and encourage younger scientists and colleagues. Joan Guinovart was much more than an outstanding scientist. He was a bridge between cultures, a champion who understood the social and political interconnection of science. His greatness as a researcher was matched by his humility as a person, leaving a legacy that inspires all who seek knowledge to build a better world. His example reminds us that true greatness lies in the ability to serve others with passion, integrity, and respect. Yes, we lost a deeply admired scientist, a person of universal stature. But above all, we have lost someone who makes you laugh endlessly, someone who makes you believe that there really are good things in the world. That is eternal friendship. This is a quote by Pablo Neruda that better describes Joan. Through this symposium, IUBMB and FEBS wish to express their deep appreciation for Joan's remarkable impact on both organizations. And above all, we celebrate a life that enriched science, strengthened our community, and touched the hearts of so many. Thank you very much.
Just, sorry, I had to put my talk back on there. I've got some slides. Let's see. Great. Well, thank you very much. I was asked to, say a few words about the contribution that Joan made to the International Union of Biochemistry and Molecular Biology. And I just want to start saying I think I met Joan about in the late 1990s when I was going to conferences on signal transduction pathways. And then he invited me to join the scientific advisory board of the IRB, which was it was the first time I'd been invited to do something like that. So I used to come here twice a year, or once a year probably, for about 10 years. And it was such a wonderful experience to see this institute and how amazing the sciences was, and his interaction with the scientists and the amount of time that he put into building and creating a world-leading institute in Barcelona. And I think IRB has become one of the most well-known Spanish institutes in the world. And I think this is really largely due to the pioneering work that Joan did in building and creating this institute. And this was a picture I took in 2022 when he invited me for lunch in the Barcelona Yacht Club. It's a very lovely location, and it was the first warm day of the year. And then he took me for a lovely walk along the beach afterwards, and it was a really spectacular day. And I remember him telling me about his philosophy on life. He really told me that there's talent everywhere in the world, that everywhere's got talent, but opportunities are very limited for lots of people. And his job was to try and build opportunity for the scientists in Barcelona, the children, and the researchers to come here and achieve something. And that was basically his his underlying philosophy and everything that he did. So he was a huge contributor to the IUBMB. For over 25 years, he dedicated a lot of time and effort to serving in various committees. He then served as the treasurer, and then he became president-elect, and then he became the president, and then he also at the same time spent over 20 years running becoming the editor-in-chief of the IUBMB Life, which is the main journal of the IUBMB. That brings in a substantial amount of income that we can use then for supporting fellowships, congresses, and other activities, educational activities, that we run. And even now, he's over 10 years since he's been president in the IUBMB. A lot of the fellowship schemes, a lot of the way we operate, the way we have monthly EC meetings, and our interactions with other regional organizations around the world. Everything we do is basically a legacy of what Joan set up during the time he was president. And you know, everyone tells me he was an incredibly collaborative you know, leader, and you know, his approach was always to get, you know, consensus and to make transparent and collegial, you know, decisions. And you know, his focus was to support the young scientists, promoting biochemistry, you know, especially in developing regions. He really loved going to Asia, South Africa, and also you know, South America. And he was also dedicated a huge amount of time, as I said, forging relationships. These are the other bodies, you know, that run biochemistry and molecular biology, you know, in the Americas, Africa, Asia, and Oceania. And he really sort of integrated these and made sure that the fellowship schemes went especially to those countries, and you know, and gave opportunities to people in those countries. And in fact, I've met so many people over the last few years who've been able to come to Europe or go travel to other countries through IUBMB fellowships, and that's really played such a transformative role for the many people that I've talked to in their, you know, in their careers. He was also very passionate in education and training, and you know, and he was very dedicated in providing opportunities to everybody all over the world in his actions. And I just got some other sort of photographs here that I wanted to go through. That you know, he was you know, especially he was passionate to help young people. I know he ran a school. I think every Saturday morning, in the IRB, I don't know if they still do this, but they open their doors to local school children to come into the building to learn about science, and you know, and the research that goes on here. And he was personally involved in driving this. And even the last few years of his life, he was dedicating all of his time to you know, to nurture, you know, science for the young people in Barcelona. And then he was also, a very good diplomat. He, you know, he really had fantastic relationships with scientists all over the world. He had this incredible network that he brought together. And, you know, I remember this dinner, I was actually present there. And this was, you know, obviously Miguel and Ilona that are here today, you know, who runs the fellowships, you know, committee for the IUBMB and the conferences. It's it was a lovely evening, and Joan did you know, was really telling us about how we need to work together and collaborate. And you know, and he was also very important. He really saw a lot of that. The humanistic and ethical vision of science was incredibly important, you know, for him and his philosophy. So I just want to come to the end here. I have you know, a couple more slides, a slide in the video I want to show you. And you know, so I think his I think his commitment to human leadership and exceptional sciences is his legacy. And you know, he really helped create a better world for the biochemists, you know, and the molecular biologists. And then he had a lot of friendships, and I've got some statements here, but I want to leave as much time as possible for the scientific presentations today. So I'll skip through this, and I just want to go to this video. So this video was I had dinner with Susan Taylor and Alexandra Newton, and I think this was in 2023, and I think this was the very last time that I saw Joanne, and we were just coming to the end of the dinner. And for some reason, I never do this. I had a feeling that I wanted to video this conversation he was having with Susan Taylor. And it was a very detailed scientific conversation, you'll see, but I think it shows a lot about Joan's personality and listening, being very diplomatic. Maybe he didn't agree with everything that Susan Taylor was saying, but he does it in a very nice and friendly way. And this was my last time I actually saw Joanne, and I'll just play. It's a 45-second video, I think. I don't know if we have sound. Okay, anyway, that's all I wanted to say. So we'll switch that off, yes. And and I think we're excited now to get onto the science, and we'll hear about the beautiful work that Joan has done in his research career, and Alexander will introduce the seminar. Okay, thank you.
So I'm just here to chair the morning session and keep our speakers on time. But I will say that Joan, when I became president-elect, he was the past president, and he became, at that moment, my forever mentor. I just turned to him for everything. I learned so much from him. He was humble, his sense of humor was amazing, and every time I asked him a question, he would go, well, go to statute 4.6.7 on page 53. He was an encyclopedia of knowledge, an absolute scholar. He knew ancient Greek. I speak Greek. He would often send me little notes in Greek. Just an absolutely remarkable person. So it's my great honor to introduce our first speaker, the legendary Philip Cohen, who is going to give us, I think, an overview on glycogen, but then speak a little bit about his own research from glycogen away and back to glycogen through ubiquitination, if I'm not incorrect.
Okay. Put it on there. Where's he going? He's supposed to be. Right. Very good. Okay. Well, 51 years ago, I met my scientific hero for the first time. Thank you. Carl Corey. And the first thing he said to me was, Philip, glycogen has been the greatest gold mine of biochemistry. And by that he meant that a number of people who worked on glycogen made discoveries of very general significance. And in fact, 6 of them were awarded the Nobel Prize. So, of course, first of all, there was Carl himself, and his wife, Gertie, who won the Nobel Prize for Medicine or Physiology in 1947 for discovery of the course of the catalytic conversion of glycogen. They discovered glycogen phosphorylase, how it broke down glycogen to lactate in muscle and to glucose in the liver, and how lactate produced in the muscle was reconverted to glucose in the liver. Gertie, by the way, was the first lady to get the Nobel Prize for Medicine. However, the Corys also got things wrong. For example, they thought that glycogen synthesis and glycogen breakdown were catalyzed by the same enzyme, viz. Glycogen phosphorylase. And this was shown to be wrong by this gentleman, Louis Leloir, who discovered the enzyme glycogen synthase, and more importantly, he discovered its substrate, UDP glucose, and went on to find the many roles of UDP glucose outside of glycogen metabolism, including, for example, UDP glucuronic acid. So he's also the founder of what we know about drug detoxification as well. He was also the only Argentinian to receive the Nobel Prize for Medicine for work done in Argentina. There have been other Argentinians who've won the Nobel Prize, but they did their work outside of Argentina. Okay, so, and then the Corys didn't discover how glycogen was broken down in response to adrenaline, it was actually one of Carl Corey's PhD students who did that, Earl Sutherland, who of course discovered cyclic AMP, the first second messenger that mediated in the system he was studying the adrenergic control of glycogen breakdown. And then the Coreys in the 1940s, they found that glycogen phosphorylase existed in two forms, an inactive and an active form, but they had no idea what the difference was between these forms. And it was actually one of Carl Corey's postdocs who made this discovery, Edwin Krebs, who when he stopped fighting in the armed forces in the Second World War, where he worked actually as a clinician in the war, he then joined Corey's lab and learnt about phosphorylase. But when he became an assistant professor at the University of Washington in Seattle, he teamed up with Edmund Fisher, shown on the right here, and the two of them formed this wonderful team that led to the discovery of the first example of enzyme regulation by reversible phosphorylation. In other words, that was, what the two different forms of glycogen phosphorylase really were. So I got into the glycogen game in 57 years ago now, when I joined Edmund Fisher's lab. When I, a couple of years later, set up my own lab for the first time, to me it seemed that the really big unsolved problem in the area was how did insulin work. When I started, this was sort of what was known about. It was known that insulin had a receptor, but it hadn't been purified or characterized in any way. And then there was the seminal work of Joe Lana that Anna will say more about later on. But glycogen synthase, Lana found, was inactivated by a phosphorylation mechanism. And what insulin did was to promote the dephosphorylation and activation of glycogen synthase, and that's how glycogen synthesis was stimulated by insulin. Well, what was in between was still a sort of a black box. So I sort of started working on this in the 1970s, and in the late 1970s, a PhD student of mine called IUBMB, a Malaysian PhD student, he discovered an enzyme that we called glycogen synthase kinase 3, or GSK3 for short. This sort of was a very strong inactivator of glycogen synthase. And then a postdoc of mine a few years later, Peter Parker, he found that insulin stimulated the dephosphorylation of glycogen synthase at the sites that were targeted by GSK3. So it looked like GSK3 would be important in insulin signaling. And it was in this year when Peter published his paper, that 1983, that I first met Joan Guinovart. I got invited to give 3 lectures on glycogen not in IRB Barcelona, which didn't exist at the time, but at the University Autonoma Barcelona, where the head of biochemistry was Claudio Cucilio, who actually had been a student with me at University College London some years earlier. I don't remember very much about my conversations about glycogen or about the lectures I gave, but it was probably something to do with what I'm showing on the board here. The one thing I do remember is that Joan took me to the biggest champagne manufacturing plant in the world, the Fresnay champagne plant. And why did we go there? Because Claudio Cucilio, he knew the owner. And so we had a personal tour of the Fresnay champagne factory from the owner. So that was what I really remember about my first meeting with Joan and the fun we had, you know, at Fresnay. Okay, so anyway, eventually in the 1990s, we did sort of eventually find out how GSK3 was regulated. And on the right-hand side, there's my student Darren Cross, and he's the guy who found that GSK3 was phosphorylated and switched off by a kinase called AKT, also called protein kinase B. And then a very young Dario, at that time a postdoc in my lab, who had been working with Darren Cross, went on to then find that PKB was also inactivated by a phosphorylation mechanism, and that it was activated by a phosphorylation mechanism, and then he went on, Dario, to find the enzyme that did it, called 3-phosphoinositide dependent protein kinase 1, or PDK1 for short, because of the key discovery that PDK1 could only activate PKB in the presence of the insulin second messenger PIP3. And at about the same time, or just 2 or 3 years earlier, there had been the discovery by Lou Cantley of PI3 kinase, and by others that its key substrate was PIP4P2, and hence the product was PIP3, the insulin second messenger. And Ron Kahn, of course, in the 1980s had discovered that the insulin receptor was a tyrosine kinase, and that it worked by phosphorylating these insulin receptor substrate proteins, which became phosphorylated and recruited, PI3 kinase. So finally, by the mid-late 1990s, it became clear what the outlines of insulin signal transduction were. And so I decided, as it was no longer a black box, I should leave the area and go on and work on something else and find another black box. And I sort of became very interested in how the innate immune system was controlled. And that's what I've been working on for the last more than 20 years now. And I'm still working on that. But that's how I got into glycogen. So it turns out that the innate immune system is regulated by an amazing interplay between phosphorylation and ubiquitination. You have kinases that activate E3 ubiquitin ligases. You have ubiquitin chains produced by E3 ligases that bind to and activate other kinases, and that's how the whole system is controlled. So I'm not going to tell you anything about that, but to jump a few years to tell you about something we've discovered very unexpectedly starting about 7 years ago. So, and to do that, I first have to introduce one E3 ligase called LUBAC, the linear ubiquitin assembly complex. It consists of 3 proteins called HOIL1, HOIP, and Sharpin, and it was discovered by Fazu Iwai in Japan. And he not only discovered this complex, but he discovered that one of the components of this complex, HOIP, was an E3 ligase, a ring in between ring domain E3 ligase with two ring domains, which produced a type of ubiquitin linkage that had never been discovered before, called a linear ubiquitin oligomer, or we prefer to call it a MET1 link ubiquitin oligomer, because it links the C terminus of one ubiquitin to the N-terminal methionine of another ubiquitin to form a real peptide bond, not the isopeptide bonds, you know, that nearly every other E3 ubiquitin ligase forms between the side chains of lysine residues. Y also discovered that these MET1-linked ubiquitin oligomers are bind very specifically to particular proteins, such as NEMO, the regulatory subunit of the I-kappa B kinase complex, which is very important in activating the two key transcriptions of the factors of the innate immune system, NF-kappa B and IRF5. Okay, but you may note here that there are these two other components, and what do they do? And most of my talk is now about HOIL1. Some of you may have noticed that, like HOIP, it also is a ring in between ring E3 ligase, and it has actually a cysteine residue in the same position as the catalytic cysteine in HOIP. But no one had been able to find out what it did for many years. Some people said it was inactive, like a pseudo E3. Some people said, well, it made MET1 link ubiquitin chains, but it was so pathetically slow, it couldn't possibly be very important, and HOIP was really the important one. Anyway, to cut a long story short, 7 years ago, Ian Kelsall in my lab, who's shown here, discovered that it's a very unusual E3 ligase. It actually attaches ubiquitin to serine and threonine residues in proteins, forming ester bonds. So Lübeck is really a unique complex. It's got these two E3 ligases, both of which do things that no other E3 ubiquitin ligases do at all. And for example, Ian went on to show that serine 365 is auto-ubiquitilated by HOIL1, and we went on to find other substrates in the innate immune system. But a few years ago, Alicia McCrory, who was my 50th PhD student to graduate in 2023, she found that HOIL1 can also join two ubiquitins together to form ester bonds involving the C terminus of ubiquitin being joined by ester bonds to threonine 12, 14, 20, or 22 of another ubiquitin. Okay, so obviously we wanted to know why was this important in innate immunity, and a postdoc, Svetlana Petrova, who still works in my lab today, actually using a knock-in mouse that we'd made in which we replaced HOIL1 by a mutant that has no E3 ligase activity. She showed that actually this E3 ligase activity of HOIL1 actually has positive and negative effects on innate immunity, which depend on the ligand receptor and the cell type. This is sort of rather unusual, but it's actually consistent with the finding that HOIL1 deficient humans display a combination of immunosuppression and autoinflammation, which is sort of pretty unusual. Okay, but that is actually not why these patients die. The patients who are deficient in HOIL1, who, by the way, for people who work in the glycogen field, that we'll come back to in a moment, call RBCK1. So HOIL1 and RBCK1 are the same enzyme. I won't say why the gene is still called RBCK1, but the E3 ligase I'll refer to as HOIP. Okay, but what these patients who are deficient in HOIL1 do is they develop cardiomyopathy during adolescence, and they die from heart failure in early adulthood, unless they are given a heart transplant, pretty quickly. So why is that? So the cardiomyopathy and heart failure in these people is caused by the gradual accumulation of insoluble deposits of glycogen with few alpha-1,6 branch points called polyglucosan bodies, or PB for short. And so we looked at our HOIL1 knock-in mice that had no HOIL1 E3 ligase activity, and we discovered in a variety of tissues, like particularly heart and brain, that between 6 and 12 months of age, they developed sort of very large amounts of deposits of these polyglucosan bodies. And that indicated that the E3 ligase activity of HOIL1 had a completely different function outside the innate immune system to prevent the accumulation of unbranched glycogen. So the question is, what was the mechanism? So, as you can see, much to my great surprise, after 20-odd years after leaving the field of glycogen metabolism, I found myself back studying it again about 3 or 4 years ago. So this is the question I want to just sort of discuss with you as the last part of my talk. Why is HOIL1 required to prevent the accumulation of polyglucosan bodies? And Ian has to... Kelso has, I give him great credit again for making a leap, and that was, well, if HOIL1 ubiquitinates the hydroxyl groups of serine and threonine residues forming ester bonds, perhaps it ubiquitinates other hydroxyl groups in proteins, including glycogen. And so using a small unbranched glycogen oligomer, maltoheptose, with 7 glucoses linked by alpha-1,4 glucosidic bonds, he showed that HOIL1 ubiquitinates this small peptide. So I just show you this here. This is just a HOIL1 catalyzed ubiquitination reaction. It has all the usual goodies in it, but here's ubiquitin, and this is ubiquitinated maltoheptose. You can see it. The ubiquitin is very rapidly converted within, a couple of minutes to the monoubiquitinated species. But this requires you to add small MET1-linked ubiquitin oligomers, which are produced by HOIP. If you do not add these into the reaction, what you find is that the reaction is incredibly slow. Even after 2 hours, you can see there's only a small conversion to the monoubiquitinated species. So adding these MET1-linked oligomers speeds up the process way over 100-fold. And then Ian made another very important finding, and that is that if you actually look at the binding of HOIL1 and HOIP to immobilized unbranched glycogen, he actually just used amylose resin that you can buy, from Sigma and other places. And then you just mix it up and spin down the amylose resin and see what comes down. He found that nearly all the HOIP came down in the pellet, but the HOIL1 remained in the supernatant. So HOIP, and so we sort of formed the idea that HOIP does sort of 2 important things here. It forms the MET1-linked oligomers that act as an allosteric activator of HOIL1, and it also is important in bringing Lu back to unbranched glycogen. And Elisha McCrory, in collaboration with Steve Matthews, a very good NMR spectroscopist in London, found that the ubiquitination site on maltoheptose is actually the 6-hydroxyl group. So actually, what is being ubiquitinated is the hydroxyl where the branch points in glycogen actually start. So based on these observations, we came up with a working hypothesis that unbranched glycogen glucosaccharides, they're occasionally formed by errors of metabolism. If they're not removed rapidly, they become longer, more insoluble, and they precipitate as PB, causing cardiomyopathy. That Hoyle-1 is, when this happens, when you don't get branching to operate, Hoyle-1 is targeted to the unbranched glycogen by the HOIP component of LUBAC. I should mention that HOIP does not bind to normally branched glycogen. It binds specifically to unbranched glycogen. And this enables Hoyle-1 to ubiquitinate the C6-hydroxyl group of glucosaccharides. And then our hypothesis was that this marks them for uptake into the lysosomes, where they're hydrolyzed to glucose by lysosomal acid 1,4-glucosidase, or GAA. So, okay, but this is all based on in vitro studies, so it's all very nice biochemistry, but that doesn't mean it's correct. So, so the question is this hypothesis right or wrong? And really the key thing that was missing here was that we'd never shown that unbranched glycogen gets ubiquitinated in cells. So, and these are the two people who I'm now going to present the evidence, which is not yet published, about the ubiquitination of glycogen in cells. So, first of all, there's Lin Chen, who actually is a surgeon in China, who diagnosed a patient with Hoyle-1 deficiency and saved his life by giving him a heart transplant operation. As a result of that, he became extremely interested in the science and read all the papers, including mine, and then wrote to me saying, you know, he'd like to give up being a surgeon for a year and come and work with me, and that his university hospital would give him a fellowship for 1 year. So, so he joined the lab, and actually he'd never done any experiments in the lab. He became very good at making CRISPR knockouts in cell lines, and in fact, nearly all the knockout cell line work I'm going to show you in a moment, he made the cell lines. Unfortunately, he then had to go back to China, so he never was able to do the interesting experiments, and that was left to Val Morfin, a postdoc in the lab. Okay, so the problem is the following, right? If you just, so we would do, by the way, we're doing all of this in HEK293 cells. And the problem is with HEK293 cells that, well, if you have normal the normal cells, there's almost no unbranched glycogen. It's all normally branched, so you can't find the branching. However, if you use a Hoyle-1 knockout cell line, then of course Hoyle-1 can't add the ubiquitin to the unbranched glycogen because it's deficient in Hoyle-1. So what do you do? So, you know, so when Lin came, I said, you know, what we've got to do is to make a cell with a large amount of unbranched glycogen, otherwise we'll never find out anything. So I said, you make a branching enzyme knockout 293 cell, and for good measure, knock out alpha-1,4-glucosidase in lysosomes as well to prevent any of this unbranched glycogen getting degraded. So that's what he did. And now I show you the results of the first experiment that Val did. So actually just want you to lanes 1 to 4 are experiments with normally branched glycogen from the wild type cells, and lanes 6 to 9 are with unbranched glycogen purified from the double knockout cells with no GBE1 or GAA. Okay? And I should mention that the way we purify glycogen is not the way you read in the textbooks. You know, this is native glycogen, and it's washed incredibly extensively with SDS to remove all non-covalently bound proteins from the glycogen. This is a sort of key point. Okay, so look at lane 6 here. So what we've just done is taken the unbranched glycogen we've purified from the cells. We have not done nothing to it. We just stick it into SDS. We run an SDS gel, and then we immunoblot for ubiquitin, and you don't see anything. And the reason you don't see anything is that, of course, as you all know here in an audience like this, that the glycogen has molecular weight of millions, so if you don't do anything to it, it's all stuck at the top of the stacking gel. It never gets in. So what you have to do in order to get any proteins attached covalently into the gel is you have to digest with amylase, human salivary amylase. And when you do this and you repeat the experiment and you blot for ubiquitin, lo and behold, masses of polyubiquitin is attached to the unbranched glycogen, which is released from the glycogen by the amylase digestion. But if you do the same experiment with the wild type, from the wild type cells with normally branched glycogen, you don't really see anything, at least, you know. So, at the very least, there's an enormous increase in the amount of ubiquitin attached covalently to unbranched glycogen. Okay. So the first thing we wanted to know is, well, what's in these ubiquitin chains? What are the linkage types? So this is a simple problem to address by mass spectrometry. Won't go into the technology, but we found that there were 4 major types of ubiquitin oligomer in these ubiquitin chains attached to unbranched glycogen. So there was attached to methionine 1, serine 20. So methionine 1, so we had MET1 linked ubiquitin. The only E3 ligase in biology we know that does that is HOIP. Serine 20, ester linked ubiquitin. One of the very few E3 ligases we know that does this is HOIL1. And our hypothesis said that HOIL1 is involved in bringing, HOIP is involved in bringing HOIL1 to glycogen. But we also saw Lys11 linked ubiquitination, an isopeptide link, and also another isopeptide link, Lys48. So but those were the major ones. Occasionally we see a little bit of ester linked threonine 12, and sometimes a little bit of Lys33 linked, but these are much lower abundant than the ones I'm showing here. Okay, so how do we prove that this MET1 linked ubiquitin, this serine 20, are attached by HOIP and HOIL1, the 2 components of LUBAC? So, fortunately, Lin Chen had made a HOIP knockout 293 cell and made a double knockout of HOIP and GBE1. So we repeated the same experiment with this, and now we're getting to the last sort of 3 months' work. So okay, what Val has done here is he's isolated unbranched glycogen from GBE1 knockout cells, and he's also isolated unbranched glycogen from HOIP GBE1 double knockout cells. Okay, and then we've repeated the same experiment, no treatment where you hardly see anything, digestion with amylase to release the covalently bound ubiquitin, and then we do another trick, which is not to add amylase, but to add hydroxylamine. For those of you who are chemists, hydroxylamines, is a way of cleaving ester bonds specifically. So it cleaves the ester-linked ubiquitin, but it doesn't cleave any other type of ubiquitin linkage. It doesn't cleave MET1 link, which is a peptide bond, and it doesn't cleave isopeptide bonds to lysine residues. Okay, so if we just take a normally unbranched glycogen, then digestion with amylase to degrade all the glycogen releases the unbranched polyubiquitin chains. Hydroxylamine, which really cleaves ester bonds without hydrolyzing any glycogen, that also releases a lot of ubiquitin chains, implying that there's an ester bond linking the glucose in glycogen, or a glucose in glycogen, presumably a C6 hydroxyl group, to glycogen. So that's that. Now look to see what you get with the HOI1 double knock-in. So obviously untreated gives nothing, but look, amylase releases polyubiquitin chains, but hydroxylamine releases nothing. So that means that in the HOIP GB1 double knockout, there's nothing linked to glycogen by an ester bond. All right? So that means that HOIP, which we believe is bringing HOIL1 to the the unbranched glycogen, the HOIL1 is then unable to link the first glucose to ubiquitin, the first ubiquitin to glycogen. So why are we getting any release of a ubiquitin chain with amylase? Because there's no, in this scenario, there is no ubiquitin attached to the unbranched glycogen directly. Well, this means that the amylase is releasing ubiquitin from another protein that's attached covalently to glycogen. And you've probably guessed what the protein is. It takes me back to some of my work 40 years ago, where my student Julie Pitcher discovered that purified preparations of glycogenin were of glycogen synthase were in a one-to-one complex with the protein that turned out to be glycogenin, the protein found by Bill Whelan to be attached covalently to glycogen. And Julie also showed that glycogenin was a UDP glucose requiring glucosyl transferase that catalyzes its own auto-ubiquitination and then adds some further glucoses to that. And Carl Smith, a postdoc in my lab, in collaboration with my colleague Mike Ferguson, the first time we ever used our now obsolete fast atom bombardment mass spectrometer, he found that this glucose, the C1 hydroxy of glucose, is attached covalently to tyrosine 195 of glycogenin, which is a sort of an ether, a glycoether link, which is also resistant to hydroxylamine. So now, in the same mass spec analysis that I showed you before, we also found that glycogenin attached covalently to glycogen is also ubiquitilated, and it's ubiquitilated. We found two ubiquitination sites. They're the last two lysines from the C terminus of ubiquitin, which ends at residue lysine 342 and lysine 344 are ubiquitilated in unbranched glycogen, but not in normally branched glycogen. And Peter Roach, another great glycogen researcher, had discovered that there were in humans, two glycogenin genes, GYG1 and GYG2. I've just shown you GYG1, but in 293 cells, there were actually much lower levels of GYG2, but last week, very clear result, when we scaled up the prep, we could see ubiquitination of GYG2 as well. Also, the penultimate lysine from the C-terminus. I've sort of shown in bold are the residues that are identical in the C-terminal domain of GYG1 and GYG2. And as Elton will tell you later in his talk, it's this C-terminal 30 residues or so of glycogenin that binds to glycogen synthase, as Julie Pitcher had sort of shown this complex in my lab, nearly 40 years ago. Okay, so that's where we are at the moment. Still got some way to go, but you'll see that in a much more elegant way later on. David Commander will show you a very beautiful way method he's discovered to show more clearly the direct attachment of ubiquitin to glycogen, which sort of shows this by an independent method. So, okay, final point before I stop. So we think MET1 is introduced by Hoyp, the serine 20 by Hoyle 1. What is ubiquitinating lysine 11 and lysine 48? And what is ubiquitinating the lysine residues on glycogenin? Because it ain't Hoyp and it ain't Hoyle. They don't do that. And I can see Matthew looking at me intently, and he obviously knows what my guess is, what it might be. It's an E3 ligase called malin. This is sheer speculation at the moment, although we've just made a malin GBE1 double knockout 293 cells to test this hypothesis. So hopefully we'll have an idea shortly. But malin is an E3 ligase, and the loss of malin E3 ligase activity, or the protein, or Lafforin, causes Lafora disease. These proteins have been claimed to interact with each other, and it's been also suggested that Lafforin may bring malin to unbranched glycogen, and another suggestion made by Peter Roach, in a similar way to we think Hoyp is bringing Hoyle 1 to unbranched glycogen. So this is sheer speculation, but perhaps malin is at least one of the other E3 ligases ubiquitinating the lysine residues that are attached covalently, directly or indirectly via glycogenin to unbranched glycogen. And that's the end of my talk. It's just a picture of our building, mainly to point out to you that my office is here. Thank you. Thank you very much indeed.
Thank you very much. We're a little bit out of time, but maybe there could be one question while our next speaker sets up.
Elton.
Have you checked for interaction with glycogenins? Ubiquitinated forms of glycogenin, do they still bind yet?
No, we haven't done that. I'm going to tell you something that's very strange, though. Right, and always in the top 10 hits in the mass spec, when we do the mass spectrometry, well, the top hit is always keratin, as in, coming from people's keratin in the air and people's fingers. because we're adding amylase, that's there. And it turns out that human saliva has other proteins called cytostatins that are obviously contaminating our salivary amylase. We see them if we just take salivary amylase purified and do a mass spec on it. You see all these. So those are irrelevant proteins. The only other two proteins in the top 10 are ubiquitin, glycogen 1, and for some reason I don't understand glycogen synthase. Because obviously glycogen synthase binds to glycogen and it binds to glycogenin. But as far as we know, they binds non-covalently and should have been eliminated by washing away extensively, the unbranched glycogen. So this would be very heretical to suggest that somehow there's a bit of the glycogen synthase somehow gets attached in an unknown way covalently to glycogen synthase, but we've tried to get rid of this this glycogen synthase attached to SDS-washed glycogen in many ways, and we haven't been able to do it yet. So, okay. Anyway, but we don't know. I didn't really answer your question, but I don't. No, I don't know. But as you know, I phoned you. I phoned you to ask you whether any of these ubiquitination sites are essential for, for the binding to glycogen synthase, and you told me no. So you've answered your own question.
Thank you very much, Philip. So it's my pleasure to introduce our next speaker, who is actually a trainee in Joan's lab. So Jordi Duran will speak next. Thank you.
Thank you. So yes, hello everybody. It's hard to speak after Philip Cohen. The contrast is going to be sharp. But anyway, I'll do my best. And I will try to explain today and to show you how in Joan's lab, we challenge a number of dogmas about brain glycogen and about, in particular, the cell-specific roles of brain glycogen in both in physiology and in pathologic conditions. So, as you all know, Joan had always dedicated his life to study glycogen, but in particular liver glycogen and at some extent muscle glycogen. But he became interested in brain glycogen at let's say, the last years of his career. And at that point, what was known about brain glycogen is that you don't have much glycogen in the brain. So people thought it was not very much important, the role of glycogen in the brain, compared to other tissues like the liver or the muscle. And also, the idea was that this glycogen, this brain glycogen, is accumulated only in astrocytes and not in neurons or other cell types in the brain. And Joan, as you probably know, became interested in brain glycogen because of this ultra-rare disease, Lafora disease, that affects very few people in the world, like 1 in every million people is the prevalence, more or less. And this is a very dramatic and severe disease that affects children. It starts as a progressive myoclonus epilepsy, but then it progresses very rapidly with severe neurodegeneration and all sorts of neurologic problems derived from this neurodegeneration, and sadly, death 5 to 10 years after the onset. So and of course the name of the disease comes from, because it was described for the first time at the beginning of last century by Dr. Gonzalo Rodriguez Lafora, a disciple of Ramon y Cajal. So the hallmark of the disease is the accumulation of these polyglucosan bodies, these so-called Lafora bodies, these PAS-positive aggregates that we can see in the brains of human patients and mouse models of the disease. And this, again, this glycogen is not normal glycogen, but poorly branched glycogen that is not only poorly branched, but also phosphorylated and probably ubiquitinated. And so it aggregates in the form of these polyglucosan bodies. So the disease is caused by mutations in two genes, laforin or malin, that were already mentioned by Philip Cohen. One is a phosphatase with a carbohydrate binding motif. The other one is a ubiquitin ligase, as Philip mentioned. So we still don't know exactly how these two proteins prevent the accumulation of these aggregates, but the idea is that, as Philip Cohen mentioned, that laforin probably binds to glycogen and recruits malin, that probably ubiquitinates some proteins, maybe glycogenin, maybe other proteins in glycogen metabolism, and this prevents accumulation of these aggregates. So, well, when we started working on the disease in Joan's lab, it was not clear whether this glycogen that was accumulating in the brain was indeed underlying neurodegeneration and all the pathological traits of the disease. So to study that, some years ago, what we did is that we generated a mouse model of the disease, the malin knockout, and we crossed it, we combined it with a glycogen synthesis-specific knockout. We used a conditional knockout that we can, and we used nestin-CRE to delete glycogen synthesis specifically in the brain. So as expected, of course, these animals, the malin knockouts accumulated these PAS-positive aggregates that we described before. And of course, in the double knockout, since they cannot synthesize glycogen, these aggregates were not present anymore. And well, to make a long story short, what we demonstrated with this double knockout back in 2014 is that, yes, indeed, if we prevent the accumulation of glycogen, we prevent all the pathologic traits of the disease, including neuroinflammation and neurodegeneration, the epileptic phenotype. We did all sorts of behavioral tests, electrophysiological measurements, all of them confirming that glycogen accumulation underlies pathophysiology of the disease, right? So the idea is this. As more or less as Philip Cohen was saying, in poorly branched glycogen is probably generated as a side product of the normal glycogen turnover. Malin and laforin, through a not completely known mechanism, prevent the accumulation of this aberrant glycogen. So if there is mutations in malin or laforin, this poorly branched glycogen accumulates in the form of laphora bodies, and this induces toxicity, epilepsy, neuroinflammation, neurodegeneration, etc. So I forgot to mention that when we started working on the disease, it was the dogma was that these laphora bodies accumulate in neurons, only specifically in neurons. And this was a bit puzzling because, as I have also told you, that in normal conditions, glycogen in the brain is stored in astrocytes. So we don't understand why then in laphora, glycogen accumulates in neurons, right? That was not, matching, right? And actually, already at the first description of the malin knockout that we did in 2011 at the mouse model, we already saw that there are laphora bodies also in astrocytes. We already described that. But we saw that as a curiosity. We didn't pay much attention to it. We didn't see the importance at that time of these astrocytic laphora bodies. We just mentioned it as a, we have already also seen some aggregates in astrocytes, but we didn't pay enough attention to it. And it took us maybe 7 years to really realize that these astrocytic laphora bodies might be important. And actually, we also consider this possibility because if this is glycogen synthesis immunostaining, all these brown aggregates are again laphora bodies, but if you look at the control brain of an aged animal, you also see some glycogen aggregates. This has been known for decades. This is the so-called corpora amylaceae, glycogen aggregates, and these corpora amylaceae are known to accumulate in astrocytes. And these, of course, are very similar to the ones that accumulate in laphora disease, at least to some of the aggregates that accumulate in laphora disease, right? So we started thinking, okay, how important are these astrocytic laphora bodies? So this is the article that we published in 2018, in which we described that, yes, indeed, in the brains of the malin knockouts, these laphora bodies accumulate not only in neurons, but also in astrocytes. So and actually in some regions in the brain, the number of astrocytic laphora bodies greatly outnumber the number of neuronal laphora bodies. And actually, I would say that 95% of the laphora bodies that accumulate in the brain of these models are astrocytic and not neuronal. So we called the astrocytic ones CAL for corpora amylasia-like, because they are very similar to corpora amylasia. We called the ones that in neurons NLB for neuronal laforobodies. You can see that they're actually different. The ones in neurons, we only have one big round aggregate close to the neuronal nucleus, and in astrocytes we have more different in shape and in size in the same astrocyte. So this, yes, contradicted the dogma that laforobodies are only present in neurons. Actually, the lab of Pascual Sanz also reached the same conclusion more or less at the same time, and I think he's going to talk later about the role of these astrocytic laforobodies. But then we said, yes, okay, there are also laforobodies in astrocytes, but are these astrocytic laforobodies important? Because, of course, if there is neurodegeneration, maybe these astrocytic laforobodies are not important, and the ones that actually play a role in the disease are the neural ones. So to answer that question, we did again the same strategy, but in this case, knocking out glycogen synthase only in astrocytes to generate this double knockout in which this is a model of the disease, but cannot synthesize glycogen specifically in astrocytes, right? So, as expected, if we see the brain of the myelin knockout, we have both NLBs and CALs in several regions of the brain. If we have the myelin knockout and we knock out glycogen synthase only in astrocytes, of course, we still have the neuronal laforobodies, but CALs are not present anymore. And in some regions of the brain, you have more neuronal laforobodies, like in the cortex, but in other regions, you have more astrocytic laforobodies, like in the cerebellum. So these mostly disappear. These stay, right? So we have a model now with neuronal laforobodies, but not astrocytic laforobodies. So what is the consequence of preventing the accumulation of astrocytic laforobodies? So we saw that neuroinflammation was completely prevented as in the full knockout. So this is GFAP immunostaining, IBA1 immunostaining showing neuroinflammation, also neuroinflammation markers. We saw that neuroinflammation was prevented, but not the epileptic phenotype. So the epileptic phenotype of the disease stayed more or less the same. So the conclusion of this is that, yes, indeed, laforobodies accumulate in astrocytes, also in astrocytes and in neurons, and that this excessive glycogen in astrocytes underlies neuroinflammation, but not epilepsy. So in this paper, we, in a way, divided the disease in two different diseases. What that comes from the accumulation of astrocytic laforobodies, which underlies neuroinflammation, and we also saw that this underlies metabolic alterations, autophagy impairment that we see in these models. But the epileptic phenotype was not due to this astrocytic laforobodies because it was still present in our double knockouts. So we hypothesized, of course, that this epileptic phenotype must come from the accumulation of laforobodies in neurons, right? So the question was then, in which neurons? And this is what we are trying to understand now in my current lab. And so we focused our attention on parvalbumin interneurons. First, because these were neurons, in the first description that we did of the myelin knockout model in 2011, we showed that these parvalbumin interneurons accumulate laforobodies. This doesn't mean that these are the only ones that accumulate laforobodies, but we saw that at least these neurons were accumulating laforobodies, and also that they were progressively lost in the progression of the disease. So these neurons were in 11-month-old animals were disappearing almost by half. And it makes sense that if you have lost parvalbumin interneurons, which are inhibitory, you have an hyperexcitatory phenotype that is coherent with epilepsy, right? So what we have done in the lab is that we have generated a mouse model in which in this case, this is also a conditional animal, but it's a conditional knock-in in this case, in which we would express a superactive form of glycogen synthase, specifically in parvalbumin interneurons. This is the work of Nuria Moral in the lab. She's in the room, a great PhD student. So what Nuria has seen is that indeed these animals in which we induce the accumulation of glycogen, specifically in these parvalbumin interneurons, have a profound epileptic phenotype. So even at doses of kainate that don't induce any seizure in the control animals, these animals have a high number of seizures. So these animals have indeed an epileptic phenotype. Also, they have an increased excitotoxicity after the induction of this epilepsy, as seen by fluorogate B staining or a gene biphosphoric immunostaining. And also not only the epileptic phenotype, but also other, and Nuria has shown other other neurological impairments in this animal, including motor coordination problems, memory problems, anxiety-related changes in several of these behavioral tests. So, yeah, indeed, this confirms that the accumulation of glycogen in neurons might underlie epilepsy in Lafora disease. These neurons could be basically parabolic interneurons, and also this accumulation of glycogen in these parabolic interneurons would underlie not only epilepsy, but also ataxia and cognitive impairment, and maybe other neurological alterations that are coherent with the disease of the patients. So, yeah, conclusion so far, glycogen accumulation in neurons and in astrocytes underlies the pathology of the disease. And of course, this way and of course this was confirmed by many other groups around the world, like they confirmed this role of glycogen in the disease. So we altogether identified that preventing this accumulation, of course, represents a therapeutic approach for the disease. And actually, I'm not going to talk about this, but in my current lab, we are trying to generate treatments for the disease based on preventing the accumulation of these Lafora bodies. For instance, nanoparticle-based treatments for the disease. But why is this important? Not only because we identified the reason of the disease and the pathophysiological mechanisms, and this way we found possible treatments, but also because we have learned a lot from Lafora disease. And better than William Harvey, let's have Joan himself tell us that. And for that, let me put you 30 seconds of this documentary that we produced to disseminate, like a couple of years ago, to disseminate on the importance of biomedical research to find cures for rare diseases. Let's hear Joan. And this would be.
La malaltia la va descriure el doctor Lafora. A començaments del segle XX han hagut de passar més de 100 anys perquè poguéssim arribar a entendre quines eren les bases moleculars, quin era el defecte que realment causava aquesta malaltia tan terrible. Una altra característica interessant de l'estudi de la malaltia de Lafora és que les solucions que es proposen poden servir també per curar i per trobar tractament per a altres malalties relacionades. I a més també d'incrementar el nostre coneixement bàsic sobre els mecanismes cel·lulars. És a dir, és clar que l'estudi de les malalties minoritàries no solament té un interès específic per aquella malaltia en qüestió, sinó que transcendeix molt més enllà i ens incrementa el nostre coneixement sobre les bases moleculars de la vida i pot arribar a trobar solucions per a altres malalties que inicialment no s'esperaven.
I do miss him. So yes, as Joan was saying, thanks to Lafora disease, or thanks to these studies that we did on Lafora disease, we have identified pathologic mechanism that could be applied to other diseases, because Lafora disease is not the only condition in which glycogen accumulates in the central nervous system. There are several other conditions, including several other rare diseases like Pompe, APBD, ALS, but also much more common diseases like Alzheimer's disease or Parkinson's, diabetic retinopathy, and neuropathy, in which the accumulation of glycogen in the central nervous system has been described. So what we have learned in Lafora disease might be important for these other conditions, right? And in fact, regarding ALS, we published a couple of years ago, we published that indeed in mouse models of the disease, in mouse models of ALS, glycogen accumulates in the spinal cord. And if we prevent this accumulation of glycogen, we prolong the lifespan of these animals. If we increase the accumulation, we shorten the lifespan, indicating that indeed maybe the accumulation of glycogen might be in the CNS, in the spinal cord, actually in astrocytes. We showed that this accumulation was taking place in astrocytes. And that was associated to the inflammation that is also associated to ALS. So yeah, indicating that this astrocytic accumulation of glycogen might also play a role in ALS. And regarding Alzheimer's disease, we have in my lab now a project founded by the Pascual Maragall Foundation to understand the role of Alzheimer's in the metabolic reprogramming of astrocytes at the early stages of the disease. And for instance, several other groups are working on the role of glycogen in Alzheimer's disease. So what we learned in LaFora might be important for other neurodegenerative conditions, including Alzheimer's, which is the most prevalent neurodegenerative condition. But also, I've told you that when we started working on the disease, the role of brain glycogen was in a way overlooked, probably because of the low concentration and the low presence of glycogen in the brain. But we, over the years in Joan's lab and also in my lab now, we have published a number of papers demonstrating the importance of this brain glycogen. So to make a very long story short, what it has shown is that brain glycogen plays an important role in memory formation, synaptic plasticity, and even in the resistance to epileptic seizures. And I have also told you that the dogma was that this brain glycogen only accumulates in astrocytes and not in neurons. But this is another dogma that the lab of Joan, in a way, challenged, because we demonstrated first in vitro, then in vivo, that neurons also have an active glycogen metabolism, and that, for instance, if we knock out glycogen synthase in KMK2 neurons, these animals have also problems. This impairs memory formation and learning-dependent synaptic plasticity. So indicating that this glycogen plays a role in the disease, and that, this glycogen plays a role in the normal functioning of the brain, and that also part of this glycogen is also accumulated in neurons, and the neurons have an active glycogen metabolism, again contradicting one of the dogmas of the field, which is that glycogen is only present in astrocytes. We are continuing these studies in my current lab, and for instance, we are now, also Nuria is also working has also already many results with this animal model in which we have knockout glycogen synthase in parvalbumin interneurons, because we think that LaFora disease is, in a way, a readout of the normal functioning of the normal importance of glycogen in the brain. So we think that the cells that accumulate LaFora bodies in the disease are the cells that have probably a higher turnover of glycogen in normal conditions. So if we see LaFora bodies in parvalbumin interneurons, probably this means that parvalbumin interneurons in normal conditions have a high turnover of glycogen synthesis, and that this is important for their function. And again, I'm not going to show this for the sake of time today, but Nuria has interesting results confirming that glycogen is important for the normal functioning of parvalbumin interneurons in normal conditions, let's say. So, yeah, conclusions. Brain glycogen is important both in physiological and pathological conditions. In pathological conditions, the accumulation in neurons and astrocytes underlies LaFora disease. So we identified a way to treat the disease by preventing this accumulation, and we identified that this accumulation might also play a role in other conditions such as ALS, Alzheimer's, or other conditions, and also that this brain glycogen that had been overlooked for many years, now it's clear that it plays an important role in memory formation, synaptic plasticity, and resistance to seizures, and also that neurons have an active glycogen metabolism. And for instance, we will see this afternoon the talk by Mark Claret, where he will show the importance of brain glycogen in POM-C neurons in the control of feeding behavior. So let me finish with another dogma that the lab of Joan contradicted, which is that when we draw glycogen, we always put in the middle glycogenin. This was supposed to be essential for the synthesis of glycogen. It was believed that without the priming role of glycogenin, glycogen synthase would not be able to synthesize glycogen. So to corroborate that, some years ago we also generated a glycogenin knockout, a mouse model. So mice only have one glycogenin, differently from humans that have glycogenin 1 and glycogenin 2. Mice only have glycogenin 1 or one glycogenin, let's say. So we knock out that glycogenin some years ago, and surprisingly, what we saw is that these animals are still able to synthesize glycogen. In fact, they accumulated much more glycogen in the muscle and in the heart. the same amount of glycogen, more or less, in the brain and in the liver. So you can synthesize glycogen without glycogenin, and this was, again, a dogma that the lab of Joan challenged. Last year, we also published that glycogenin is completely dispensable for the glycogen metabolism in the liver. So if we knock out glycogenin, we have normal liver glycogen metabolism, and glucose homeostasis is normal in these animals. So yeah, again, a dogma that was completely challenged by the results that originated from Joan's lab. So yeah, that's all. A big part of what I've shown was done here at Joan's lab at IRB Barcelona by many other people, but these were the last participants of the lab. This is my lab at IQS. We are more people now, and actually more about to come, so we should update the picture. And I want to thank, of course, the collaborators and the fund. And let me finish by saying that we, together with Matt Gentry, published this in memoriam article to Joan a few months ago. So yeah, let me say that everything I know about glycogen, I owe it to Joan. So as the last words of the article say, gracias por Joan. Thank you.
I think we have time for a couple of questions.
Obviously, as you know, the knockout of glycogen in 1 results in a sort of similar disease to the knockout of Hoyle-1. You get cardiomyopathy and heart failure. So, so the glycogen is not normal in.
No, but yes, but in the patients you have in the patient, human patients, you have that, but not in the mouse. So in the mouse, the glycogen that the mouse accumulates is more or less, well, I would say it's normal, and everything that we have analyzed from glycogen in the mouse model is normal.
Even in the heart.
Even in the heart, yeah. That's so the difference probably is that humans have the 2 isoforms. So maybe when you have one, then the other tries to compensate and messes up with glycogen metabolism in the heart.
I've just made a glycogen 1 GB1 double knockout, and I'm anxious to know whether there's, or whether in a glycogen 1 knockout, whether, for example, you have covalently attached ubiquitin, because that would imply that it's not normally branched.
So we didn't see any aggregates I mean, we saw normal levels of glycogen in the hearts of the glycogen in knockout, and we don't see any aggregates. So we haven't properly analyzed. So we have analyzed muscle glycogen and liver glycogen, the structure, the shape, and everything. We probably didn't have paid so much attention to heart glycogen, but yeah, I would say that most likely in our animal, the amount of glycogen in the heart was actually increased, but not aggregated.
I mean, humans get cardiomyopathy and
Yeah,
heart
sure.
failure, even though they have glycogen 2 still there.
Exactly.
So
but
it's
that's
not compensating.
No, I'm not saying that it is compensated. What I'm saying is maybe precisely because there is another glycogen then the glycogen that is generated in these hearts is different. In the mouse, where you only have one glycogenin, if you knock it out, you don't have another glycogenin to try to compensate and do a bad job trying to compensate. I don't know. That's my only explanation, because again, in our animals, complete devoid of glycogenin, the heart glycogen seem to be, well, at least not very different from the control animals. But again, this is a bad model in the sense that mice only have one glycogen, and humans we have 2. So you cannot directly compare what happens in mice to what happens in humans. What you can say with our model is that glycogen synthase can synthesize glycogen without glycogen, right? So that's as far as you can go. How it compares to the human diseases, that's not a good model to compare, because again, we only have one glycogen in the mouse.
Thank you very much.
Thank you.
I think to stay on time, we'll invite the next speaker. So it's my pleasure to now introduce Pascual Sanz, who has been a longtime collaborator of Joan's.
That's right. Well, first of all, I would like to thank the organizers for inviting me here in this magnificent event. I would like to show you how I got involved in Joan's research. And when in 2006, I was happily working in yeast, trying to figure out the regulation of one of the main energy regulators, that is AMPK. We work with the yeast ortholog, that is SNF1. So I was collaborating with Graham Hardy in Dundee University, Marian Carlson, and Matthew Bolin, trying to figure out different regulators of this protein kinase, specifically with the role of different phosphatases. Then, one day in a meeting, I was approached by these two guys, Joan Guinovart and Santiago Rodriguez de Cordova, and tell me, why don't you join us in an effort to try to understand the molecular basis of Lafora disease? And I said, what Lafora disease is? Because I didn't know this disease at all. Lafora disease, as you have seen previously, was described for the first time by the Spanish neurologist Gonzalo Rodriguez Lafora, and he drew in the necropsias of the patient that he had the accumulation of these granules inside the neurons. So he called this disease a disease that was related to the accumulation of polyglucosans in the brain. The disease is terrible. It's an ultra-rare disease affecting less than 4 patients in 1 million people. It's a particular type of progressive myoclonus epilepsy that develops myoclonus, epileptic seizures, and neurodegeneration. It starts when the patient is in the childhood or early adolescence, and unfortunately it has a life expectancy around 10 years. There is no treatment yet, and this is the reason that Joan and myself wanted to work in this disease, try to provide the basic knowledge to implement different therapeutic approaches. The disease is an autosomal recessive disease with 2 genes that are mutated. The one is EPM2E that codes for the laforin, and the EPM2B that codes for an E3 ubiquitin ligase. So you know that how persuasive Joan was when he tries to convince you that you have to work with him. And he told me that we had to explore the different phases that glycogen had in the brain. And I remember in a seminar that he gave in our institute that the glycogen should have 3 different phases: the good phase, the bad phase, and the ugly phase. And that's what convinced me to join the group and then start working together. As a researcher working in yeast, we started with using what we know, that it was the all yeast 2 hybrid system. And by using this technique, we were able to define that laforin and malin was making a complex. But more importantly, we saw that laforin interacted with one of the regulatory subunits of PP1 phosphatase, that is R5-PTG. And that laforin and malin could really ubiquitinate R5-PTG and then target it for degradation. So our working hypothesis at that time is that in the absence of laforin or malin, you get a fully activated R5-PTG PP1 complex that dephosphorylates glycogen synthase, and that makes glycogen synthase fully active. So you get more glycogen accumulation. That was the beginning, and since then, our group has been studying the molecular basis of Lafora disease. We described that there was a proteostasis dysfunction with both proteasomal and autophagy defects in this disease. There was also a mitochondrial dysfunction that gives rise to an increase in the amount of ROS in the cells, and that gives a kind of oxidative stress. And we also published that the glutamatergic transmission was affected because the transporters that were taking care of taking up glutamate were not present at the level of the surface of the astrocytes. And finally, we gave different evidences that neuroinflammation should be treated as a new trait in Lafora disease. But today I want to convince you that astrocytes really make a role in the pathophysiology of Lafora disease. As I said, Lafora draw these big black spots inside the neurons. So we wanted to revisit this observation and did what you have already seen in the talk from Jordi, that you get this PAS staining. But PAS staining is something chemical, and you cannot really address which cells are accumulating this structure. So we, with the help of Matthew Gentry, we developed a technique of immunofluorescence using a specific antibody that recognizes glycogen and polyglucosans. And using this technique, we got very surprised because we saw the accumulation of glycogen inside some neurons, but most of the staining was outside these neurons. And what we saw is that they are mainly inside the astrocytes. You can see here the accumulation of these red particles inside specific cells that are positive for, in this case, for glutamine synthase, in this case, for glial fibrillary acidic protein, a specific marker for astrocytes. We also saw the same for microglia. So in a word, glia are the main accumulators of polyglucosan bodies. We described this thing as Jordi said, and it was not back-to-back, but more or less at the same time. We published these results, and as I said, with the help of Joan Guinovart. But the question we wanted to answer is, why LD astrocytes accumulate more glycogen than the regular ones? Obviously, we cannot do this thing using mouse, using systems, unless you have an in vitro system that can really help you to understand the different actors in this possible reaction. So what we did was set up an in vitro assay by purifying astrocytes from either Laforin or Malin knockout mice. And under specific conditions, you can see here that these astrocytes that are pure, pure cultures of these cells contain these polyglucosan accumulations that are absent in the control astrocytes. So now that we have this system working, then is when we can really study which component is the one that is affected in this astrocyte. The first one was to check if glycogen synthase was affected. So we did the activity, we measured the activity of the glycogen synthase, and it was similar as wild type. We also checked by Western blotting the different forms of glycogen synthase, and in all the cases, we didn't see any difference at all. Then we checked for the other part of the equation, that is the glycogen phosphorylase, and we saw again the same. We didn't see any change in the activity or any difference in the amount of the enzymes that are present in our astrocyte cultures. So with these negative results, we thought that it was the best time for checking and target metabolomics of this astrocyte and check which metabolites were affected in the laforin or malin knockout versus the control. And we saw an accumulation of different metabolites like glucoxyphosphate, fructose 1,6-D-phosphate, or gluconate, all connected to the upper part of the glycolytic pathway. That was telling us that probably this astrocyte had an increase in the amount of glucose that was taken up on them. And this was really what we saw. When we checked for the glucose uptake in this astrocyte, we saw an increase in the amount of glucose that these cells were able to capture. Then the next question was why glucose uptake is so enhanced in LD astrocytes. And by doing an untargeted proteomic approach of proteins that are ubiquitinated by malin, but not in a malin inactive form, we described that there was one protein that is a nucleotide exchange factor that was, in a way, being ubiquitinated by malin. So in the absence of malin, Prex is fully active, and that makes that the small GTPase, RAG1, is fully activated, and the activation of these GTPase activates the transporter, in the case of the astrocyte, the GLUT1 transporter. And that was our, let's say, observation that the glucose could really enter the cells because the absence of malin. So as a conclusion of what I have seen, or what I have shown you before, is that in the absence of laforin and malin, we have two different branches. In a way, Prex is fully activated, so you get more glucose inside the cell, and at the same time, the absence of this negative regulator of glycogen synthase makes glycogen synthase more active, and that results in an accumulation of glycogen. All right, what I have shown you is what happens with the astrocyte model after 2 weeks of maturation. We wanted to extend this maturation to see was a negative effect, or if it produced, the death of the astrocytes. And so we maintained the cells for longer, and you can see here that when you increase the time of maturation, you get these forms of polyglucosans that are larger than the ones that you get at 2 weeks. What is even more interesting is that when we apply super resolution microscopy to these particular aggregates, we saw that they are formed by small, very tiny, small aggregates that get together and give rise to these polyglucosan bodies. And surprisingly, this is what you can already observe in vivo preparation of the mice. So in both cases, you get the aggregation of these particles making these polyglucosans. And these polyglucosans are decorated by the same time of the same kind of proteins. For example, the P62, that is a regulator or an effector of the autophagy pathway, and also you can decorate these polyglucosan bodies with ubiquitin. And we know that ubiquitin is necessary there because it's the first signal that the cell has to try to release these compounds from inside the cell. So I want to share with you our hypothesis that by working with the in vitro model, we can really reach conclusions of what is really happening in the in vivo model. This aggregated structure of the polyglucosan bodies remind us an hypothesis that the group of Matthew Gentry published some years ago, saying that this aggregation of the polyglucosan could be the result of first the accumulation of small particles of unbranched polyglucosans that got together, and then finally got this big aggregate. And you have already heard this morning that these aggregates are toxic. So we wanted to check what was the toxicity that was related to the accumulation of these polyglucosans. So to answer this question, we did a kind of unbiased omic approach by doing an RNA-seq of the brain of mice that had 16 months of age. We chose this age because at that time, the disease in the mice is already present and is quite severe. So by doing this kind of technology and using both brains from Malin and Lafferty knockout, because we wanted only to focus on those RNAs that were similar in both cases. So we saw that most of the targets that we described were related to upregulated genes, and most of them corresponded to genes related to the immune system, inflammatory response, and phagocytosis. And you can see here, we choose some of these targets to validate the RNA-seq, and I want you to check that in some of the cases, in most of them I will say, there is an increase in the expression of these markers with time. So that means that the disease is really a progressive disease with the accumulation of these inflammatory markers. And secondly, when you check who is the cells that are really expressing these markers, you get that they are mainly produced by astrocytes and microglia. So the conclusion of this experiment is that we saw an increase in the amount of RNA corresponding to inflammatory mediators, chemokines, cytokines, and also complement proteins. There was an increase in the expression of these mediators. And the next question was, what is the main pathways that are related to, to the expression or to the effect of this toxicity? To answer this question, we use a technique to check for the upstream regulator of this whole bunch of different mediators, a technique that is a kind of, let's say, bioinformatic technique, that is the Glycogen Ingenuity pathway analysis, that in a way suggests the different upstream regulator for a whole bunch of different RNAs that you may have. This is the list that we got from the analysis, and the first one that was more prominent was the TNF-alpha signaling pathway. The TNF-alpha is, I don't have time to explain all the components of this pathway, but I want just to stress you that some specific components of the pathway, the ones that we analyzed, were upregulated in the in vivo brains. And we also saw that astrocytes and microglia were the main actors in this expression when we performed the co-immunofluorescence analysis of using markers of these cells and also markers of the inflammatory mediators that we were analyzing. The other component that was very important was the IL-6 signaling pathway, and again, we analyzed different components of the pathway, and again, we saw an increase in the amount of the signal for this pathway in preparation from the brain of Malin knockout mice, and in this case, it was astrocytes, the one that were upregulated. It has been described in the literature that when TNF and IL-6 pathways are increased, the permeability of the blood-brain barrier can be disturbed. In a way, it loses the permeability and allows the entrance of different components of the blood system, either proteins or either cells. So we checked first for the proteins, and we didn't see the accumulation of albumin in the brain parenchyma. We didn't see either the accumulation of granulocytes or macrophages, but we saw an accumulation of T lymphocytes, specifically the one that are CD8 that are related to the cytotoxic T lymphocytes. In the same way, if the permeability of the blood-brain barrier is affected, one should expect that some markers released by different components of the brain could really reach the blood. And that is what really we observe. We observe an accumulation of CL-CL10 and S100 beta in the blood from these mice that we were analyzing. I want to point out that these two markers are mainly produced by astrocytes. So again, suggesting that astrocytes are making a main role in the pathophysiology of the disease. So the working hypothesis we have so far is that by accumulating glycogen inside the astrocytes and microglia, you get the release of some components that are pro-inflammatory, but at the same time, they release components that are chemoattractive for different peripheral immune cells. These peripheral immune cells will infiltrate the brain parenchyma that will aggravate the neuroinflammation that is present in the brain, and in a way that will produce the neurodegeneration of the brains that are around. The neurodegeneration will release different damage-associated proteins that close the circle, giving a positive feedback of this situation. The next question was, okay, we have set up an assay that can provide you some clues about the role of polyglucosans in an in vitro model. Can we prevent the glycogen accumulation in this system? And the answer is yes. After many trials with negative results, we end up with two compounds, that is ICAR and metformin, that can really diminish quite strongly the amount of polyglucosans after only 24 hours of administration of these drugs into the culture. The most important thing of this is that for the first time we see some way to eliminate this polyglucosan accumulation. It is surprising that both of them act on AMPK, so probably AMPK is doing a positive role in eliminating these compounds. And what is more important is that when we treated these cells with a combination of any of these compounds and an inhibitor of glycogen phosphorylase, we lost the positive result, suggesting that this glycogen phosphorylase a positive regulator of the degradation of the polyglucosans that are being accumulated inside the astrocytes. So now we are doing a kind of a screening, a multi-screening of different drugs with the idea of using this in vitro system and checking for a decrease in the amount of polyglucosans after only 24 hours of action of the drug. And this is like last week. We have now identified 3 compounds with a very low IC50 that suggests that the activity of these guys are like 100 times more effective than the one that metformin or ICAR had. In the next meetings, I will give you more information about these compounds. With these compounds, what we want to do next is try to see if they are effective, not just in cells, but in mice. So for this reason, we have set up in the lab a kind of a pipeline to check if the benefit effect of different compounds on malign knockout mice. So we get mice when they are 3 months of age. We treat it with different compounds, either in drinking water or by administration by intraperitoneal injection. And after 2 months of administration, we do a battery of different behavioral tests, and then we do the histopathological analysis of them to check how effective the action of these compounds was. We have an experience already in this setting. I have to tell you that So far, we have described 4 different compounds that can really ameliorate the behavioral test that we check with the animals, but only one of those was able to reduce the amount of polyglucosans that were observed in the mice. Unfortunately, we sent an application to both the European Medicine Agency and the Federal Drug Administration for asking for an orphan drug designation for this compound, and we got it. So now, all the clinicians can really use this compound to treat Lafora disease patients. Finally, what I have told you so far is everything about mouse models. We want to go a step forward by using human samples. To do this kind of thing, we decided to use iPS cells, and we constructed an iPS cell that was isogenic, but contained one mutation that is the most prevalent mutation in the EPM2B. We want to differentiate neurons and astrocytes to work with them and then confirm if all the features that we have observed in mouse are really present in these human models. Just as a glimpse of what we are going now, we have been able to differentiate neurons. You can see here the marker of MAP2 and also astrocytes. And the final step is to obtain brain organoids that we have already them, but we have to analyze and check if all the pathophysiology that we have described for the mice are also present in the human samples. All this work couldn't be possible without the help of Chelsea Hope. So I want to really appreciate the work that this patient association has, and give a support to give a kind of help to fight Lafora. And finally, this is the group that has been involved in the work with all the funding agency, but my talk wouldn't be finished without a mention to Joan Guinovart. So I want to really to express my deepest gratitude to Joan for his extraordinary mentorship. For him, I was El Chique de Valencia, that means the boy from Valencia. That is an expression meaning a friendly relationship. So, gracias, Joan. Thank you very much.
You mentioned on 2 occasions that that you saw in the mail-in and the Lafora knockout elevated CXCL10.
That's right.
So CXCL10, of course, is IP10.
Yeah.
Interferon regulated induced protein 10, and so that is made by type 1 interferons. It's not made by the TNF or NF. Well, it's not made by the TNF pathway. So I was just wondering whether you had thought whether anti-type 1 interferons could possibly have an interesting role in treating Lafora disease for that reason.
Well, very nice question. The point is that when we checked for these compounds, we checked in the literature for which were the proteins, the cells that were mostly releasing these compounds to the bloodstream. And always I saw that astrocytes were good component. Also, lymphocytes do the same. So you are right. It's a question of a pathway of interferon that maybe is inside these cells, the one that makes them to produce the compound. We are now having in the side pocket different strategies to combat CXCL10 by using anti-CXCL10 antibodies that are available. But we haven't started yet because when you are using an antibody technology, the point is that these antibodies cannot cross the blood-brain barrier. So it's good for any kind of inflammatory reaction that is in the periphery. But if you want to solve something that is neuroinflammatory, maybe that is not the right way to do it.
Was one of your very potent molecules fenformin?
No, it was metformin.
No, I know.
Yeah.
But fenformin is a much more potent derivative of metformin, and therefore I thought that might be one of your much more potent molecules. I'm trying to tempt you to say what they were.
we were thinking in that molecule indeed, but when we read the literature, they said that when they use fenformin for diabetic patients, they had to withdraw the application of this compound because of hepatic toxicity. So we wanted something.
This is why it's not used, I know, but this only happens in a very small percentage of patients, and for things like Lafora disease, fenformin would be an approved drug.
Yeah, it's already approved. Maybe the metformin. I don't know for the fenformin because you have to go to these trials with the EMA regulators, and if you say that you're going to use one drug that has been withdrawn from the market, probably you don't get a good result of the evaluation.
All right, thank you very much.
You're welcome.
So we had a nice introduction to Chelsea's Hope Lafora Children's Research Fund. So it's my pleasure now to introduce Kit Donahue, who actually trained in one of the scientific co-organizers of this meeting, Matt Gentry. Thank you, Matt. We've forgotten to acknowledge you so far. So she trained in this lab and then went on to do a lot of advocacy and worked with Joan. So she'll give a short talk before our little break.
So thank you for that introduction. She mentioned I'm Dr. Kit Donahue. I'm the Executive and Scientific Director for Chelsea's Hope Lafora Research Fund. I've been in that role for about 4 years now, and whether you all realized it or not, when you came to a glycogen conference, you actually got a pretty good primer on the scientific mechanisms of Lafora disease. So that's not what I'm going to be presenting today. I'm instead going to give you all a little bit more of a perspective from the patient community as you work on elucidating these critical mechanisms for understanding glycogen biology. It has very real impact and life-altering implications for our patients. So to just tell you a little bit and give you a glimpse into the window of Lafora disease, I'm going to play a clip and let a couple of our board members who are also parents of children with Lafora disease describe the impact it's had on their children.
Lafora disease is a glycogen storage disease which causes intractable seizures and early childhood dementia. It is neurodegenerative, so our children go backwards, losing all their abilities to do academics, sports, and live their lives and live out their dreams. They lose their ability to walk, to talk, to feed themselves.
You know, she has fun and fear of missing out. She was very social. She wants to be the kid that she was. You know, she's very scared. And she feels very strongly about finding a cure.
So as you can tell even from those few short seconds, the devastating impact this has on families, children who develop normally until they're in their early teens, and then with the onset of seizures and the debilitating progression of onset of neurodegeneration in a very short period of time. You see 3 different patients here, pictures side by side. Within 5 years apart with each of these pictures. The dramatic change in their behavior, their ability to walk, and their cognitive decline. Lafora disease, as with many rare disease communities, are incredibly dependent on the quality of the science that helps them understand the mechanism in order to identify potential treatments. And so what I would like to share is just a brief history of Lafora as we're working to reach the goal of treating. Our mission at Chelsea's Hope is to improve the lives of those suffering from Lafora disease and to work towards reaching a cure. And so for any rare disease, there's sort of 5 steps towards building up to a cure. Of course, it begins with defining the rare disease, and for Lafora, as you all heard, back in 1911 was when it was first described, these sugar-like aggregations that were responsible, characteristic of this disease. It wasn't until the early 2000s, though, that the genetic drivers EPM2A and EPM2B were identified, and that made Lafora already one of the fortunate rare diseases. Out of more than 7,000 defined rare diseases, only about 30 have been able to identify their genetic drivers, despite predictions that upwards of 90% of rare diseases have some sort of genetic driver that underlies the condition. And thanks to a strong network of very passionate researchers, some of whom had personal connections to Lafora disease, within the decade, they were able to define the function of these genes and the proteins that they coded for, which you already heard so much about this morning, with the ultimate understanding that these 2 genes coded for proteins that regulated the shape of glycogen, and when they lost their function, you started to accumulate glycogen in its aberrant form, forming these Lafora bodies. Now, the final 2 steps are often interchangeable. You go back and forth between developing a better understanding of the disease mechanism and developing interventions. And so this was the state of Lafora disease when Joan entered the scene and started his investigations. And of course, as you heard so eloquently from Jordi, since his passion was looking at glycogen synthase, he began to investigate the impact of Laforan and Mao and their loss of function on glycogen synthase and glycogen synthesis in the cells. And again, as you saw from Jordi's presentation, this active form of glycogen synthase that was discovered within the mouse models for Lafora disease showed very early aggregations within the astrocytes, and that was a very surprising find at the time. It had not been the focus of research. And then as the mice aged, you then found these accumulations in the neurons themselves. And it was the accumulation that they were identifying in the paravalubin positive neurons, again, as Jordi mentioned, that they were able to associate with this correlation, the accumulation of the Lafora bodies in the neurons, and what that led to for a possible explanation, as these are inhibitory neurons, that might be connected to the onset of seizures in Lafora disease. They then went on to establish part of Joan's lab, looking at what would happen and proving definitively this link between this accelerated accumulation of glycogen and neurodegeneration. And so they expressed an active form of muscle glycogen synthase in a mouse model, and very quickly were able to illustrate this overaccumulation of glycogen, and importantly at the same time illustrate the loss of Purkinje cells within the cerebellum, which helped the patient community understand a possible mechanism that would link to why their children were starting to exhibit ataxia and lose a lot of their motor function. And so, once again, as you've heard from several speakers now, this opened the window for a potential therapeutic intervention for Lafora disease and what would happen if you could decrease glycogen synthase activity. And as it's already been illustrated in several talks, it has been shown that if we can regulate glycogen synthase activity and therefore control the synthesis of glycogen, slow its accumulation, we have the potential to intervene in Lafora disease. And not only that, but if we can intervene and slow this glycogen accumulation, it has significant impacts on testing for markers for neuroinflammation and the markers for neurodegeneration. So that hit for the patient community all of these symptoms that they were experiencing every day in their children. They're now seeing the science behind it through Joan's presentations, helping them understand on some level what's happening, and helping them figure out where they can best plug in to support the research and the patient, the full patient community to bring a cure to the clinic. It was at this point that an international collaboration began. We've already heard Joan's passion for collaborations and bringing in scientists and encouraging them in their work to support the patient community. And so Dr. Guinovart's project specifically was working on defining the therapeutic window for intervention in Lafora disease. And many of the other projects happening simultaneously in the other labs were focused on developing platforms that would allow us to target glycogen synthase and glycogen accumulation as two different methods of coming in to create a cure for Lafora disease. And in 2021, this paper was published in Neurobiology. This data was actually presented at the 2019 symposium in Madrid, and that was actually my first interaction in person with Joan after having collaborated in Dr. Gentry's lab. And he gave a very important discussion with families, helping them understand that the science behind this is very solid for reducing the glycogen accumulation by targeting glycogen synthase. But he was very clear with the patient community, you had to have early intervention because their studies clearly showed that while they could knock out muscle glycogen synthase at different points within this mouse model, the level of Lafora bodies that had already been accumulated was maintained. And whatever amount of neurodegeneration, but also neuroinflammation that was being driven by these Lafora bodies would then remain in the patient. And so that sparked a really important discussion, and from that formed the priorities of the patient community, that not only did they want a therapy that was going to reduce the activity of glycogen synthase and reduce accumulation of glycogen, but that some sort of possible enzyme therapy, like ones worked on by Dr. Gentry and Dr. Brewer, could be utilized to help ablate the accumulation of Lafora bodies and help rescue maybe some of those in later stage to allow for a later intervention within Lafora disease. And so as these platforms have been developed, there's just one final one I'm going to share with you, the one that has reached furthest and is now in the clinic. It was an antisense oligonucleotide that I had the privilege of collaborating on in the validation process as part of the Gentry lab with the Menashen labs and Ionis Pharmaceuticals. And this followed exactly the predictions and the pathways for intervention that were identified and supported through the work of Joan, where if you develop this glycogen synthase antisense oligonucleotide, you can knock down its activity by more than 50% in the brain, and you can essentially halt glycogen accumulation. And what we saw very clearly in the mouse models and tests was that you had a decrease in the size of the Lafora bodies and the number of the Lafora bodies that were present compared to controls. And when this data was shown to the patient community, despite running into the typical struggles that many rare diseases come into when it's not determined to be profitable to take to the clinic from a pharmaceutical perspective, the patient community themselves have rallied behind to sponsor and fully fund a safety study that began at UT Southwestern Medical Center in December 2024. And so what I hope I've been able to illustrate to you in these few short minutes is just the incredible impact that your research on the basic science, the basic biology of glycogen can have on the patient community. And like Jordy, I was very struck by the interview that Joan gave as part of the Fighting the Rare documentary. I'm not going to play it for you all again, only to leave you with the final thought of what Joan would encourage us all to do to continue to work on developing good science and making sure that we are helping to translate that science into impact for these very vulnerable patient communities. So with that, I would like to thank the hosts for inviting me to talk on behalf of Chelsea's Hope and thank all of our scientists and clinicians and our advisory board members who help us work towards reaching a cure for Lafora disease. Thank you all.
I want to thank all the speakers for such a nice tribute to Joan. I hope he's up there listening to all of this. So we're running a tiny bit behind schedule, so maybe we'll take a 20-minute break and come back here in 20 minutes. Thank you.
Hello? Okay. Hello? Start. Okay. Okay, we are starting soon, so maybe. I'm gonna go ahead, yes. Good afternoon, everyone. Welcome to the midday session. My name is Ana Victoria Lechuga. I'm a junior group leader at the IUBMB Barcelona, and I will be chairing this session. So this session will focus on different aspects of glycogen metabolism, ranging from metabolic adaptation and exercise physiology to molecular regulation across systems. We have an excellent lineup of speakers, and I would like to kindly ask you both, speaker and audience, to help us to stay on time so we can have a little bit of discussion. So 25 minutes talk plus 5 minute Q and answer. And I will stand like 2 minutes before the 25 minutes. Yes, so let's start. It's my pleasure to introduce our first speaker, Alan Saltier from the University of California, San Diego, who will talk about glycogen and adapting to metabolic stress. So Alan, the floor is yours.
Can you get this? Thank you very much. You know, the 25-minute, you know, that doesn't account for me, too. That can't be right. Okay. Anyway, it's really an honor to be here and a pleasure. Thank you very much to the organizers for inviting me. It's a great opportunity for this tribute to Joan, who's really a giant in our field. So it's really great to be here. And I also just want to take a second to point out that we've lost other giants in our field over the last few years, and specifically Patricia Cohen and Peter Roach and John Lawrence are the three people that I think about quite a lot in our field. And John Lawrence especially was a close friend of mine, so I really do miss these guys. And they all had a major impact in our understanding of glycogen and glycogen metabolism. So I'm going to just start off talking a little bit about, just to put this in context, about some of the things we've been doing in my lab over the last few years since I started out just a couple of years ago in this field. My lab has been interested in trying to understand how metabolically active cells, especially fat and liver cells, decide whether or not to take up and store energy or burn it or release it for other cells to burn in response to hormonal or nutritional cues that arise in fasting and feeding. And then kind of to layer on top of that question, we're also interested in how these pathways respond to metabolic stress. And when I say metabolic stress, I mean both anabolic stress, like obesity, and catabolic stress, like starvation or cold. So we've been working on this, trying to dissect and understand the signaling pathways from these hormones that control these processes, worked on a lot of different aspects of this process, including the regulation of glycogen. And I want to point out that, and I'm not going to talk about this today, but our studies on really trying to understand the pathways that control the metabolic processes in cells, and glycogen and triglyceride metabolism and other things, remains incomplete. I mean, there are still big gaps in our understanding of the signaling pathways downstream from the insulin receptor and the beta receptor. So we don't ever understand everything. And Kay, I think, can attribute to that, and Dario and other people who work in this field know that we don't really know everything. For the young people in the audience, I just want to emphasize there's still a lot to learn about these pathways. Now, we really started in the glycogen area trying to understand the regulation of glycogen metabolism by insulin. And I'm not going to talk about this today, but we also pivoted in the last couple of years to trying to think about not just how glycogen metabolism is regulated, but how glycogen itself controls other processes in the cell. So glycogen, it turns out, is a cue, especially in fat cells and liver cells,
to the overall energy status of the cell. It serves as kind of a signaling molecule in its own right. Now, this first came up to us in fat cells, and I'm not going to talk today at all about fat cells. I'm going to focus exclusively on liver. But we published this paper a couple of years ago. This is work from a talented Israeli postdoc in my lab, Omer Keenan. And what Omer found was that fat cells really are interesting when it comes to glycogen. Now, if you just look at a white fat cell, you can barely detect glycogen. So it doesn't look like a player in glycogen metabolism. It doesn't really use glycogen as a way to store energy. And in fact, John Lawrence and I used to talk about this all the time. The fact is that fat cells are exquisitely sensitive to hormones that regulate glycogen. In vitro, you can really see a big change in the accumulation of glycogen, but in vivo, they don't really do anything. And Omer found out that with prolonged sympathetic activation of white fat cells, you see a big accumulation of a cassette of enzymes and proteins involved in glycogen metabolism. So that after 6 or 7 days of a beta agonist in vivo and in vitro, you start to see accumulation of glycogen and upregulation of all the genes that are associated with glycogen metabolism. Phosphorylase, synthase, the scaffolding proteins that I'll talk about, AGL, all these guys go up. And the reason they go up is so they can be turned over, it turns out. And the turnover is necessary for the generation of ROS through the activation of NOX4 that is permissive for activation of MAP kinase pathways that control the transcription of genes involved in uncoupling ATP to generate heat in white fat cells. So in other words, this pathway has evolved as a way for the cell to ensure that there's enough energy on board for them to survive uncoupling of the synthesis of ATP, which is a serious issue. So in this sense, glycogen is a signaling molecule in the fat cell that triggers these other processes in the cell, not really just a way to store energy. So that's all I'm going to say about this. This is really nice work. If you're interested in the transcriptional pathways that are involved in the upregulation of these genes, we just published a paper a month or two ago in Nature Communications where we've dissected all these pathways. Now today I'm going to spend most of my hour and a half that I have left for my talk to talking about the hepatocytes and how glycogen plays an important role in hepatocytes. And I'm going to tell you two related stories today. And the first is in the context of anabolic stress, in the context of obesity. And in this case, glycogen plays a positive role in telling the cell that it's a good time to store energy when it's up. In the context of catabolic stress, and I'm going to talk about starvation today, glycogen is depleted and tells the cell, you better burn some energy now because we need energy in the body in the form of fatty acid oxidation, and you better make some sugar in that cell for the rest of the body. So this is what I'm going to talk about today. So we started this work, I would say in the 1990s, and my lab also was on a 2-hybrid insanity rush, I guess, trying to clone all kinds of interacting proteins. And among these was the gene PTG that you all know about, one of the scaffolding proteins that had been identified in the 90s by the Cohens and by the Roaches and other people. And we worked a lot in the 90s, late 90s, on trying to understand the biochemistry of the scaffolding proteins. We made chimeras and mutants and summarizing these papers. I like to tell people that today, all of these papers would have probably been one paper, actually, which is kind of sad. But in reality, that's the case. Now, I think that a really important experiment was done in the late 90s by my friend Chris Newgard. That kind of got a little bit under the radar screen, but very important paper, I thought. So Chris used an adenovirus to overexpress PTG in liver in mice. And what happened was the liver, as Chris described it, the liver turned into like a vacuum for glycogen. So you got high expression of the scaffolding protein, and it really drove the accumulation of glycogen in liver to heroic levels. And I didn't realize it at the time, but this was the first cue that the transcriptional control of the scaffolding proteins had a big effect on glycogen metabolism. Okay, before that, we're all thinking about direct effects of hormone action, but it actually turned out that the levels of the scaffolding protein was probably the biggest impact, had the biggest impact on accumulation and turnover of glycogen. So we started to, it took a while for this to sink in my own brain, I have to say, but eventually we started looking at this in the context of obesity in mice. And we'd been doing these experiments for other reasons in my lab, feeding mice a high-fat diet, looking at accumulation of different things. And we noticed, I think for the first time, that glycogen levels really went up, especially in a fasted state in response to high-fat diet in liver in mice. And really just in liver, not in fat. And this was accompanied by an increase in expression of PTG and in parallel the increase in expression of SREBP1, which is known to be involved in lipogenesis. And in fact, it turned out that this parallel, so this is again, SREBP, as you all probably know, is regulated by the mTOR pathway. It turned out that the activation of the mTOR pathway, either in fasting and feeding or a normal and high-fat diet, was paralleled by an increase in SREBP and an increase in PTG and the accumulation of glycogen. So we started to wonder if these things were linked, and I won't show you all the data, but I'll show you both in a negative and a positive sense. If we activated the mTOR pathway in liver by knocking out the TSC gene, which is a negative controller of mTOR, we saw that there was an upregulation of SREBP, which is known to be downstream of this pathway. And the scaffolding protein was also upregulated. And if we added shRNAs against SREBP, as you can see here, we blocked both the transcriptional upregulation of PTG, shown here, and the protein levels as well. So PTG is driven by this, the mTOR-S6 pathway that leads to activation of SREBP. Likewise, glycogen accumulation in liver also increases when you constitutively activate the mTOR pathway, and this is also blocked if you knock out SREBP. Okay, this is glycogen and synthesis of glycogen. So this pathway seems to be regulated by the SREBP pathway, and this is probably why it's elevated in high-fat diet. So just to summarize, hepatic glycogen levels are increased in obesity. Hepatic PTG expression and glycogen synthesis are increased by obesity through the mTOR S6K SREBP pathway. And the question is this somehow important in metabolism? So we created knockout mice of PTG. We started with whole body knockout mice, then we went to a liver-specific knockout mice, and the results were identical. And you can see here, not to our surprise, the glycogen levels are reduced in the knockout mice in liver. But to our big surprise, the lipid levels were also reduced in these mice. So this is just a triglyceride accumulation. In the knockout mice, you see it's reduced in high-fat diet. This is just an H&E stain. You see the steatosis seen in liver in response to high-fat diet is reduced. It's not completely eliminated, but it's reduced. So this was a surprise and confusing, and we wondered how PTG or glycogen might be controlling the lipogenic pathway. And so we actually looked at this, and in the knockout mice for PTG, where the glycogen levels were low, we noticed that there was a reduction in activation of the mTOR pathway, as characterized by increase in S6 kinase activity. This is just the quantitation of this data. Now, if you're following me, you're probably confused because I just told you that mTOR SREBP increases the expression of PTG. Now I'm telling you that PTG is necessary for the activation of this pathway. So come on, Alan, you're obviously very confused about this. And I think the answer is that there's a positive loop involved in glycogen metabolism. So in response to overnutrition, the mTOR pathway is activated. This gives rise to induction of SREBP. SREBP controls the enzymes involved in fat accumulation and fatty acid synthesis. It also induces PTG, which increases the glycogen levels. And then glycogen is somehow a positive controller to provide the sustained activation of the mTOR pathway. And this leads to the accumulation of triglycerides. Another way to think about this is that when energy comes into the liver, the first choice for storage is glycogen. And we all know this, actually. This is true in muscle and liver and fat. The first place we go to store energy is glycogen. It's easily mobilized in glycogen. But glycogen reaches a threshold, and when the threshold is reached, it then tells the cell to switch the accumulation of energy to lipids. Okay, so it's a positive loop that promotes the anabolic activity of the liver. But the big question is, how is this working? What's the sensor of glycogen? Now, we did a lot of RNA-seq analyses in these mice, in the knockout mice of PTG, where the glycogen levels are low, and we noticed a couple interesting things. One is that there's a big upregulation of genes that were controlled by PPAR alpha, which is a process involved in fatty acid oxidation, and a downregulation of genes involved in lipogenesis. So we started looking at some of those genes, and indeed, in the knockout mice, there was a big increase in oxidation genes, all of these under control of PPAR alpha, and a downregulation of the lipogenic genes, all of these in control of SREBP. So we started to think that there must be something that senses glycogen that is a negative controller of lipogenesis and a positive controller of fatty acid oxidation through PPAR alpha. And what was that? Well, the most obvious answer was that it was AMP kinase, okay? And Graham Hardy had shown years ago through elegant studies that glycogen actually bound to the beta subunit of AMPK2 inhibits activity as a dominant inhibitor. And these studies were very, very nice and really showed this in a convincing way. So we started to look at AMPK in some of our models, and indeed we showed that in all models of steatosis that we've been studying for other reasons in the lab, there was always a downregulation of the activation of AMPK. Ruben Shaw had showed that if you infect cells with a constitutively active form of AMPK, there's a reduction in steatosis. And then finally, in the knockout mice, in the PTG knockout mouse, where glycogen levels are low, there's a dramatic increase in AMPK activity. So this is all consistent with the Hardy data suggesting that glycogen is an important controller of AMPK activity. So what we think is happening is that this positive loop controlled by glycogen is regulated through AMPK activity. That AMPK, as we all know, inhibits the mTOR pathway. When glycogen accumulates, it de-inhibits this process. So what about the fatty acid oxidation pathway? Well, we also looked at that. We focused our attention on genes that we know that are involved in liver and fatty acid oxidation. PGC1 alpha is one of these. We looked at expression of the gene in knockout mice, and you can see there's a dramatic increase in PTG expression, in PGC1 expression in PTG knockout mice. And this is reduced by adding an inhibitor of AMPK. So even with respect to the fatty acid oxidation arm of this process, we think that glycogen is inhibiting fatty acid oxidation by blocking AMPK, which is normally involved in activating this process. So this is our overall scheme. What we think is happening now is that glycogen is a positive effector of anabolic properties in liver. Okay, it inhibits AMPK, thus blocking AMPK's ability to increase the fatty acid oxidation. So this is reduced when glycogen is high in obesity. And likewise, the lipogenic pathway is activated by glycogen because the negative effect of AMPK is blocked when glycogen is high. Okay, so in the anabolic state, in the obese state, glycogen is pushing us to store more energy. Okay, so that's just a summary. Now, what about the glucose function of liver and what's happening in catabolic stress and starvation? So I'll just remind you that we have this amazing ability to maintain a constant level of glucose in the blood even after prolonged starvation. So in the case of starvation, when we stop eating, the glucose starts to go down. You can look in the purple line. After a few hours, it kind of hits a steady state, and then it stays level. Now, the steady state is maintained by the liver primarily due to the breakdown of glycogen first, but the glycogen is exhausted at some point. And so then the liver kicks in by making its own sugar. And it does this after the glycogen is gone, okay, and starts to come out. Now, there's a two-part component to this. The first part is that gluconeogenesis, the generation of sugar by the liver, is fueled by just an increase in the substrates that are coming from fat cells. So lipolysis is driving the gluconeogenesis in the early stages of starvation. But later on, there's another thing that happens, is that the liver kicks in and starts to make its own enzymes to control the production of glucose. Okay, so the gluconeogenic enzyme production is regulated by glucagon, and in this case, glucagon activates the pKa pathway, phosphorylating and activating Krebs. Krebs gives rise to intermediate transcriptional regulators. PGC1 again comes up as works as a cofactor with HNF4, NR4A, and there are other genes that are involved. Together, these genes control the expression of the gluconeogenic genes like PGC1 and glucose-6-phosphate, phosphatase, and other enzymes. So we wondered whether glycogen levels might, in the scenario of prolonged starvation, might also influence the expression of these genes. And of course, I'm asking this question because the answer is yes. So we went back again and looked at the RNA-seq data, and among the pathways that we noticed was a pathway controlled by HNF4. HNF4 is the major transcription factor that controls a lot of these genes, and these are upregulated. So we thought, aha, maybe glycogen depletion is really controlling the regulation of the gluconeogenic genes. And it's not a completely crazy idea because GWAS studies have actually shown PTG really does associate with a high log score with fasting blood sugar and insulin levels. So it's not a completely crazy idea. It's probably also true in humans. So we took a three-pronged approach to this question. We first manipulated the glycogen levels in the liver by, as I just showed you before, by knocking out PTG, by overexpressing the scaffolding protein, or by adding an inhibitor of the breakdown enzyme that would also result in accumulation of glycogen. So I'll start first with the knockout of PTG in a case in which the glycogen levels are reduced. You can see in this case, we looked at the sensitivity of the liver cells from the knockout mice in vitro to glucagon, and we see that the glucagon responsiveness of these cells is dramatically improved when it comes to the regulation of the gluconeogenic genes, as you can see here in the knockout mice. So the glycogen is depleted and the gluconeogenic gene expression goes up in response to glucagon. We can also just add a permeable a cyclic AMP and we see the same thing. Now importantly, the cyclic AMP levels themselves are not controlled by glycogen. Nothing upstream is controlled, but the glucose production is enhanced if you knock down the glycogen by knocking down the scaffolding protein. So that's one side of the coin, and what happens then if you increase the accumulation of glycogen, and of course we see the opposite effect. We see the stimulation of the gluconeogenic gene program by the glucagon is reduced, as you can see here in all three cases. And AMP kinase activation, or AMP kinase inhibitor blocks the positive effect of glycogen depletion on the regulation of these genes, showing that it's also, as I showed you before with the lipogenic gene expression, is all happening through AMPK. And we also made a double alpha-1, alpha-2 AMPK knockout in liver cells, and we showed that the same thing, that in these cells, the gluconeogenic gene expression is repressed in response to glucagon or cyclic AMP. So what we think is happening is that glycogen is modulating the responsiveness to glucagon, okay? And when glycogen is replete, it's telling the cells that we don't need to respond to glucagon because the blood sugar levels are going to be just fine because the glycogen is very high. But when we lose the glycogen, AMPK is activated, and then AMPK somehow plays a permissive, important role in the regulation of these genes. Now, the question is, what is the mechanism of this? And we're not completely sure yet, but we know it's not upstream. We know it has nothing to do with the glucagon receptor, PKA activation of Krebs, so it's got to be something downstream. And I won't spend a lot of time to show you this today, but we focused our attention on the Krebs cofactor, CRTC2. This is a gene that was discovered by my colleague Mark Montminy at the Salk Institute. And we noticed that there was a very nice AMPK consensus sites in CRTC2. So we looked at phosphorylation. We were able to show that these sites were hit by AMPK. And I'll just quickly go through this, and what they're doing is to increase the stability of this coactivator. Okay, this is a cyclohexamide experiment. So you can see here that in the knockout in the knockout cells of PTG, where the glycogen levels are reduced, CRTC2 stability is increased, and we see the same thing if we provide cells with a direct activator of AMPK. You see there's a big increase just in the protein levels of CRTC2 that we don't see in the AMPK knockout. And I'm skipping over a lot of the other data. We published this stuff a few months ago in the JCI. So this is what we think is happening. We think what was happening is that when glycogen is depleted, AMPK gets high. AMPK now has a permissive role by increasing the protein levels of this cofactor for Krebs, allowing for a full responsiveness to glucagon. And of course, this is what you would want. If you're depleting glycogen, you need the liver to kick in and make the glucose in another way. And this is the mechanism for that. So another way to think about this is that in the fed state, when insulin levels are high, there's another pathway I didn't talk about, but the CRC2 levels are depleted. In the fasted state, when glycogen levels are lower, AMPK is higher, promotes the stability of this protein and promotes the actions of glucagon. So again, this is just a summary of what I've told you about today. We think glycogen is a signaling molecule. It's not just an inert way to store energy, but it's also a way for glycogen to cue the cell to do the right thing at the right time. In the fat cell, it's to make sure that you have enough energy on board to undergo the beiging of white fat cells. And in the liver, in the fed state, in the anabolic state or the obese state, accumulation of glycogen is telling cells to store more energy. In the fasted state, it's telling cells to burn more energy by increasing fatty acid oxidation and increasing the synthesis of glucose for other cells in the body. So I'll just stop right there. Thank you very much. Let me just acknowledge some of the people that have been involved in this work. We did a lot of these studies in collaboration with my colleague at the Salk, Ron Evans.
Recording in progress.
And now you tell me. And a lot of this work was done by fellows in my lab, Bi-Chin Zhang and Binbin Liu, and I'll just stop there and take questions. Thank you.
Right, Dr. Cullen. I would like to ask you whether cells that have a lower concentration of glycogen, such as muscle cells or neuronal cells, whether you would see also this, this effect of glycogen inhibiting AMPK.
So it's a really good question, and I was intrigued by the previous talk because I thought maybe this is also happening in brain, actually, especially with the anti-inflammatory effects. Okay, so we think that we have a lot of data on this now in liver that AMPK is an anti-inflammatory, and we published this in Cell a few years ago. So we think glycogen is pro-inflammatory, actually. So it could be that there are other signals going on It could be happening in muscle. We haven't looked in muscle. So I really can't talk about that. But it could be happening in muscle too.
Yeah, great talk. So have you recapitulated anything in vitro to see if different glycogen architectures had different impacts on AMPK?
That's a very good question, Matt. So I think you probably know that the branching state of glycogen is very important in its ability to bind to AMPK. And I glossed over it a little bit, but AGL is markedly transcriptionally controlled both in liver and fat. So I think we're missing, a lot of us haven't paid as much attention as we should. I think it's an under-investigated area as to the control of the debranching of glycogen. And I think AGL is really a great target for us to study.
I was just wondering, Alan, whether you thought that this inhibition by glycogen is simply sequestering AMPK away from the glycogen so that it can't serve its other functions or some of its other functions.
Yeah, that's a very good question, and a reviewer has asked me that question. And,
I promise I was not the reviewer.
Okay. Yeah, and I just can't answer it. I mean, it could be sequestering or it could be catalytic inhibition, or both, right? The sequestering idea is very interesting because, as we know, and as you guys have been showing today all day long, glycogen is not in solution in the cell. It's found in bodies, and it really could be it's sequestering and degrading even possibly AMPK. So I think there's a lot to learn about that. It's a very good question.
Thank you so much, Alan. So now we need to move to the next speaker. So unfortunately, our next speaker cannot be joining in person today, but we are really glad that she can connect online. So it's my pleasure to introduce Luke Sulo from the University of Copenhagen, who is going to be talking about glycogen and glucose in motion, the metabolic demands of exercise. Thank you, Luke.
Hello everyone, and greetings
thank you, Ulrike. So we have some time for questions. Can you hear us? Okay, perfect.
Thank you very much. Really nice talk. So the two different types of exercise that you did, can you predict which type is better, the high intensity versus the prolonged?
You mean the human?
Correct.
The human study, yeah.
Yeah. And can you replicate that in the mice as well?
Yeah, I think we could. I mean, we haven't done. I think I think the electrically stimulated muscle contraction model, where we have done the glycogen resynthesis experiments in, will recapitulate maybe more either the sprint or the resistance type exercise. Whereas the in vivo exercise running protocols, where we see this reduction in muscle glucose uptake during exercise, I think that more is sort of towards the endurance type exercise we did in that human study. I mean, long-term aerobic exercise, it very efficiently builds up glycogen in the days after. So I think in that setting, maybe that would be a more relevant setting to study these mechanisms in, if that answers your question.
Well, I'm going to have a question. I have a question also that is more related about the molecular mechanism related to the ROS production and signaling. So in your knockout, you mentioned that, I mean, you have like depletion in the ROS production related to the glucose uptake. But is there like kind of also imbalance of different reactive oxygen species in this situation? Or is more about abundance, or like if there is some kind of like adaptation in this context?
Response of exercise training to glycogen buildup, because we know that exercise training long-term will increase the storage of glycogen in the muscles. So we don't know that for the NOx2 deficient mice. If repeated exercise bouts with repeated increases in bursts of reactive oxygen species would bring in the glycogen needed. But we know it from the RAC1 knockout mice, where we have done an exercise training, like a longer-term intervention. And while we didn't see this glycogen resynthesis right after one bout of contraction, it looks like long-term exercise can still increase muscle glycogen to similar degrees as in the wild-type mice.
Okay, thank you. So we don't have any other questions. Thank you so much for your presentation. We can move. Okay, so our next speaker is Marcelo Green from the Institute of Molecular Biology of Barcelona, IUBMB CSIC, who is going to talk about the molecular basis of glycogen metabolism regulation in bacteria.
Okay, now yes. For the invitation to contribute to this lovely day, right? And to all the people that are participating in the organization and of this meeting, right? So today we will pass a little bit to the glycogen and bacteria, right? And I will talk about the structural basis of glycogen metabolism regulation mainly. So just a brief introduction. So this story started quite a long time ago. At the time, I performed my PhD in Buenos Aires, in Argentina, in the Leluar Institute, right? So this was the place where the nucleotide sugar were discovered, as Philip mentioned early, And because of that, Louis Leloir obtained the Nobel Prize in Chemistry in 1970. So the first nucleotide sugar to be discovered was UDP glucose, right? But they didn't stop there. They continue their research and identify UDP-N-acetyl glucosamine, UDP mannose, and UDP-N-acetyl galactosamine, for example. But at the very beginning, they didn't know the function of the nucleotide sugars, right? And they they didn't know the chemical structure of the compound. But they realized that UDP glucose indeed can act as a glucose donor in the context of an enzymatic reaction, which was the trihaloase phosphate biosynthesis. And a second example came up through the sucrose biosynthesis, but maybe the main relevant at that time was the change of the paradigm in glycogen synthesis by means, as Philip mentioned, it was thought that glycogen phosphorylase, it is the enzyme that catalyzes a reversible reaction and was indeed in charge of biosynthesis and breakdown. But they discovered the glycogen synthase, right? And some years later, myself and another PhD student from the Leloir Institute, so 50 years later, we had the possibility and the chance to provide the first crystal structure on this enzyme, right? We'll talk about a little bit on that. So all this work was performed in a really, really modest environment, right? For political reasons. So Louis Leloir resigned from the university and rented a house, right? And transformed this house into a very small institute. So you can see here the rooms transformed in 3 labs, a bathroom, kitchen, a place where they could store the chemical compounds, and a very small library, right? And working according to what we know in Argentina, the first law of Leloir, number of tubes per ideas remains the constant, right? They didn't have quite a lot of funds to work with, and they needed to be quite really creative, right, to perform the experiments. Here is a picture of Luis. He was a bench researcher, right? Very systematic in terms of 9 a.m. to 5 p.m. every day, and Saturdays bibliography. That was his almost his entire life. He worked until his last day of life, right? And then a very small group, just 5 people here plus a visitor. And you can see a picture of how the laboratory it looked like at that time, all made in wood, right? Here is the, the entrance of this very small house, but on your right, you have a big one. Well, in the big one, some years before, his mentor, who was the first Nobel Prize in Sciences in Argentina, Bernardo José, moved before, right? So you had a couple of houses with 2 Nobel Prizes, one awarded and the other one to be awarded, working together, and the institutes connected through a couloir behind, right? That was the situation, right? So really familiar. And here is after some years, the privates and the government, the Argentinian government, contributed with necessary funds to build up this construction where I did my PhD, right? So Luis, essentially, his main contribution, as Philip mentioned, was to establish the basis of the glycogen transfer reactions, right? A big, big contribution with the discovery of nucleotide sugars. In our field, the discovery of the glycogen synthase. He was proposed for a second Nobel Prize later on because of the discovery of the precursor oligosaccharide for glycoprotein synthesis, our lovely glucose-3 mannose 9 and acetylglucosamine 2 linked to dolicol paraphosphate. He didn't get it, but Armando Parodi took over all this research. I did my PhD with Armando, and Armando was a key player in the discovery of the quality control mechanism of glycoprotein folding in the ER. Essentially, he discovered this enzyme, a folding sensor in the ER, And working in this enzyme, I started to work in glycogen synthase, just by chance, right? And I purified the glycogen synthase from Agrobacterium tumefaciens. And then during a very, very short stage in Pasteur Institute with Pedro Alzari, I have crystallized the enzyme, right? And then together with Alejandro Busquiazo, we saw the structure, we contributed the structure of the first enzyme. So which were the key messages at that time? So the key messages, imagine that the only enzyme reported from the pathway was glycogen phosphorylase, right? The first allosteric enzyme to be solved by David Badford and Louis Johnson in UK, and then Robert Fletterick. Glycogen synthase, indeed, it is comprised by two Rosman-Fold domains, so the C-terminal domain, the N-terminal domain, and in the middle you have the catalytic center, right? Usually the C-terminal domain, it binds the nucleotide sugar, and the N-terminal domain will bind the oligosaccharide acceptor, right? So again, this is the question at that time was how a glycogen synthase it looks like. So that was the question again at that time. So another very important message was that there is quite a lot, an important flexibility, inter-domain flexibility between the N and C terminal domains, which indeed it is really important for regulation in the context of eukaryotic enzymes, right? The other key message was that if you compare the course of a glycogen synthase and a glycogen phosphorylase, they were the same, right? They were really, really similar. One enzyme processing the glycogen in the biosynthetic way, and the other one breaking down it. So at this time, during all this work, two names came very often in the laboratory. One name was Joan Guinovart, and the other name was Peter Roach, right? So during this work, during all this work, structural work, we realized also that Joan Guinovart was trying to crystallize the glycogen synthase here. And this was made with a lovely, really lovely professor, Ignacy Fita, with whom I had the chance to share office, which is really, really nice. And together with Juan Ferrer, they contributed the crystal structure of a glycogen synthase from Pyrococcus avisi, right? Which it is a homotrimeric assembly, and in principle, this assembly is made by the N-terminal domains with the C-terminal domains free, not involved in the intersubunit interactions, and so you have quite a lot of, you would expect quite a lot of flexibility there, right? And they were doing cryo-EM at that time. So this is a negative staining, this is 2006, right? Then Joan and Ignacy, they continued to work on the enzyme and reported a high affinity glycogen binding site away to the active site, and looked at the processivity mechanism of glycogen synthase. Then, as an independent researcher, what I did is to focus in another key player in the regulation of bacterial glycogen synthesis, biosynthesis, which is the ADP glucose paraphosphorylase, right? And the ADP glucose paraphosphorylase, it catalyzes the synthesis of ADP glucose from glucose 1-phosphate and ATP, right? And this enzyme is allosterically regulated by metabolites that at some point they will reflect the energetic flux within the cells. Essentially, activators are metabolites that will represent signals of high carbon and energy content, like, for example, FVP, and inhibitors like AMP will indicate low metabolic energy levels, right? So we solve first the crystal structures of AGP from E. coli in the presence of AMP, the negative regulator, and FBP, the positive regulator, right? So as you can see, it is formed by, it is a homotetramer that can be viewed as a dimer of dimers, right? And each protomer or each subunit is composed by here a glycosyl transferase domain, so a Rosman fold domain where the catalytic, the catalysis will occur, and then a regulatory domain, left-handed parallel beta helix, right? And we found by X-ray crystallography that the two allosteric regulators are going to the same place, right? Which is formed by an L-beta helix domain from one protomer and a catalytic domain from another protomer coming from different dimers, right? So according to that, we proposed at that time a model of action or the interpretation on how this enzyme could work. And so here is the regulatory, here are the regulatory sites. Here you have what we call this a sensory motif in red. So you have it is a chain of amino acids which will make very short secondary structure elements, and which is pretty well connected with the catalytic side. So one interpretation is that the negative signal made by AMP will indeed be transmitted through the sensory motif directly. So here is one of the catalytic residues. So if you imagine any change in the conformation of these residues will impact the catalysis. And, but at the same time, we found that this residue, arginine 130, it is stacked against the nucleobase and most probably transmitting the signal through two regulatory loops that we could not model at that time. We had no density to provide the model, right? And regarding the positive signal through FBP, according to X-ray crystal structures, we thought it was just transmitted through the sensory motif. Just a note, we mutated this arginine 130 per alanine, and we found that you can have so this mutant displayed a very dramatic increase in the enzymatic activity and overproduction of glycogen in vivo. So we had at that time the location of the allosteric regulators in the enzyme, but not the mechanism. And because of that, we solved the same structures by cryo-EM, right? With the goal to see the loops, right? And so we solved the two structures of, in the presence of FBP and AMP at 3 Armstrong resolution. And two differences between cryo-EM and X-ray crystallography. The first one was the location of the FBP, right? By X-ray crystallography, the FBP was more solvent exposed compared to the FBP found by cryo. And the second, if you compare the location of FBP with the AMP, they overlap, right? And this accounts, so this explains why the sensitivity to inhibition by AMP is modulated by increasing the amount of FBP. And this is the second surprise that we had, right? Which is the conformation of the RL2 loop. The RL2 loop, indeed, is a loop that will cap the active site, will interact with ATP, right? Which is one of the substrates of the reaction. And the funny thing is that in the case of the negatively regulated enzyme, these loops RL2, they are visiting or interacting together, right? Far away from the active site in a non-competent manner. And then in the presence of FBP, they will go to the active sites and interact with ATP. So, and this correlates with, again, on affinity for ATP, right? And this is a very simple but very beautiful mechanism. This is the point where I crossed Joan physically for my first time, right? So he was able to see before publishing these structures here, in fact, in IRB. And it was really beautiful to see his reaction, his eyes, typical from a 100% scientist, right? It was very, very nice. And then the third project, it is unpublished data. It is already accepted in Nature Communications, but you would expect to have it publicly available maybe in 15 days, right? Which is glycogen phosphorylase. And glycogen phosphorylase and its relationship with what we call the phosphoenol pyruvate sugar phosphotransferase system. So in bacteria, the PTS system catalyzes the transport of sugars, like, for example, glucose. And this, in this system, there is a very small protein called HPR, which plays a very central role. And why? Because it will be shuttling phosphoryl groups, right, between different components. And one of the components are the E1 protein and the E2A protein, which will give the specificity for the sugar. So essentially, imagine that you have glucose up to here. It will be phosphorylated and transformed in glucose 6-phosphate. And then this will enter into glycolysis. Then at the end, phosphoenol pyruvate will be generated. And this component will be used by E1 to be autophosphorylated. Then E1 will transfer the phosphate group to HBr in a histidine residue, histidine 15, and then HBr will transfer this phosphoryl group to the E2A system, to the E2A protein, right? So something to keep in mind, so the activity of GP is strongly enhanced by the unphosphorylated HBr, but remains unaffected by the phosphorylated form. And also HBr was found to interact with many other proteins that participate to the central carbon metabolism. So we would like to see the mechanism on how HBr could control the glycogen phosphorylase activity. So we saw the cryo-EM structure of the E. coli glycogen phosphorylase. If you take a look on the native mass spectrometry profile that we perform in Oxford University, essentially we have three main species, monomeric, dimeric, and tetrameric. But if we take a look on the structural assemblies that we can solve by cryo-EM, we have the homodimeric and homotetrameric. So the tetrameric assembly can be described as a quasi-symmetric dimer. Essentially, imagine that you have, my hand is a dimer, and my other hand is the other dimer. So essentially, the active sites are facing to you. Essentially, they will be occluded in the context of the tetramer. So, and that's the reason, because in the context of the bacterial and eukaryotic enzymes, it is believed that it is a resting assembly, right? Non-catalytically one, right? So, and then it is quasi-symmetric because you have like a rotation of 30 degrees, which indeed will provide quite a lot of movement, right? And we don't understand why it is so flexible, right? Things to be understood in the future. So this is the dimeric version of the protein. So as you can see, the dimer is supported by a very extensive number of interactions here. So you have a GTP fold domain here, a GTP fold domain here, and you are seeing, so the two groups that will accept the oligosaccharide are facing to you, right? And then you have the upper region. If you turn this 90 degrees, you have an upper region, a central region, and a bottom region. In the upper region, there will be the entrance to the maltooligosaccharide residues. And then if you compare the glycogen phosphorylase, this bacterial glycogen phosphorylase, with the eukaryotic one, all the allosteric regulators in the eukaryotic one are in the bottom, right? And it will communicate and transmit the signal through an alpha helix to some residues here, which will in fact cap the entrance of the active site. Here is the protomer, a typical GTP fold enzyme, we found the PLP, which is a cofactor of the phosphorylated reaction. And then Louise Johnson came up again, right? And forever. By means, she was, after working with the eukaryotic glycogen phosphorylase, she also made an exercise with MALP. So in E. coli, you have two versions of phosphorylases that could act in, one in maltoligosaccharides, so linear alpha-1,4 linkages, and another one in the whole glycogen particle. So she made a really nice exercise with the MALP and with making complexes and so on. And she distinguished two main components of the regulatory mechanism. But not the regulatory mechanism, on the interaction with the maltoligosaccharides. Two loops called 280, 380, it doesn't matter, but these loops here and these loops here, essentially they will, the conformation, so the conformational changes of these loops will provide the affinity or not to the maltoligosaccharides in the context of the linear oligosaccharide or the glycogen particle. So we made a structure of glycogen phosphorylase in the presence of HBr. If we take a look on the mass spectrometry data, a broad range of multimeric species, but essentially the majority are in a ratio 2:2, so 2 glycogen phosphorylase, 2 HBRs, and 4 glycogen phosphorylase and 4 HBr. And essentially, the HBr will interact with the N-terminal domain of GLGP, of glycogen phosphorylase, very close to a site called glycogen storage site, right? Very, very close to the loop 380. If we take a look on the interactions, it is mainly made guided by electrostatic complementarity. So you can see this here. So here you have negatively charged residues coming from the glycogen phosphorylase and positively charged residues coming from the HBr, right? And a really, really nice thing, if we zoom up in this interface, right, we will identify a couple of regions at this interface. The first one, it will be involved by a hydrophobic interface, right? Here in orange, you have glycogen phosphorylase. Here you have a phenylalanine, which will be stacked or anchored into a pocket made by also phenylalanine residues from the hydrophobic phase of glycogen phosphorylase. But the other point, which was really, really interesting, is the histidine 15. So the histidine 15, indeed, it will have the phosphate, right? The phosphoryl group. And this phosphoryl group will be released and be in contact with another partners, right? So taking a look on the structure, on the other side, on the side of the glycogen phosphorylase, we identified a pocket of 3 histidine residues, right? Histidine 432, histidine 806, and histidine 809. And taking a look on the density maps, we realized that maybe there was an unexplained extra density on one of these histidine residues. And we made a very simple exercise. It was reported by Albert Heck. He made a really nice histidine phosphoproteomic study. And if you go to the raw data, you will find just one histidine residue to be phosphorylated, and it was 806, right? Which corresponds to the histidine where we could see an extra density. So essentially, you have a release of phosphate, of a phosphoryl group from this histidine HBr to the glycogen phosphorylase. Then we performed quite a lot of mutagenesis analysis, we can kill the enzymatic activity, we can disrupt the tetramer, which will become a dimer and be more active, but maybe we can discuss this later. And this is one of the final slides. There are some structures reported with HBr, and different partners. So, for example, this is our structured glycogen phosphorylase. Then you have the E1 HBr complex, the E2A specific for glucose, specific for mannose, and specific for mannitol. But you can identify that the electrostatic complementarity also dominate this kind of interaction. By means, these proteins, they have different folds, right? But the electrostatic complementarity is there, and in all cases, you have a release of phosphate from HBr to the other ones, right? Conclusions of this last part. So collectively, this research, I think, reveal a complex, very complex regulatory mechanism for glycogen phosphorylase, and most probably for glycogen biosynthesis in bacteria, apart, for example, of transcriptional regulation. So, for example, oligomeric plasticity, the regulatory and allosteric compounds, then phosphorylation, and so it seemed to be a very dynamic system, right? So it appears also that bacteria have evolved different strategies to control this glycogen biosynthesis and breakdown. So next step in our lab is to try to visualize how not just glycogen phosphorylase it works, but also other enzymes of the pathway, but in the context of the particle, right? How they interact alone or together, and could, for example, process the alpha-1,4, process the alpha-1,6, again, in the two directions. Very challenging project, but we will go in that direction. Just to thank all the collaborators in the context of this last project, and then a final word to Joan. By means, I just crossed hands with him twice in my entire life, right? So very few. But if I would kept something from him, it was, of course, his manuscripts, lovely manuscripts, and then his eyes. By means, plenty of energy. And when I was explaining to him or showing to him how, for example, AGPAs was working, he was like a child, and you can feel it. And this is something very, very, very nice to see in a researcher, right? So that's it. Thank you very much for everything, and I'm happy to take questions.
Okay, so we have time for one quick question.
I assume that very nice talk. I assume that the role of pyridoxal phosphate in the enzyme is quite different from the role in transaminases, for example, right? Or because it's a catalytic role also, right?
Yes, it is a catalytic role, and it is conserved also in eukaryotes. One thing, I think last year, there is a manuscript. I'm sorry. Yeah, there is a manuscript where researchers from Chile they identified glycogen phosphorylase that it doesn't use PLP.
I see.
So they made an exercise taking a look on possible regulators, but you could have another kind of catalytic mechanism in that context. But this is just one first manuscript.
Yes, and then with respect to the histidine phosphorylatable, well, that's very much reminiscent of the two-component system of bacteria, right? So do you have aspartate acceptors on the other side or not?
No. Indeed, if you take, if you take a look, we need to go further in this research, right? Because if you take a look on the conformations of the proteins in the unliganded form with respect to HBr, and by means the conformations are the same. So we have in mind a possible way in which the enzyme could be controlled or activated, but this is not proved according to these structures, right? So I think that we will need to catch up or work a little bit more on that direction.
I think there is something thermodynamic there. I mean, for the thermodynamics,
yeah.
I think it's the system phosphohistidine, phosphaspartate. It's one that makes it reversible, I think.
Yeah, to my point of view, there is another thing that we are missing here, which is a glycogen particle. So essentially, if you have, in principle, glycogen phosphorylase plus HBr, you gain 5-folds on affinity for glycogen. And I think that this is a very old question, but not answered.
Looks allosteric or something like that, right?
But, well, how is the glycogen particle bound to these kind of enzymes? Not regarding the catalytic site, regarding the surface. And in the context of the glycogen particle, so how, well, could biosynthetic enzymes interact each other in the context of the glycogen? So, part of the, a big variable there is missing. So, of course, we were working for years trying to produce this kind of understanding, and this is great from many, many different labs. But I think that at some point, glycogens should, come up.
Okay, so thank you very much for the great talk. Let's move to the last speaker of this session, Kei Sakamoto from the University of Copenhagen, who is going to talk about physiological control of glycogen synthesis and utilization in skeletal muscle. Lesson from Joan Guinovart.
Does laser pointer work? This one. This one. No. No. Now. Okay. All right. Thank you.
Okay. What is your pleasure? Excuse me.
Yeah.
What is?
Should be this one. Okay, so while setting up, so I would like to thank the organizers for inviting me for this tremendously important event. So that's it's a real pleasure actually to honor Joan's legacy with his family and friends and colleagues and also glycogen lovers. So I know it's a bit now, kind of a challenging task for me because people are getting very hungry. So please use your liver glycogen ketone bodies to keep your brain working. Okay, bear with me for the next 25 minutes or so. So what I'm going to talk about today is a physiological control of glycogen metabolism, which is quite a fundamental process. And I'd like to tell 2 stories about this in conjunction with my actually, interaction, and then also a story with Joan Guinovart in terms of his, lesson he gave to me, and then also his insights that shape, and also encourage my science. So that's, I think many things have been said. So I was here 2 months ago to also honor his legacy in terms of global impact and societal impact for Joan. So the conclusion of the symposium was that he is an outstanding scientist with no doubt. He's a great leader, a mentor, and also most importantly, he's a very nice guy. So that was an important message to convey. So Marcelo already mentioned as well, but if I describe him, he is full of passion, genuine curiosity, and also love for the science, and then particularly, the macromolecule called glycogen. So that's, I first met him in 2009, actually for a common friend called Professor Louis, Louis Hughes, sorry. Maybe I'm becoming hypoglycemic actually myself. So so there was actually the symposium to honor. He's actually a transition to emeritus professor. So so we had a garden party at his place on Saturday, and so that you can see that I was quite young, 7 years ago, and so you can see Joan looking at the session quite seriously. And also you can even see Dario in the coffee break talking to Joan here. And so that's actually, I had a conversation with Joan because I knew he was working on glycogen, and so I started working on glycogen. So that's so I started with some very kind of random Scottish Japanese culture, and when it comes to speaking of glycogen, his face totally changed, and we instantly actually clicked, and then we became friends for longer time. So that's so Philip already gave a great overview of the discovery of insulin signaling pathway in terms of regulation of the glycogen synthase enzyme. So I did my PhD at Jocelyn Davis Center, where many of the key proximal insulin signaling has been identified there, and I moved to MLC protein phosphorylation unit, and first actually worked with Dario as a postdoc. So that's so I had opportunity to work on insulin in terms of regulation of the glycogen synthase, so as also exercise. So as Philip mentioned, this actually very complex regulatory process was beautifully resolved to some extent, but we shouldn't take it for granted because if you look at the timeline of discovery of insulin 21 to the discovery of the speed K1, it took over 70 years, so it's a long-term processes. So that's Philip said that's okay. This has been solved, but since actually majority of the data are from in vitro and cell culture studies, so Dario had idea that we need to do proof of concept in animal models. So that's he was particularly interested in looking at the insulin regulation of AKT-mediated GSK3 phosphorylation in the regulation of the glycogen synthase in muscle and liver. So that's there was a talented PhD student at McManus who was tasked actually for this project. So that's he generated insulin-insensitive GSK3 alpha-beta knocking. So where AKT phosphorylation sites are mutated to replace to alanine. So that's PKB or AKT is no longer able to control or deactivate GSK3. So that's glycogen synthase cannot be dephosphorylated and activated if that is really the case. So that's I had the honor of doing the actual experiments because I was trained for the physiology in Boston. So that's we injected actually mice with the wild type and GSK3 knocking with the insulin and looked at insulin signaling pathway, glycogen synthase activations. As you can see, GSK3 was nicely phosphorylated by insulin, which was associated with dephosphorylation and robust activation of the glycogen synthase activity. So beauty of this experiment was that the insulin response was totally blunted. So that's as a postdoc, I was less astonished. A single amino acid on the GSK3 is a major controller of the glycogen synthase activation, so which is quite remarkable, in our hand. So, however, what was puzzling to us was that when we measured glycogen content in fasted and fed animals, so as you could see, there was no difference between wild type and glycogen synthase knocking. So that's so I think the interpretation of the result was that okay, maybe there is a compensatory pathway, for example, insulin-independent exercise pathway, which was sufficient to be able to accumulate glycogen. So, however, I was not that convinced in this result. So that's when I became a group leader at the MLC protein phosphorylation unit. So I followed up this experiment. So I had my first PhD student, Michal Boskila. So that's I was still on bench PI, so that's we did the experiment together. So what we did was that we wanted to measure de novo glycogen synthesis by stimulating isolated skeletal muscle in the chamber in the presence of insulin and also tracer glucose, so that we can measure glucose incorporation into glycogen as a readout of glycogen synthesis. As you can see, in the absence of the insulin, there is no differences between the glycogen formation and in the presence of insulin, so you have a quite robust increase. However, there was no difference between wild type and GSK3 knocking. So this means at least this phosphorylation event is not necessary for the synthesis of glycogen, which was quite puzzling because, as we saw, there was no activation of insulin by phosphorylation. So that's this was an insight actually from Joan. So that's when I looked at the literature back in '86. So Joan already actually implicated that glucose 6-phosphate plays a key role in deposition of the glycogen in both muscle and liver. So that he wrote a beautiful review article in '97. So that's this is a scheme. So insulin stimulates glucose uptake through AKT pathway, glucose translocate to the membrane, so that glucose gets inside and get phosphorylated by hexokinase to form G6P. So this actually acts as a precursor for glycogen synthesis. At the same time, G6P is known to be a allosteric activator. So that's he pointed to the role of G6P, and then he humbly concluded in this actual article that the increased G6P concentration in cell may account for activation of glycogen deposition. So that was his conclusion. So that's I also want, so and then he actually invited me right away after we met. So this was my title and 2009. So to give actually a lecture at IRB. So I also want to acknowledge that Jørgen Jensen, who is from Norway, who is present today, so he also helped me in terms of developing this concept of the role of G6P. So thank you very much, Jørgen, about this. And so that's here actually comes. So we have two regulatory processes through reversible phosphorylation. We talked through insulin-mediated G6 phosphorylation, and then glucose transport actually increasing G6P. So that's allosteric activate. So that's why we already showed that we had a model to look into this. So however, the challenge is that how you can study G6P mediated. Because since this is not a covalent, actually coupled or the high affinity binding, if you actually homogenize the tissue, so this G6P binding is readily dissociated, so that you cannot look at the effect of the metabolites in vivo. So that's I came up with the idea, okay, so then how actually G6P regulates glycogen synthase. So this was actually Peter Roach initially proposed that there was no crystal structure. Actually, as Marcelo mentioned, it was not early 2000. So that's actually Peter looked into the sequence of conservation from yeast to the different species. So there are clusters of the arginine and composed, positively charged arginine to bind to the phosphate as a negative charge to comply G6 binding pocket. So that's so since actually this there was no clear information about specific amino acid that is important to regulate this actually the event. So that's my PhD student Michał did a mutagenesis analysis for single, double, triple expressing hex cells in the presence of glycogenin, and then try to identify a mutant where it retains normal catalytic activity and then still can be regulated through reversible phosphorylation to regulate controls activity. And so that was the criteria. So she found one of the mutations, which is arginine 582A, is the most suitable actually mutation. So with this basis, and so that's there was a structure a few years back. So it was nice to see that it's actually comprising G6 binding pocket, and then arginine 82 is indeed an important binding residues of glucose 6-phosphate. So that with that, actually, we made G6P resistant knock-in animals to study the role of G6P in vivo. And so that's John was, of course, always kind and enough to say, okay, you're a very clever guy. So that was his comment I still remember. I appreciate this. So this is actually a summary of our 3 years of the work. So that's I already showed that the insulin effect on glycogen synthesis using GSK3 knock-in. And however, if we use G6P resistant knock-in animals, so basal is already reduced. And then, so if you look at the absolute value, then there was a profound decrease in insulin-stimulated glycogen, glucose incorporation to glycogen. So we even made a triple knock-in closing with GSK3 double knock-in to show that there was additional decrease actually of the glycogen synthesis. So that's, as you could see, that it looks so, even though GSK3 is an important regulator of phosphorylation-dependent activation, the in vivo, the glucose uptake-mediated G6P accumulation appears to be important allosteric regulator of the glycogen synthesis. So that's thanks to Joan for initially actually proposing this and also inspiring my research on that. So that's, then I think you're asking also what about glycogen synthase GS2 in the liver form. So that's we immediately collaborated, and then there are a number of people, his team contributed this work. So that's we generated similar knock-in animals of glycogen synthase liver isoform for arginine 582 RNA mutation knocking animals, and then we could also demonstrate that the G6P plays a key role in actually accumulating glycogen in the liver. So that's actually, I then moved to actually the Lausanne in Switzerland. So I had an opportunity to take up a position at the Nestle Institute of Health Science. So that's, I shifted my focus onto LKB1 and PK pathway in terms of gluconeogenesis and then also glucose uptake. But Joan was always checking in with me and then occasionally invited me to actually IRB. So that's, I gave talks several times, and then last time I spoke actually here was 2019, and then you can see I was talking on glucose uptake in muscle, but he was still grateful for me to cover and discuss science. So that was very kind of him, as usual. So that's I switched the gear and then talk about my final piece of the book. So that's as already Philip mentioned. So that's the discovery of the reversible phosphorylation for biological actually regulatory mechanism. Awarded a Nobel Prize. So that's I had an opportunity to have a photo session with Ed. So this is actually Philip's milestone, achievement party in Grand Castle. So that's he was 92 years old, and so that's he was still very keen to actually know about glycogen metabolism. So that's as you probably most of you know, that Ed and Ed are working on glycogen metabolism. So that's he was working on the series of phosphorylation events in controlled hormone-mediated glycogen breakdown by muscle glycogen phosphorylase, also gene named PYGM. So that's so Joan was also asking, okay, so what about the degradation in people? And then that's I also made a promise to Ed that I'm going to work on this. So when I moved to Copenhagen, I revived my glycogen program and tried to study this. So, however, as also Philip mentioned, so Corey and Coley already had the discovery of allosteric regulation of the glycogen phosphorylase through 5'-AMP. So during muscle contraction, actually when ATP is generated by two molecules of ADP, so AMP is formed. So that's that can allosterically activate PYGM or glycogen phosphorylase. And then, as has been already described, so hormone epinephrine or adrenaline produce cyclic AMP, activate PKA, and then phosphorylate the glycogen phosphorylase kinase, and then that leads to phosphorylation activation of glycogen phosphorylase through N-terminal single serine residue of 15. So that's we are curious about what is the major contribution of this mechanism. So that's Devska actually took this role. So that's we took a similar approach what we did for glycogen synthase. So that's okay. Let's make the phosphodeficient mutation, and then also, if possible, make AMP-resistant mutant animals. So that's the latter part was not trivial because we didn't have such information. So the Elton, actually the Kiraj, who will be speaking later, has been extraordinary help. So he's my long-term collaborator and friend. So that's there are a multitude of structure available. And also this one is in the presence of G6P and AMP. So that's he helped me to design the the mutant which may be insensitive to insulin. So that's to the AMP. So we made a multitude of mutants and expressed in bacteria and purified them. So the first criteria is that enzyme is stable, and then these mutants all underwent the kinetic assay by AMP. So that's, as you can see, wild type. The EC50 is low micromolar, and then one of the mutation we intensely worked on is varin 46 to threonine. So that's, as you could see, we did a series of additional mutations to refine, and came up with finally to the to threonine mutation, which actually is activated at 50% after millimolar concentration, which never happens in physiological context. So also Elton was kind enough to be able to also crystallize this mutant in the presence of G6P, and also did a biophysical assay to demonstrate that this mutation doesn't change the G6P binding, however change the affinity to AMP quite substantially, also the consistent with also enzymatic data. And then with this basis, we made a knocking mutation of the mice, hoping this is AMP insensitive in vivo. So that's this is our scheme of making these animal models, and then do initial validation to see the enzyme activity, and then also do incubation work by hormone, and then also AMP mimetic, and so do the exercise running, and then do glucose tracer study in the end. So that's, it was important to show that this mutation for both lines did not actually alter the body composition or body weight or other general metabolic parameters. So that's, they also had the normal physical activity. So there was no overt phenotype actually detected under CHO diet, for example. So that's, we did actually incubation experiments with epinephrine. That's, we initially looked at protein expressions because sometimes a mutation causes instability of the protein in the tissue. So it was not the case. So that, as you could see, AMP-resistant mutation and then also serine RNA mutation, both actually expressed at the normal level compared to wild-type enzyme. And as you could imagine, that's, there was no phosphorylation because of the RNA mutation. So it was good to see that the protein is expressed in a stable way. So that's, we did kinetic analysis, like what we did for bacterial enzyme. So this is increased concentration of AMP. So that's, as you could see, you need millimolar concentration to activate this AMP-resistant mutation in muscle lysates. And so that's, if you mutate serine to alanine, in the absence of AMP, the enzyme is totally dead. So, however, if you actually increase the concentration of AMP at even very low, actually micromolar, then actually this enzyme is pretty much active, and then fully active around 100 micromolar of the AMP. So that's so this is kind of the ranges of the concentration during high intensity of the exercise. So that it seems the S2A mutation is functionally pretty much okay by AMP. So that's, we did a proof of concept experiments to see if hormone-mediated GS activation is actually altered in this animal model. So that we incubated with epinephrine and then look at pKa and then also glycogen phosphorylase kinase and then also PIGM. So that's, it is quite easy to see. This is pKa substrate antibody. Epinephrine increases both wild type and mutant. So as phosphorylase kinase phosphorylation and PIGM in wild type. However, of course, this is ablative. So in the absence of AMP, and epinephrine can increase in the wild type, and then which is not the case for the mutation. And if we look at AMP-resistant mutation, so there was totally similar in terms of signaling and also PIGM activity. So that's, there was no alteration in terms of phosphorylation-dependent activity in this animal model. So model have been pretty much validated. So that's, we wanted to look at the glycogen. So there are different muscle types. So that's typically there are fast twitch, the glycolytic fiber types, and then also slow twitch oxidative fiber types. So that's, this is a cross-section of the mice, and staying with myosin heavy chain. So as you go green, darker part, it's more glycolytic, and then only soleus muscle contain the slow twitch and oxidative fibers. So in any cases, so the gastrocnemius, EDL, soleus, all had about 30% of increase in glycogen. And interestingly, the serine to RNA mutation, phosphodeficient, had a quite substantial increase in all muscle types. So that's, if you look at actually at the same scale, you could see that the S2A mutation causes about 5-fold increase, which is quite substantial. And so that's, we wanted to consult with the two clinician scientists who are working on glycogen storage disease, macular diseases. So that's, they did analysis in terms of initially glycogen staining. So you can see that's the carbohydrate staining or PAS staining. You can see quite dense the signal in the S215. And there was no actually abnormality in terms of the morphology and also the size of the fibers. So, but if you look at the closely, there are kind of mosaic type of glycogen accumulation in muscle. So that's, and so that's Thomas did a counter staining with the myosin heavy chain. So as you kind of see, if you have a type 1 slow twitch, and then your glycogen accumulation is lower. If you have type 2X, which is glycolytic muscle, you have a higher glycogen. So there was no shifting of the fiber typing. And so that's, there was a proportional increase of the glycogen, in the both oxidative and glycolytic fibers. So that's also glycogen is not actually delocalizing in one place. So that's Joachim Nielsen has been studying this for decades. So that he has hypothesis that location matters in terms of function of the glycogen. So there are subcell colema and also actually also interfibril glycogen accumulation. So that's he did EM actually analysis on this. So that's he did entire quantification. So you could see some particles of the glycogen at different localization. And in ST15, as you can see that the particle size and chain lengths are dramatically increased. So it's very visually clear. So after quantification, then the size and particles are larger, but it's equally increased. So there was no distinct changes in particular localization of the glycogen. So that's with this, we wanted to see the functionality actually of these animals, and particularly wanted to see the glycogen degradation pathway in these animals. So that's we did a treatment exercise around 60% of max speed for 6 hours, because 1 hour, because we knew that this deplete about 50% of the glycogen. So as you can see, 3 different muscles, wild type deplete nearly 50%, and there was a modest increase in the basal, but the glycogen depletion was quite similar. So this is a delta from resting to exercise. So that's all 3 muscles had similar decline of the glycogen in both wild type and knock-in. So that it seems this mutation did not affect glycogen degradation. And this condition, as we checked the AMP levels increased, and AMP activation was also increased. So that's actually the exercise regimen was sufficient to increase the AMP at least in concentration. So that's then actually we also did a similar experiments using phosphodeficient. So this is 2 muscles. So that's you can see that the wild type had 50%, as you already show. So in the knock-in mice, there was no decrease in the glycogen degradation. But however, this is tricky to interpret, because since you have burst, high glycogen, the glycogen architecture might have changed. So the stoichiometry of the glycogen phosphorase to also recognize the glycogen particle will be different. So that it's difficult to interpret this data. So that's we spent additional year to actually close with the inducible muscle glycogen knockout, actually kindly donated by Joan and Jody. So that's we induced actually a glycogen synthase knockout for 6 weeks. And we could nicely reduce profoundly, closer to wild type levels. And did the exercise, exercise, so still there was no decrease. So, but also one thing I want to point out that this means that there is substantial glycogen degradation in the absence of glycogen phosphorase activity. So this means there has to be proteasome or actually the glycophagy happening to degrade glycogen. So this may be important thing to look into. Nonetheless, so this, even you kind of normalize glycogen content, these mice are almost deficient in degrading glycogen. So that's we are now doing a C13 trace analysis to look into where glucose goes, and then what's the actually the substrate utilized during exercise. Because if you cannot degrade glycogen, you need to use either fatty acid or glucose to be able to sustain exercise during exercise, to have repeat ATP. So that's so I talked about 2 contexts of glycogen synthesis and also degradation during exercise. And then so the first story I articulated that glucose 6-phosphate is important physiologically to stimulate glycogen synthase, inspired by Joan. And on the other hand, for the glycogen degradation, the phosphorylation-dependent activation is important for both basal and exercise context. So it's an interesting actually aspect to look into reversible phosphorylation versus metabolite regulation in physiological context using this type of knock-in technology. So that's, I just want to wrap up. So that's, this was mainly done by Dipska, who is a postdoc, a researcher actually in my group. And then I already actually acknowledged numbers of key collaborators who did a really nice work. So that's, I would like to also finish my talk, actually thanking Joan again. So that's the 2 elements apart from his outstanding scientific mentorship. So that's his passion and curiosity, love for science is really infectious to everybody. We can see the energy, of course, today. And of course, I think, he is such a, great, person in terms of his hospitality, leadership, and also mentoring that you could see, from the point of view of family, colleagues, and mentees. So everybody says he's such a nice person to care, every single people who are involved, to work with him. So that's a very important component. So that's also, this has been said, Joan normally likes to use the analogy of glycogen, the good and bad and ugly. So glycogen can be good, but could be bad or ugly. So our promise to Joan, I think entire glycogen community should be, dearest Joan, that's we will keep glycogen as a good, not the ugly. So that's a hope we can achieve this in the next 20 years or so. Thank you very much. Yeah, thank you.
Time for question.
I just need one. That was terrific. Thanks. So two questions.
Yeah.
So the first thing, in the PGYM mutant that you have that's not susceptible to degradation, what happens to the glucose uptake in those livers, in those muscles?
Yeah, so we measured both insulin-dependent and AMPK-dependent glucose uptake, and then which was normal.
Contraction dependent?
So contraction, we haven't done that because I was surprised because there are papers showing also glycogen content is important component. So I hypothesized that hyperaccumulation of glycogen would lower, but in mouse skeletal muscle that was not the case. So we do more study, and also we have also GYS1 knockout animals to deplete glycogen. So we try to look into lack of glycogen versus hyperglycogen to see the insulin and in NPK responses for the glucose uptake. So that was quite important component. Also, somebody asked the question. So that is the data so far.
Okay. And then getting back to the first part of your talk,
Yep.
I'm really glad that you pointed out the deficiencies in the GSK3 model. And we saw this in fat cells 25 years ago with Matt Brady published this. And then in liver, glucose uptake is not increased by insulin.
Right.
Of course. So I think so that's a second thing. And then I think the third thing is that rapamycin really blocks insulin-dependent glycogen synthesis. This was shown by John Lawrence. We reproduce this many times. Other people have seen this. So I think we still don't understand the mechanism. So I'm just wondering if you have any ideas. I think there are transcriptional events. We keep thinking this, in our old ways of looking at it, that there's a short-term effect and it can't be transcriptional. But I'm not sure about that. So I'm just wondering what you think about the mechanism.
Yeah, so I also thought about this. Also, I think you also nicely showed also PTG, actually regulation by mTOR pathway, and then also we had also a bit of paper with Paul Tichel, you probably saw, with the JCI. You also commented this was not new, and also he showed that mTOR-dependent PTG and also regulation in that context. So what has been difficult is that, as you also point out, since actually glucose uptake doesn't increase, and then it is more to kind of the hyperglycemia, GLUT2, and then so that's. But also, if you try to do this in hepatocyte context, also it totally changes. So we did also kind of the glucose flux analysis, meaning using a C13, C14 glucose to look into the mechanism of how glucose is utilized under different contexts. However, since as soon as you isolate hepatocytes, you alter gene expression or protein expression of many enzymes. I don't have a clear picture as yet, so I think we need to do more extensive, perhaps possible proteomics analysis or whatever to see the alternation of the key enzyme, and also coupled with the tracer study to look into that's the glucose tracing to see where it enriches and different fasting feeding condition. Yeah, if there are funding available.
So here there is one question. Two questions. Very quick, please, Antonio.
I'm gonna be quick. Okay. I just want to ask you whether in your S15A experiments, whether you notice differences in the exercise performance in your mice under conditions in which there is no glycogen depletion, and whether you measure ATP or creatine phosphate.
Yeah. Okay, so that's very interesting. So that's so the exercise performance we did 60%, 80%, and so it doesn't seem to change much. But I think it has been to some extent known by also Peter Loach's so glycogen is not a limiting factor for exercise in mouse. not in human, because mouse only have 10% of glycogen compared to human. We measured actually nucleotide extensively. Interestingly, AMP was increased. I was expecting to be really reduced. So now we are looking into mitochondrial functionality, also respiration, to see if there are changes in the mitochondrial function. Because, as you could imagine, since glycogen cannot be utilized, mitochondrial function could be altered. So we would like to see if there are any changes in mitochondrial functionality, also coupled with this nucleotide changes. So you maybe help me in terms of mitochondrial part, actually. Please, Antonio. Yeah.
So I think we need to move now, because if not, you're not going to have time for lunch.
Everybody's
so thank you so much.
Yeah.
Also, a little bit of information. Lunch is going to be outside where the coffee break was. The next session is going to start at 3 p.m. So see you later. Thank you.
So I'm enjoying it, yes.
No, no, I have. Okay, yeah. Hello, good afternoon. We should start if we want to finish on time. Okay. Hello once again. My name is Jerka Dumic. I'm coming from the University of Zagreb, but here I represent FEBS. And it was great privilege of mine to be able to contribute to organization of this event jointly with Alexandra, Francisc, Antonio, and Matthew. We did our best to make a good program, and definitely Joan deserved that. So I think that we can be all proud on all of us, but mostly on Joan and his legacy. So as a common biochemist, I'm not in the field of glycogen. I know that the postprandial glucose level is going up. So hopefully our body will do a good job and store our excess of glucose. However, if our attention starts getting stored, not used, it will be quite challenging to be awake. However, we have 5 fantastic speakers, and I'm sure that they will keep your attention alert. So first of them is David Commander. So please, David, the stage is yours.
Thank you so much. Yeah, look, I'm new here, right? I would like to thank the organizers for having me at this meeting. And I thought because I have very little connection to this field as a whole, I will just briefly introduce myself, and then you will sort of see where we're going. So I work at the Institute WEHI in Melbourne in Australia, and I'm running a ubiquitin signaling division there. We are interested, so the nice thing with ubiquitin is that you can work on anything. And we are interested in inflammation, neurodegeneration, particularly Parkinson's disease and cancer. And as a medical research institute, we are focusing on human biology. I am super excited to be here. I'm a newcomer in this field, thank you very much for this invitation. I'm just sitting there and I'm taking a lot of notes to really learn here. I'm a student here, right? This is very important for me to understand how you think, what your problems are, and what we have missed. What we have developed is a method to detect ubiquitinated glycogen in cells and tissues, and that's what I want to share with you today. And there's so many questions, right? And I really would like to understand where our interpretation of the data does not make sense. And that is really important for me. And again, I think it was fantastic to be reviewed by experts to really set us straight on this journey. And I think that's really important for if you are a newcomer in this space. And again, my model is still rapidly evolving, so you will see that later. And then again, another thing to say here is obviously that I've been at meetings like this before, and everybody shares a story about the person that is the legacy of whom is celebrated. I don't really have a story to tell here because I didn't have the chance to meet Joan. Or so I thought. And then last year what happened was, I'm traveling to ubiquitin meetings. I travel a lot, and I met, new people all the time, right? I mentioned the fact that we work on ubiquitinate glycogen to somebody I hadn't met before, and he just said, oh, I worked on glycogen during my PhD. And then he looked at my email, half an hour later I'm getting an email basically saying, hi David, I read the abstract of your talk, super cool. I used to work on the E3 myelin during my thesis, and he shared the link. David Vilches. So he's now working in the protein homeostasis field, and I'm sure many of you know about him. it's always interesting to see where people are, right, when they come from a lab. And that's always a very, very important sign of a scientist who have say, what's the legacy in terms of the people. But then the other thing that I found remarkable, I remember that my PhD advisor always asked if glycogen was ubiquitinated. I'm very happy that this has been proven. I wonder if the Laforan-Malin complex also promotes ubiquitination of glycogen, right? So this was beautiful because I really felt that we can indeed discuss that today. And, I emailed back to him, I recognized that last also on the paper, and I was just invited in this memory, maybe by a reviewer. Anyway, so let's start this. So ubiquitination is something that over the last 2 decades or so has become very, very complex. There's many different types of ubiquitin chains, many different types of branched ubiquitin chains, and these are proteins these balls here are proteins, not sugars, okay? So, we basically have contributed to making this a very, very complex and very powerful regulatory system that we have in all of our cells and that regulate essentially all aspects of biology. We've reviewed the structural biology perspectives. There's I think 250 structures in the PDB with ubiquitin in it. And that if you want to get up to speed on this, that's probably an interesting place to start. But what what you will associate with ubiquitination is a protein modification, right? We all know that ubiquitin is attached to proteins to degrade them. That's the textbook 101. Let's just forget about all this, because what you will learn about today is the fact that ubiquitin is not just attached to proteins. If you look at the chemistry, that's a lysine, it's ubiquitinated. That's the serine and threonine, that's ubiquitinated. Philip showed this earlier today. So this is protein modifications that we know about. But if you look at these chemical groups that are ubiquitinated, they exist in other molecules as well. This is lipid A in LPS. Now that OH group was suggested to be ubiquitinated. Philip already discussed his work on maltose ubiquitination. Phosphatidyletanolamine can be ubiquitinated on its amino group. So these are the 3 papers that sort of started this field of non-protein ubiquitination. And since then, there really has been an explosion with very high-profile papers that are effectively suggesting that a lot of molecules can get ubiquitinated. So all of these papers here on the top have shown this in vitro. And I'm not super excited about that at this point, because in vitro we can ubiquitinate anything. Okay, so we are a ubiquitin lab, and I can ubiquitinate any molecule you give me with the right, tests and stuff. These papers are a little bit more interesting in a way, because they really show literally a Western blot, 1 Western blot in the paper, or 2 maybe, where they effectively say, here's a Western blot, that ubiquitin has now been shifting a little bit up the band, suggesting that it's coming from a ubiquitinated non-protein. And phospholipids, the same story. There's a couple of Western blots showing that's a clear species that in cells is a ubiquitin that clearly has been shifted up, showing that it's lipidated. And then more recently, there were some interesting papers on ADP ribosylated proteins that are ubiquitinated on the ribosyl group. And also drugs are ubiquitinated if they have an amino group, for example. A very interesting 2 studies here, and they've shown this in cells. So the question that you have to ask is, just how big is our system, right? I mean, if we now talk about molecules that are ubiquitinated, and in this review, very short, 2-page review, we discuss it quite critically and basically say, can ubiquitinate and does ubiquitinate, that's a big difference. But also we point out that we are completely blind towards non-protein ubiquitination. There's no mass spectrometry method to look at this. There's no test. And what is completely unknown is the scale and the relevance of these modifications should they exist in cells. So what's missing is a sensitive and quantitative method to detect non-protein ubiquitination in cells, and that is what we wanted to generate. And this is basically the method. That's basically the idea, right? If you have ubiquitin attached to a small molecule, then we can take the ubiquitin off in a particular way that we remove, we keep the C terminus of ubiquitin attached, and we generate GG-modified small molecules that we should be able to detect by mass spectrometry. That sounds very straightforward. It took these two gentlemen the last 4 years, so both of them worked on that together, and they didn't do anything else. Marco has just finished his PhD, and Simon is our mass spec expert. He's a senior mass spec expert, and who developed, both of them developed the methods that I'm going to show you. The trouble is that we are looking at proteomic quantities of non-protein substrates, for example, metabolites. So that's a bit of a problem because we have much less of proteins in a cell compared to many of the metabolite species that we're looking at. And there's no methods to do this. So we developed this with what we call ubiquitin clipases and with sortase, and I'll show you this on the next slide. So what is a clipase, UB clipase? So this is a name that we gave an enzyme that we discovered in 2019 or described in 2018, 2019, which was a viral deubiquitinating enzyme that cleaved ubiquitin, but it didn't cleave it cleanly. So it basically left two residues attached to the substrate. But it doesn't really remove anything else. So it only removes ubiquitin, so the proteome stays untouched. That's important. So it generates effectively out of the ubiquitome, it makes a GGome. Now we have small extensions to wherever ubiquitin was attached. This was extended by a study from Kai Hoffmann, who basically showed that the same activity can be found in bacterial effectors. And the enzyme that we're using now is this enzyme BP-JOS, which is a super good, an amazing clipase from bacteria that we use. So what is sortase? Sortase is another bacterial enzyme, sortase A, that is a transpeptidase. So you can give it a peptide, and it loads the peptide onto its active site. And then if it finds an N-terminal amino group, an N-terminal glycine amino group, it will discharge the peptide onto that N-terminal amino group. So it's basically adding a peptide onto onto a stump. So we are generating GGs in this method, and these can now be extended with whatever we give it as a peptide to do this. So it's basically a perfect complementation for these two methods. So here's our workflow, and I'm just going to show you this once, but it's hopefully is self-explanatory. It looks complicated, but it's actually not. We take a cell lysate, and we filter the cell lysate to get rid of all small molecules. We are then having all of the proteins left, including the ubiquitinated small molecules, and then we clip. When we clip, we release new small molecules where the ubiquitin was attached to whatever it was. And now we filter again. We throw away all the proteins, but what comes through the filter here are small molecules that now have a GG molecule attached to it. So here we then had these sort of soups of small molecules with GGs, and we wanted to do metabolic mass spec. And we realized that method wasn't sensitive enough. We didn't have enough material to look for these proteomic amounts. And that is where the sortase came in, because now here, so this doesn't work here, but here what we now do is we are extending this GG modification with a peptide that we define, that we call a clip tag. And now what we are dealing with is actually a peptide with a small molecule attached. And now we can use normal mass spectrometry, high-resolution mass spectrometry to effectively do cleanup steps, HPLC, and we basically get a 1,000-fold sensitivity increase. So that is the method in a nutshell. We can do this now in targeted ways. Okay, so the way we developed the method is that we basically made standards, and we added the standards into our cell lysates. So here we basically take a maltose, the ubiquitinated, then we add it to the cell lysate, and if the ubiquitinated GG maltose can be clip tagged, we can see it in the end, right? We can do this with heavy labels, so we can quantify. And that's how we develop the method. So let's look at glycogen ubiquitination in cells. We've already heard about glycogen ubiquitination. We were intrigued by these two papers that came out that discussed HOL1, RBCK1, that showed when you have mutations in this protein, you start to see polyglucosan bodies in brain. And then Philip's paper went on to show that HOL1 can ubiquitinate glycogen in vitro. We already heard all about this. In neither of the two papers, as Philip also mentioned, was there any indication that Hol1 does ubiquitinate glycogen in cells. So that really is something that we wanted to look at. My student basically said, okay, if we want to do this, if we want to optimize our chances to see this, okay, sorry, no. First of all, let's show you that we can very nicely reproduce Philip's data, as of course, you would expect. This is glycogen, sigma glycogen. We ubiquitinate it with fluorescent ubiquitin. We can then amylase treat this, and you can see that the ubiquitin smear is sort of getting smaller here. We can clip this, and this is our clipase reaction. So now the glycogen stays untouched, but the ubiquitin has gone, but it actually hasn't gone because the GG modifications are still in the glycogen. And if we put both together and we add our sort tagging step, we can now see it on the mass spec. And we can see this as clip-tag maltose. So clip-tag is a peptide that we add back. Clip-tag maltose, triose, tetraose, pentaose, etc. So these are now from in vitro modified glycogen digestions. So Marco said, okay, we need to optimize our chances that we might be able to see this species in cells. And what he developed is a recruitment system where we take a liver cell line and we are just taking a CBM20 domain, GFP, and FKBP, and then we bridge that through rapamycin with FRB, cherry, and Hol1 to recruit the ligase directly to glycogen. So it doesn't mean that, okay, so here's what this looks like. This is the CBM20 signal. It's diffuse in these cells. When we add rapamycin, we start to see dotted structure here, these granular structures. We've recruited the E3 ligase, and now if we look for ubiquitin, we can see that ubiquitin is now heavily enriched in these structures. This is dependent on the ubiquitin system, so this is the E1 inhibitor of ubiquitin, so ubiquitin E1, and if you have that, then there's no more ubiquitination, and it's also not just an effect that rapamycin can do itself, so it needs to have both systems present. We can also do that in a cell where we have overexpressed GIS1 or GIS2, doesn't matter. And again, we see already dotted structures in these CBM20 domains without recruiting, but when we recruit, we get the ligase into these structures, we get ubiquitin into these structures, and effectively that's the same experiment. TAC243 is essential for ubiquitination to happen, and nothing happens with GFP alone. And now, when we take these cells that have been treated, so these are the first set of cells, we now see exactly the same species that we had. So none of this experiment showed that this was glycogen that's ubiquitinated, okay? But now we look at whether it's glycogen that's being ubiquitinated in these dots, and absolutely, because we get exactly the same species that we get from our in vitro preparation, where the ubiquitin had been added to the glycogen molecules. Great, this is the first time we've observed ubiquitinate glycogen in cells. We can play with the system. HUH7 cells, there's low glycogen. We have to have a ligase recruitment. We use SCOF3 cells, and I'll discuss them a little bit later. We can detect endogenous ubiquitinated glycogen. We can metabolically label, so you feed 13,6 goose glucose, and we get a ubiquitinated heavy glycogen. And more glycogen means more ubiquitinated glycogen. So let's ask the question whether we can see this in an animal. This was so far a very engineered system where we basically just force the system. This is just a total. So we worked on this with muscle experts from University of Melbourne, Paul and Craig. They're metabolism and muscle people, and they looked at ubiquitin. This is basically just total glycogen blot. Again, nothing that will surprise you. Looking at different tissues, but what I found super surprising already is that we saw ubiquitinated glycogen in any cell type in a mouse that had glycogen. So we can easily detect with our method, and we had a huge amount in liver. Again, I didn't really expect that. We saw the muscle results first, but that liver really was so much ubiquitinated glycogen, that was surprising. We also had some human samples, and we can see it in human muscles, so it's not just a mouse thing. And I come back to that. So that's an observation, right? I haven't told you about quantification of this anything, but really the question was, what would happen if we turn this into ideally for me, it's like observation is not relevance, right? So relevance for me comes when we can regulate it. And we did the very simplest experiment we could think of. We basically did a fasting experiment. 6 hours fasting, 24, 48, and then fasting and refeeding. Here's the experiment that you would expect. This is total glycogen. We just looked at liver here. Glycogen after 6 hours starvation goes down to about 20%. This is an experiment that really surprised me because at 20% glycogen levels after 6 hours fasting, ubiquitinated glycogen goes up. Okay, so this is not normalized. So effectively, that low amount of glycogen had now more ubiquitin attached than under the normal conditions where we already saw quite a lot of ubiquitin. This is crazy. This means that during the process where glycogen goes down physiologically, ubiquitination is increased. That really was super surprising. On the other side, when we refeed, we basically start to see glycogen going up, but these are straight glycogen, these are straight sugar balls that come up. Ubiquitination seems to be lagging behind after the refeeding, and then it takes about a day to equilibrate the glycogen signal. Okay, so we wanted to quantify this. And the way we quantified this is on the ubiquitin side. So we wanted to see, we made we took heavy ubiquitin, which was like, just labeled C13 heavy ubiquitin, and we, 15N actually, 15N ubiquitin that we generated in vitro these species here, quantified them, and then we added them to the reaction to understand how much ubiquitin has been added to these species. So this is a standard curve here, and we are looking at the same animals from like 2 animals here, so basically 6 hours fasting and 24 hours fasting. And these amounts of ubiquitin now that we detect here, the picomole of ubiquitin in the 6 hours fasting condition in the maltotetraose species. This is triose, this is maltose, so 3 different things. So you can add this all up to get an idea of how much ubiquitin was added during this process of fasting. 1.3 picomole in total. These mice have 100 picomole of ubiquitin in total. So this is a very, very large amount. This is the same amount of all ubiquitin chains that are locked up, for example, in these K11 chains that Philip mentioned earlier, in total cells. There is maybe histone ubiquitination is more abundant, but that's the only other chain. This is the only other attachment point that we know for ubiquitination. So this is crazy, right? As a ubiquitin person, this is crazy, because now I'm telling you that during fasting, about 1% of total ubiquitin is becoming attached to glucose, to a sugar, in glycogen in the liver. So this morning when you woke up, a lot of your ubiquitin was attached to glycogen overnight. This happens during glycogen breakdown. The question is, does ubiquitination cause glycogen breakdown? And again, initially we were a little bit more going out, but here we are sort of now. So now I'm, I can't believe I'm telling you how this works. But anyway, so this is, apologize for these super simplified slides. You are the experts in the room here. But effectively, we don't think that it's necessarily in the normal glycogen catabolism space. But what we were more interested in is glycophagy. Because ubiquitination is such a well-established signal for glycophagy, for autophagy processes, that you would effectively have a beautiful thing. And I don't need to tell you about glycophagy. There's GLUT transporters on lysosomes to pump out glucose that is generated here. Pompe disease is a GAA modification, so we know that this is physiologically and pathophysiologically important. And there's this interesting protein STBD1, which is so far the only reported or suggested glycogen binding domain and LEA glycophagy receptor. This is supposed to be ubiquitin independent. This is not going through ubiquitination. So we were interested in this pathway and wanted to study this further. How important is glycophagy? That's a really good question that I've tried to find out from you in discussions. And it's I would love to have the answer for that. Bufalomycin treatment removes changes the pH in lysosomes and increases glycogen levels 2- or 3-fold when you add it to cells. This is our data, but it's also published. FIB200 is a very important protein in normal autophagy. When we knock it out in HeLa cells, we see more this is more glycogen. And then this is data that was published from this Nature Cardio paper last year, where they STB1 preferentially binds to GABARAP-L1, and that I think is pretty good. But GABARAP-L1, knocking out one of the GABARAP-L1 alleles seems to be increasing glycogen in heart. So, glycophagy is a real thing, and you already know that. At this point, I was really nervous because what we had here is a system which depended on rapamycin, and rapamycin has so many effects on autophagy. So I asked the team to just completely switch the system, right, because that clearly wasn't going to cut it when it comes to looking at autophagy. And we switched to a different recruitment system, which is just 2 different proteins here that can be dimerized by a chemical called this Mundy system. And it doesn't matter how we do this, we always get nice recruitment. We always get nice ubiquitination of glycogen. But now what we could do with this system, or what we did with this system, is to effectively do a bit more imaging to understand whether these structures that we saw, they are GFP positive, meaning that they are CBM20, so this is glycogen. They recruit the E3 ligase, they are becoming ubiquitin positive, and now they also become LAMP1 positive. And when we bufalomycin treats the cells, this effect is strongly enhanced. This is quantified here, where we basically start to see a lot of co-localization of ubiquitinated glycogen particles with LAMP1, suggesting that they are co-localizing with lysosomes. This depends completely on ubiquitination. This depends on the activity of the E3 ligase that we recruit. This is definitely not endosomes. This is an endosomal marker, so we know that we are looking at lysosomes. And then, we are this is in progress here, where we now look at autophagy genes, which autophagy genes are important, and we here you can see very nice P62 staining, and we've already seen this morning from Pascal Sanz. VP2 is important, so VP2 goes to these structures. ATG13 doesn't seem to go to these structures, and that's a very important kinase component here. And this is why I keep a very open mind about what's the machinery here that we invoke. Okay, so glycogen storage diseases. Another thing I don't have to tell you anything about because you have developed these. The question was, is ubiquitinate glycogen dysregulated in glycogen storage diseases? So here we basically wanted to do this, and we used these COF3 ovarian cancer cells. We thought we were very clever, but I'm not sure we were. But basically we used this cell line from an old paper which was shown to have a lot of glycogen in these ovarian cancer cells. We were asked by the reviewers to just show whether this is actually normal glycogen, right? And for this, we did, first time in my lab, we did EM, and we saw these beautiful, I find EM amazing, right? We looked at these beautiful structures, and you can see what was a textbook version of, my student says, a textbook version of these beta particles, which supposed to be, and again, I'm telling you this, you tell me what this is. We knocked out GBE1, and we saw all of these very, very different glycogen structures. These are some gamma particles as well down here. So, and you can also see how the membranes are really messed up here. So, ubiquitination of these glycogen structures is completely different, right? So first of all, we have much lower glycogen in the GBE1 complete knockout. And that little ubiquitinated glycogen is 30-fold more ubiquitinated. So the difference here is 300-fold. Again, Philip showed the same data this morning, showing that you really get, when you knock out GBE1, you see a 300-fold increase over a wild-type situation for ubiquitinated glycogen. And now we have a system that we can play with. Again, we can now take out E3 ligases. This is a knockout of HOIP. Philip explained it this morning, and we can see a strong reduction in this GBE1 knockout when we take out HOIP. So we start to see enzymes now which are involved in this process. Okay, so here's a funny experiment because it's in the paper, but not with potentially all the information. Actually, we got four human muscle samples from Catriona McLean at Melbourne, at Alfred Hospital in Melbourne. And again, we could easily detect ubiquitinate glycogen, but these were actually patient samples where we have 2 controls and 2 McArdle disease patients. They are blinded, so I cannot tell you which of the samples is which, okay? I really don't know. But we have 2 samples which are down here, and we have another 2 samples, one is here and one is sort of off the scale increased. So effectively, that already indicates, to me, this could indicate that potentially there is a change here in a human muscle sample on glycogen as well. But again, we have to unblind the samples to really say that, right? So one thing we realize is that we are probably looking at 2 very, very different scenarios here. First of all, we have what I've shown you with the fasting experiment, is that we are looking at a physiological process where ubiquitination happens during glycogen breakdown with soluble particles delivered to lysosomes. What is the machinery? What are the chain types? I don't think we have this yet. The GBE1 knockout situation could be really quite different because polyglucosan bodies are obviously very different from normal glycogen, insoluble, and they are very, very highly ubiquitinated, which our data shows time and time again. Many of these particles do not go to lysosomes because we think because they are too big. So the question is, and these are questions for you, right? Can ubiquitin interfere with glycogenolysis? We don't know yet. Same machinery. Is that the same E3 ligases that put ubiquitin on? Is it the same chain types? That's going to be interesting. And you could really think a little bit about this as a quality control pathway for abnormal ubiquitin, abnormal glycogen, rather than what's happening up here for the wild type situation. Okay, I want to show you 2 more pieces of data which I find a little bit striking. This is otulin knockout. We published the otulin liver knockout. Otulin is a linear ubiquitin chain, a deubiquitinase. So it can only cleave the chains that Lubach is making. And you can see in the stain that these livers in these animals had no glycogen. Okay, so that's something we commented on in the paper, but we couldn't understand it. Indeed, what I can show you now is that this data from the mice did not match what we can see in cells, which is very surprising because in cells we knock out the DUB, the deubiquitinase, we see much increased ubiquitinate glycogen, but the glycogen doesn't really seem to be going away. So we don't really know what's going on. This is in the Scoff cells, but these are wild type cells. Okay, so some models, we know that glycophagy has to exist somehow because we need to get glycogen into lysosomes. We now add ubiquitin to the mix. We already understand some proteins that are involved in this process, and we think that this is a ubiquitin-driven process. The question is ubiquitinate glycogen still the same substrate for glycogenolysis? And again, this is sort of where my structural biology mind comes to play, and I'm really having fantastic discussions here. Which of the ubiquitin code is involved? Philip discussed already the ubiquitin code this morning. But these linear ubiquitin chains, we know them as very, very inflammatory signals, right? So I've just shown you like aggregates or puncta and cells, which now will be full with linear ubiquitin chains. And we know that other places where linear ubiquitin chains are heavily involved, they trigger inflammation. Right? TAC1 complex, NF-kappa B signaling, all goes through these chains that Philip and I now discussed. What are the E3 ligases? We don't know. We know that Hoyle can do it. RNF213, there's another paper in circulation suggesting it can do it as well. But we believe there might be other ligases as well. What are the deubiquitinases that removes this? And then finally, I showed you that just ubiquitination is enough to phase separate these particles. They are highly ubiquitinated, but they're also very persistent. And in live cell imaging, these things do not really go away. So potentially, and that's a very recent idea, maybe this is an interesting analogy to stress granules, which are effectively mRNA storage capacities. So I'm really wondering if there's some long-term storage compartments of glycogen possible here as well. Okay, so I've shown you that ubiquitin is a new component of physiological glycogen regulation. There's ubiquitin-dependent glycophagy. The question is, why has this not been discovered before? I think it's a very labile bond between ubiquitin and glycogen. And you know, everything above pH 6.8 is going to take it off. Many of the glycogen biochemistry protocols that we've looked at would remove the ubiquitin. Many of the mass spec protocols would remove the ubiquitin. And you know, glycophagy is relatively recent. And then I think in the ubiquitin field, we are really pretty good at putting ubiquitin on, taking ubiquitin off. And think about PROTAC drugs, right? PROTACs are effectively where we are doing this for proteins. So what happens? Can we just reduce the amount of glycogen by just recruiting E3 ligases? Yeah, 25%, that's not a lot yet. And again, my mind here is relatively wide open about, at the moment, what we actually have to do to these species to actually induce them. But I think it is a very exciting idea that we could direct, via the ubiquitin system, glycogen or particular pool of glycogen towards the lysosome at will through interfering with this. So I'm going to stop here, and I'm going to switch all the other part. I wanted to show you that we have many other metabolites that are ubiquitinated, including triglycerides. They are all going to be in the paper. Triglycerides, when you feed cells glucose, you get ubiquitinated glycerol. You can find ubiquitinated glycerol. So this is a metabolite. Okay, that's very important, I think, as well. Increases in livers after fasting. Spermine is another one we found. And you know, there's a lot of things to do for us. Happy to discuss any and all of that. Ubiquitination is really big, right? That's the conclusion here. We have to update our wording. It's not a post-translational modification of proteins. It's a cellular modification. I've shown you data on carbohydrates. I haven't shown you data on lipids. I haven't shown you data on drugs, but we have found those things in cells very easily with our new method. And we now can do this in a way, and it will keep us very, very busy. And I think, and again, this is a very big statement here in this crowd in particular, but I believe that there's, this catabolism is probably not the right word. Metabolism is probably better. Yeah, I will leave it here. This is my lab at the moment. The green people have contributed to the study, and everybody now works on non-protein ubiquitination. We've completely stopped working on protein ubiquitination. Thank you very much.
Thank you. Thank you, David. So first, thank you for staying on time, and also to left something to make the lecture even more intriguing. So, so we have time for one short question.
Have you looked at the glycogen from various tissues, from Lafura, about Lafura knockout and Mailing knockout?
No, we have not done this yet. And we are quite far away in Australia, but just one FedEx. So I'm very, very happy to look at this.
Well, we can send you tissue from all these mice, and that is easy. No, we don't need to send you the mice, just tissues.
No, that's right. And I think this would be super fun to do, right? And again, I think I would just like to, at the moment, we are just still scaping the landscape, right? What can we see? What should we look at? And so on. And there's so much to do for this. Yes, absolutely. I would love to see these samples. Thank you.
Thank you. Okay, we have to reviewer number 3. We have to allow him a question.
I didn't invite him, though. No, is it mammalian specific? The phenomenon. Could you see it in plants?
That's a great question. And non-protein ubiquitination, I'm very sure would be. My guess is yes. We looked, no, right? But I think the next. Yes, I would love to look at plants. I'm sure this is happening in plants. And plants is very interesting because they have a really big ubiquitin system, very heavily expanded. But if you look at ubiquitination sites, it's very, very small, actually. They haven't really been able to find many ubiquitination sites in plants. So I really think there's something funny going on there, and our method might sort this out. There is the yes, very rare. Yeah, and there is a paper from there's a paper from Osumi who's looked at this in yeast from the autophagy field. And in yeast, I mean, the lipid ubiquitination, for example, that was done in yeast. So it's just we haven't had the method. So
thank you,
awesome,
David.
thanks.
Okay, so I'm really pleased to announce the now next speaker, Matthew Gentry, who really contributed to setting up the program. So please, Matthew.
Yeah, just while he sets up, a thank you to IRB, to IUBMB, to FEBS, to Laura and her team for getting all of us here. Why don't we give her and the team a round of applause for... Thank you. This one here. Is that me? Yeah, perfect. So I first met Joan 20 years ago. I was a postdoc with Jack Dixon, and met Joan and Pascual and some of the other Spanish scientists at a meeting. Quickly became, I met him as a competitor, quickly became a collaborator, and then a friend and a mentor. And what I learned about Joan, and also those of you who know Jack Dixon, both outstanding scientists, both incredibly kind men, and both very forward-thinking and ahead of their time. And I think you've heard that today repeatedly. And so I actually changed the title of my talk to this, because I think a lot of the times of what we're seeing is what we're discovering, Joan kind of already did it 20 or 30 years ago. And you'll see that a couple of times through what I show you today. So just a couple disclosures company-wise and foundation-wise. One thing I wanted to do for this talk was to look back at Joan's papers. He's published over 180 papers. So I had a student grab those, put them into ChatGPT 5.4 to have it summarize all of his work. And the summary it came up with was very impressive. So you have the scientific arc of physiology to mechanism to disease. We've heard a lot about that. We've heard today that glycogen is not just an inert fuel, but Joan said it's a regulated, localized, we've heard that theme today, biologically consequential system. Alan talked about it being a signaling molecule itself. You look at the three phases of his career, this is GPT's words, and you can envision Joan when you read this creative flair of old school physiology, ahead of his time mechanistically, and from mechanism to disease. The ones that really jumped out to me was this idea of he took frogs and was studying glycogen in frogs because they have a seasonality to them. This was before knockout mice. And so he was trying to look at how was glycogen being regulated in this seasonality changes in the frogs. He looked at glycogen localization, whether it was in the nucleus or in the cytosol, very much ahead of its time, and then really defined the fact that brain glycogen does exist and is important, and you heard that from Jordi today. So what I'm going to tell you about today is a technique that we've used to look at glycogen, to rediscover some of what Joan has taught us over the last few decades. Many of you know about single-cell RNA-seqs, spatial proteomics. A more newcomer to this scene is spatial metabolomics, and this is a technique we utilize a lot in my lab, University of Florida, in collaboration with two close collaborators there. So what we can do is take a tissue from, take a tissue sample from any tissue. We're kind of tissue agnostic. This is showing a brain here of a fresh, frozen, or a formalin-fixed tissue. We can spray a matrix on that, and then through a series of steps here, we can get the small molecule metabolites, some lipid metabolites, and in-linked glycans. And the way this works is you spray matrices so that the biomolecules can be exploded essentially when a laser rasters across the tissue sample, as you see here, the laser going across the tissue. And as the laser goes across, you're capturing essentially an LC-MS spectra of all of these biomolecules. So what you have then is an XY coordinate for every one of these biomolecules. And you can take that information then and spatially depict that with these heat maps. So in the heat maps, the hot colors are high levels of this particular metabolite, and the cold colors are low levels of this particular metabolite. So when we do this technique, this came out a few months ago, we get about 30 to 50 different small molecule metabolites. We get about 200 to 300 lipid molecules, and then a few thousands of these N-linked glycans. So for every biomolecule, we have a different heat map. So it then becomes a data processing and machine learning and computer science problem of trying to figure out what does the changes in metabolism tell us about the specific disease that we're looking at. We can get near single cell resolution from this. We can tune the laser down to about 5 micron. We run most of our experiments at 50 micron resolution. So I'll just tell you one story about the glycogen storage diseases that this technique has informed us about, and then tell you another more recent story about lung cancer and the role of glycogen in lung adenocarcinoma. So you've seen the Lafora disease mouse model a number of times. This is using an anti-glycogen antibody to visualize where that glycogen is. And you can appreciate here that if you're thinking therapeutically, you've got to hit every region of the brain. Jordi and Pasquale told you have to hit both astrocytes and neurons, but you see the Lafora bodies in the hippocampus, the cortex, the cerebellum, all the way into the spinal cord. We can also look at glycogen levels by MALDI imaging. And so this is looking at a wild-type mouse. You can see a glycogen in the wild-type mouse here. We can additionally look at all of the N-linked glycans. So this is taking all 3,000 N-linked glycans, reducing them to one signal, and asking where does N-linked glycosylation happen in the cell, in the brain. Unsurprisingly, it's everywhere. The surprising piece was when we looked at Lafora's disease mice. So this is the Lafora knockout mouse. You see very similar recapitulation to the IHC here with our MALDI imaging. But what really stood out to us was this sort of lack of N-linked glycosylation. This idea that when you perturb glycogen metabolism, you also perturb N-linked glycosylation. We published this a few years ago now, but since then we've looked at about half a dozen different glycogen storage disease mouse models. And what we see in each case is that when you have aberrant glycogen metabolism, so this is Pompe disease, you have high levels of glycogen in Pompe's disease. And with that high level of glycogen, you now have low levels of N-linked glycan. Similarly, GLUT1 deficiency syndrome, in this disease, you have low levels of glycogen. You've perturbed the glycogen metabolism. You also perturb the N-linked glycosylation. So essentially what we're seeing is that when you perturb glycogen metabolism, you have aberrant N-linked glycosylation. We've also looked at the reverse. There's congenital disorders of glycosylation. In those mouse models and patient samples, you have perturbed glycogen metabolism. And why is this? As a biochemist, I teach medical school students biochemistry, right? We sort of focus on this piece right here, that glycogen is an energy driver for mitochondria. However, glycogen is also involved in being able to generate these sugar nucleotides, which are incredibly important. You heard in one of our earlier talks. These sugar nucleotides are utilized to perform N-linked glycosylation. This N-linked glycosylation is incredibly important in the brain because the proteins have to get to the cell surface, all those neurotransmitters and neuroreceptors. If they don't do that, they get stuck in this pathway. The result then is the epilepsy that you heard about from Kit Donahue in her talk. So the way we think about glycogen in the brain specifically is that, yes, it's about metabolism. It's also a signaling molecule and very important there. And that's because of this idea that glycogen perturbs N-linked glycosylation, which is important for resting potential, for action potential. All of these membrane proteins are highly glycosylated. The neurotransmitter loading, those proteins are glycosylated, and also for signal transduction that you can see. So in working on Lafora's disease, what Kit and others have alluded to is that it's an ultra-rare disease. To get companies interested in this disease specifically, what we've started to talk about the last couple of years is that there's a host of neurological glycogen storage diseases. So each of these diseases, at least in mouse models, And what's showing in patient data that they have aberrant glycogen metabolism in the brain. So the thought is, can we have one drug focused on either glycogen synthase to regulate it or to degrade the aberrant glycogen, and then have one drug to treat all of these diseases? You've got a large enough patient cohort that biotech is then interested into that. You heard a little bit from Kit about the LaFora Epilepsy Cure Initiative. This was work that Joan was intimately involved in. His lab was a big driver in this program project grant that we had from NINDS. Stunningly, this grant developed 3 drugs that are now have all gotten into patients, and I'll just kind of summarize the work the foundational biochemistry that led to this work. So we have an antibody enzyme fusion that's comprised of a Fab, a antibody fragment here, that's fused to that same salivary amylase that you heard about earlier. This can penetrate cells, and when it penetrates cells, it degrades the LaFora bodies in mouse models. So it doesn't cross the blood-brain barrier, but it can be injected directly into mice intrathecally or into their intercerebroventricular space. We've had a series of papers to show that this gets into the brain, it gets good biodistribution, it degrades the aggregates, and it actually normalizes brain metabolism in the LaFora disease mice. You heard about the antisense oligonucleotide that was developed in collaboration with Ionis Therapeutics. This was driven by Burge Menashian and his group at UT Southwestern and many others of us in the LaFora Epilepsy Cure Initiative that collaborated on that. And then there's also a small molecule that I think we'll hear about a little bit more later that was designed and developed by Maze Therapeutics, which targets GYS1. It downregulates GYS1 activity. Maze developed this for Pompe disease. Unfortunately, it doesn't cross the blood-brain barrier. However, importantly, as I mentioned, each of these drugs have now made it through large mammal testing, either canines or non-human primates, and are into the clinic. These two have gone through safety studies. This one's just now going into a phase 2. So the punchline is we don't have a perfect drug, but those basic mechanistic insights that Joan's work provided have allowed us and others to develop 3 drugs that have gotten into patients. So the take home from this first piece here is just the neurological glycogen storage diseases, a cohort of diseases, this tight link between glycogen metabolism and in-link glycosylation. And then our field collaboratively have developed multiple drugs. And then the next phase of that is getting these drugs across the blood-brain barrier. And there are unique ways to be able to do that. So one of the other things that we've looked at in our lab is cancers. You've heard a couple people mention that there are some cancers that have high glycogen levels. We screened about 60 different cancers and found high glycogen in 8 or 9 of those. The one I'll tell you about today is here, lung adenocarcinoma. Very high levels of glycogen in both lung adenocarcinoma, and this is showing Ewing sarcoma. Just as a relative, to get an idea of how much glycogen we're talking about, in Ewing sarcoma, these tissue samples have about the same amount of glycogen as a liver tissue has, so a very high level of glycogen. What we did in this story was to look at a tissue microarray. These are patient samples, biopsies from, we looked at about 150 different patient samples and scanned using the MALDI imaging to see how much glycogen is there. And what we saw was that while lung squamous cell carcinoma had elevated glycogen levels, lung adenocarcinoma had incredibly high levels of glycogen in those patient samples. And the high levels was in the tumor. It wasn't in the stroma, the vascular tissue, or the normal tissue. When we dug into this a little bit deeper, we wanted to know, and this was our reviewer 3, David, I don't know who our reviewer 3 was, but they asked, what's the genetic driver for these patients that have high glycogen? Is it just one type of lung adenocarcinoma patient that has high glycogen? Surprisingly, these are all human samples. When we looked at the different genetic drivers, every single genetic driver had high levels of glycogen for lung adenocarcinoma. Now again, this wasn't the case for squamous cell, but for lung adenocarcinoma, all of these different drivers had high glycogen. We could recapitulate this in the mouse models. So we got the mouse models for these common lung adenocarcinoma drivers, looked at the tissue, the lung tumors in those, and they too all had high levels of glycogen. So the big question is, great, so glycogen is there. Is it really important? Is it doing anything in these tissues? So to answer that question, we utilized this system here where we can induce using a KRAS P53 floxed mouse. So this is a normal lung adenocarcinoma mouse model, and then also cross that with the GYS1 floxed mouse so that when we add adenocre, we at the same time induce the tumors with P53 and also knock out GYS1 in these. And when we did this experiment, on the left you see the KRAS P53 mice, you can see the high levels of glycogen here, you can see the IHC staining in purple. The KRAS P53 mice that have glycogen synthase 1 knocked out, essentially no tumors in those mice. So we struggled to see any tumorigenesis in those mice, so it's showing the importance of glycogen metabolism in this model. Another intriguing piece of this story, which we don't fully understand yet, goes back to the phosphorylation aspect. So you've heard a lot about laforin and its role in Lafora's disease. When we looked at the lung adenocarcinoma glycogen, what we saw was, using MALDI imaging, we can also measure how much phosphate is on the different chain links. So this is looking at essentially 5 glucoses, 6, 7, 8, all the way up to 15. And in each of these cases for lung adenocarcinoma, there was a lot more phosphorylated glycogen than for lung squamous cell carcinoma. We could look at, quantify this with the imaging here. You can see the high levels of phosphate and the higher levels of that glycogen. Again, remembering that laforin is the enzyme that removes glycogen phosphate. And so when we looked by immunohistochemistry in the patient samples here, what we saw was that laforin levels are repressed in lung adenocarcinoma, but not in lung squamous cell carcinoma, matching the data that we're seeing up here as we would predict. Also, when you look at the Cancer Genome Atlas, this is looking at individual patient data for do they have high levels of laforin expression or low levels. The patients with lung adenocarcinoma that are expressing high levels of laforin have a much better prognosis than those expressing low levels of laforin. So there's some aspect of glycogen architecture, both chain length and or phosphorylation, that is a prognostic factor for lung adenocarcinoma. We can recapitulate this in mice. So if we take the lung adenocarcinoma cell line and we overexpress laforin, so we reintroduce laforin, you see the tumorigenesis is not nearly as aggressive. We can look at this in isolated stem cells from the Lafora disease mice. We see that the laforin knockout have much higher number of colonies. Those colonies grow larger. And then we can also do the same KRAS P53 experiment, this time crossing them with the Lafora's knockout mice. And in this case, we would predict that without laforin, the tumors would be much more aggressive. And that's exactly what we see. Tumor volume and tumor numbers explode. So it's not just about the glycogen, it's also about the glycogen architecture that's important in this disease. So I'll just leave you with a couple final thoughts here. In addition to the glycogen storage diseases, there are additional high glycogen diseases. I've shown you a lung adenocarcinoma story here that came out, most of that published at the end of last year. Earlier, we had published a story where the E3 ubiquitin ligase malin ubiquitinates glycogen phosphorylase, and phosphorylase then gets trapped in the nucleus. This is a story that Joan published on back in the early 90s, I think it was, showing glycogen metabolism in the nucleus. We showed how this changes histone acetylation and epigenetic regulation in this model. Ewing sarcoma is another one that we've published on recently that has high glycogen, and it's an important piece of that disease. And then lastly, lung fibrosis, a story there. And I think where we want to go and see the field go is now that we have these diseases that have high levels of glycogen, which one are targets actual drug targets versus just hallmarks of disease. The hallmarks are not so interesting, the targets are. And the targets are extremely interesting now because the Lafora disease field has developed these therapeutics that you can imagine now repurposing in these other diseases moving into the future. So with that, I'd just like to acknowledge two close collaborators at UF, Ramon Sun and Craig Vanderkoy, who we have a joint lab with, our team that does the work and our funding. And thank you all again for coming out to Barcelona and acknowledging Joan and his contribution to the glycogen field.
Thank you, David. Thank you. Yeah.
Matt, that was really, really terrific. I'm wondering about the lung cancer studies you've done when you knock down GYS1.
Yeah.
Are you looking at the reversal of a Warburg effect? Have you done any seahorse studies or anything like that?
We do, not a big change. Yeah, it's surprising to me. So for lung cancer, I didn't show the data. We also see a massive change in the in-link glycosylation in those tissues as well. So is it metabolism or is it glycosylation? We don't yet know, but yeah, kind of where to head next.
Thank you.
Thanks for your excellent talk. Just I want to ask regarding the cancer, you mentioned glycogen levels alterations and laforin level alteration and the transcriptomic level. I would like to know if there is some correlation identified with a high level or either synthase or low levels or phosphorylase of glycogen, glycogen synthase, glycogen phosphorylase, or laforin. So there is correlation at transcriptomic level that give clues who is if there is a transcriptomic level determinant of this accumulation of glycogen or not?
Yeah, so the question I think is, what about the protein levels and the transcript levels of the machinery for glycogen metabolism? We don't see a lot of changes in the protein levels for other glycogen metabolism enzymes. So laforin is definitely downregulated. For lung cancer? Not for lung cancer, yeah. The E3 ubiquitin ligase is transcriptionally silenced in lung adenocarcinoma, and that leads to glycogen phosphorylase getting stuck in the nucleus. But the levels of phosphorylase are the same in the normal tissue and the lung cancer tissue. It's not about the level, it's about the location of where the degradation enzyme is at. So essentially, you're trapping phosphorylase in the nucleus, and it should be out in the cytoplasm. So that's why you get the excess glycogen in the tissue, is because the glycogen's in the cytoplasm, the degradative enzyme is now in the nucleus. Sure.
So that's it.
Very good.
Thank you.
Thank you,
Matthew. Okay, so it is my pleasure to announce our next speaker, Marc Claret. So Marc, please.
Okay, so good afternoon. Thank you very much. And of course, first of all, I would like to thank the organizers for inviting me to be part of this tribute to Joan. I really appreciate this. I had the privilege to closely interact with Joan at two different stages of my career. So first, as a biochemistry student at the University of Barcelona, early in my 20s, and later on as a researcher. And over these years, so I realized that among the many qualities of Joan, so one of the most important, at least for me, was his ability to inspire people. So I'm sure that many of us in this room were inspired by Joan, by his research and his way to approach science and also life in general. So thank you, Joan, for your legacy. So today I would like to tell you about unpublished results that deal with the role of glycogen in the sensory perception of pumps in neurons. This is a project that we started several years ago in collaboration with Jordi Duran and Joan Guinovart. So without them, we couldn't have gone so far. And for that, I also thank Jordi and Joan for their generosity to share all the regions and the expertise. So for more than 20 years, my lab has been interested in understanding how energy balance is maintained at the organismal level. And of course, this is an extremely complex and multi-layered biological process that is influenced by many factors, so ranging from genetics to even cultural aspects in humans. So the brain is the major orchestrator of energy balance, and it does this by integrating a lot of signals. So interoceptive signals, such as nutrients, hormones, and, sorry, I cannot use the pointer. Yeah. So such as nutrients, hormones, and also the nervous system, and also exteroceptive signals coming from the sensory, environmental, and contextual domains. So the archonucleus of the hypothalamus, it's been recognized for decades as a key area of the brain implicated in the regulation of energy balance. And within this brain region, so a population of neurons called POMC stand out as a key actors implicated in the regulation of appetite, energy expenditure, and systemic metabolism. So under one of the major features of POMC neurons is that they are able to act as metabolic transceivers, in the sense that they are able to detect fluctuations in a wide range of circulating factors informing about the energy status of the organism. For example, nutrients and hormones such as glucose, certain free fatty acids or amino acids, and hormones such as insulin, ghrelin, leptin, or GLP-1. And basically, under conditions of positive energy balance, so POMC neurons get activated. They increase the expression of the prohormone POMC that is sequentially cleaved into several bioactive neuropeptides, including alpha-MSH, which is arguably one of the most important anorexigenic neuropeptides. So POMC neurons release the alpha-MSH in the axonal terminals that are distributed throughout the brain, but particularly dense at the paraventricular nucleus of the hypothalamus, where they mediate the canonical response, and they bind as an agonist to the melanocortin receptor. In this way, activating a signaling cascade that leads to reduced food intake, enhanced energy expenditure, and changes in glucose and lipid metabolism. Right, so therefore, POMC neurons are not able to monitor these fluctuations in nutrients and hormones, but also to integrate this information and elaborate precise responses in order to adjust to the metabolic demands of the organism. Right, so this is the classical framework that has guided the research in this field for more than 30 years. But about 10 years ago, 3 different teams discovered that actually the activity of POMC neurons can be regulated by the mere sight or smell of food. So therefore, they're able to also respond to the sensory perception of food in anticipation to actual food intake. So the discovery that led, the technique that led to this discovery was fiber photometry, which allows for the continuous recording of neuronal activity in a cell-specific manner in freely behaving mice by using biosensors, so calcium biosensors that are used as proxy of neuronal activation. So basically, in a seminal paper by the laboratory of Zach Knight, they showed that when the mice are exposed to a non-edible object, so nothing happens in terms of POMC neuron activity, but as soon as they are exposed to a chow pellet, so there is a rapid activation of these neurons. And this happens before actual food intake, as it can be seen in the second panel, in which the activation of POMC neurons precedes the first bite. Importantly, the magnitude of activation of the sensory perception of food or actual food intake is very similar, as it can be seen in this quantification graph here. So therefore, these findings challenge the classic view or the homeostatic view, and therefore this new sensory perception aspect of this neuron should be incorporated, accommodated in the framework. And therefore, some authors came up with this hybrid model in which homeostasis means allostasis. And it suggested that it works at 3 different speeds. So the first speed, which is a very rapid, that happens within seconds, that is mediated by the sensory perception of food. A second speed that happens within minutes, that is mediated by the chemo and mechanosensation of the gastrointestinal tract. And a third speed that happens within minutes to hours, that is the classic homeostatic hormonal regulation. Right, so therefore, POMC neurons are able to monitor energy status in anticipation. They're able to integrate and coordinate adequate responses in order to adjust for the future demands of the organism. Right, so despite the importance of this biological process, the sensory perception of food, so little progress has been done during these 10 years. And there are many questions that still remain to answer. So today, I would like to try to address a few questions, 3 of them in particular, that are shown in this slide. And the first one is, what are the molecular mechanisms underlying the sensory modulation of POMC neurons? Right, so to answer this question, so we did a very simple experiment in which we took advantage of the POMC ribotag mouse, which allows for a cremated tagging of polyribosomes and subsequent isolation of the RNA in a cell-specific manner. So in that case, in POMC neurons. So we overnight fasted those animals, and then we exposed them to either a non-edible object, so which is a piece of wood that is similar in size and color as a chow pellet, that is encapsulated within a wire mesh, or they were exposed to actual food pellet that, again, is encapsulated in a wire mesh, in a way that it can be seen, it can be smelled, but it cannot be eaten. So after these 10 minutes, we collected the archonucleus of the hypothalamus, and we proceed to isolate the mRNA to subsequent RNA-seq. So basically what we observe is that both conditions, the neo and the sensory conditions, so nicely separated in the PCA analysis, so suggesting distinct transcriptomic signatures, and we observed 99 differentially expressed genes. So 98 of them were upregulated, while only 1 downregulated. When we look at the gene ontology analysis, we found an enrichment of pathways involved in the macromolecule metabolic processes and also phosphatase regulatory activity. So when we look at the identity of some of these differentially expressed genes, so we noticed the upregulation of 3 phosphatases that are implicated in glycogen metabolism, including PTG, protein targeting glycogen, that we have talked a lot about today. So next we wondered, is glycogen therefore implicated in the sensory activation of POMC neurons? So to initially address this question, so we took advantage of the hypomap, which is a single cell atlas of the hypothalamus, and we looked for the expression of several genes implicated in glycogen metabolism. In particular, we look for glycogen synthase, glycogen phosphorylase, and glycogen branching enzyme. We look in particular in POMC neurons and compare it with astrocytes as a bona fide cells that express glycogen. And as you can see here, so we observe that POMC neurons, they do express actually the glycogen metabolism machinery, and actually to a similar extent than astrocytes. So indicating indeed that POMC neurons do express genes implicated in the glycogen metabolism. But what about the protein and glycogen per se? So in order to retain cellular resolution, so we used immunofluorescence approaches. We used antibodies, specific antibodies against glycogen synthase and also against glycogen, kindly provided by Matthew. These antibodies have been validated by others, but we also validated ourselves by using the nesting glycogen synthase knockout, in which deletes glycogen synthase in the whole brain. And basically what we saw is that indeed in the paraventricular nucleus of the hypothalamus, so there is a colocalization of alpha-MSH, which is a marker of POMC, with glycogen synthase, as you can see here. No signal in the negative control. And similarly, we also observed presence of glycogen staining in the POMC neuronal axons in the paraventricular nucleus, as it can be seen here. And again, so no signal in the knockouts. So these results indicated indeed that glycogen synthase and actual glycogen granules are expressed in POMC neuron projections. So next, we wondered whether sensory food cues are able to modulate glycogen and also glycogen synthase expression. And for this experiment, we used control black 6 mice that were overnight fasted, and again, they were exposed for 10 minutes to either the neo or the sensory paradigm. And we measured the expression of MGS and glycogen bioimmunofluorescence. What we saw is that under conditions of sensory, there is a dramatic increase in the presence of glycogen synthase colocalizing with POMC neurons, and this is concomitant with a reduction in the quantity of granules in the axonal projections of POMC neurons, as it can be seen here. So these results are consistent with an accelerated glycogen metabolism, in which this increase in glycogen synthase is, we believe it's a compensatory mechanism in order to refill the glycogen stores to be ready for the next round of sensory perception. So next we wondered what is the relevance of neuronal glycogen in the context of POMC neuron sensory activation. And for that, we generated mice lacking glycogen synthase, specifically in POMC neurons, using the Krelog system. We used a mouse that kindly was provided by Jordi and Joan. And we prepared them for fiber photometry by injecting AABs that express the biosensor, the calcium biosensor, and also an optical cannula. So after an overnight fast, the mice were exposed as usual to 10 minutes to the neo or the sensory perception of food paradigm. And this is just a figure to show that indeed, so the cannula was placed at the right place, just on top of the arcuate nucleus of the hypothalamus, where POMC neurons are located. So this is what we saw under in control animals. So basically, we saw that under neo conditions, so nothing happens in terms of activity, but as soon as a chow pellet is exposed, so that is encapsulated, and it cannot be reached, but it can be sensory perceived, so there is a rapid activation of POMC neurons. This is the quantification of this experiment, and here you can see the individual traces of each mouse. So now, so what happens in the mutant animals? So this is what happens in mutant animals. So in mutant animals, we did not see almost any signal, as you can see in the graph. So the sensory perception of food, so just provided a residual increase in the activity of these neurons, as it can be seen in the quantification graph. Right, so these results suggest that glycogen synthase is therefore required for the sensory activation of POMC neurons. But at this point, we consider the possibility that these neurons were somehow functionally dead, right? And that they were blind, basically, to any kind of a stimuli. So it is known that POMC neurons respond bidirectionally to several cues. In particular, for example, they increase their activity when they sense glucose, and they reduce their activity when they sense ghrelin. Right? So we wanted to assess whether these POMC neurons lacking glycogen so were able to respond to other signals. So basically, we did an experiment in which we provided a bolus of glucose, oral glucose to the mice. These mice are control animals. This is the expected response, so a rapid increase in the POMC neuron activity after a bolus of glucose, and a rapid decrease in the POMC neuron activity after injecting ghrelin. And basically, we saw exactly the same in the mice lacking glycogen synthase in these neurons, so suggesting that POMC neurons, or the lack of glycogen in POMC neurons, is not a negative thing, and in fact, they are able to detect the other signals, and the fact that they don't perceive the sensory cues from food is something that is very specific for this particular scenario. Right, so far I have shown you that glycogen synthase is required for the sensory activation of POMC neurons. But I listed other questions that I would like to address, and the second one is, what is the physiological purpose of sensory inputs? And we wanted to address this at three different levels. So in regards to feeding behavior, in regards to the hepatic preparation for the postprandial input of nutrients, and also in the cephalic insulin release phase. So as you know, so feeding behavior is an extremely complex sequence of events that is divided into three main phases: the appetitive phase, the anticipatory phase, and the consummatory phase. And we designed experiments in order to assess each of the phases. So we generated, again, controls and knockout animals like in glycogen synthase in POMC neurons, and we used the whole board test that has been widely used to assess exploratory behaviors in mice. They were habituated to the whole board, and afterwards we baited this whole board with food that again was visible. They could be seen, they can smell it, but they cannot reach it, they cannot eat it, right? But the number of head dips that we observed between control and knockout mice were equivalent, so suggesting that the POMC neuron activation does not regulate the appetitive phase. So what about the anticipatory phase and the consumatory phase? So we used the three-phase open field test to assess these two phases at the same time. And again, so control and knockout animals, so were overnight fasted, they were habituated to the arena, and afterwards they were exposed to the neo condition for 15 minutes, right? So and we measured the latency of these mice to enter into the square where the non-edible object was located. And afterwards, we extended the experiment by providing actual food that was fully available, so the mice could eat it. And this is what happened. So in control animals, we saw a dramatic increase between the neo and food conditions. So there is a transition, so an increase in the time that they spend in the center where the food is located. But we did not see this response in the mutant animals. And also what we saw is that the mutant animals eat much less, significantly less than the control counterparts. So together, these results indicate that the lack of glycogen in POMC neurons is not altering the food discovery capacity of those animals, but instead it is impairing the consumatory drive. So they lose sort of interest for the food. Now, so what about the hepatic preparation? So a few years ago, the team of Jens Bruning published a paper in which they demonstrated that the sensory activation of POMC neurons is able to prime the endoplasmic reticulum via mTOR activation. So they showed that after 5 minutes of exposure to a cage food, so there is a phosphorylation of mTOR and phospho-AKT in the liver of those animals. So we wanted to replicate these studies in our model and see whether the glycogen in POMC neurons was important for this effect. So we repeated exactly the same experiment, 5 minutes exposure to neo, sensory, or full availability of food, and we conducted Western blot. So as expected, the control animals showed an increase in the phosphorylation of AKT and mTOR under both conditions, under food and the sensory condition, but the mutant animals showed a blunted response specifically only for the sensory condition. So indicating that the deletion of glycogen synthase in POMC neurons blunts the hepatic engagement of the AKT and mTOR pathways. Now, what about insulin release? It's known for decades that there is a cephalic insulin release that happens before actual food intake. We know nowadays that this effect is mediated by the hypothalamus through the vagus nerve and that instructs the pancreas to release insulin in anticipation to prepare the body for the postprandial income of nutrients. So we did a very simple experiment to see whether our mice had any defect on this particular biological effect. So we used control and knockout animals, and after an overnight fast, so we took a blood sample at minute 0. Immediately after, they were exposed to the neo or the sensory perception of food paradigm, and 2 minutes later, we collected a second drop of blood to measure insulin levels. So this is what happens when control and mutant mice are exposed to the neo condition. So basically nothing happens. So there is no transition from the minute 0 to minute 2. The levels of insulin remain more or less normal. But as soon as they are exposed to the sensory perception, so the control animals, you can see an increase in the circulating insulin that we find in the blood, but this effect was not seen in the mutant animals, indicating again that the impaired sensory activation of POMC neurons alters the cephalic insulin release phase. Right, so far I show you that the deletion of glycogen synthase blunts the sensory activation of these neurons and causes several behavioral alterations related to impaired food activation, sensory activation, delayed food entrainment, impaired lipid metabolism, and reduced cephalic insulin release. We were able to confirm all these findings. So we recapitulated everything when we used 2 different strategies to complement this data. In particular, we used Cre-dependent siRNA against glycogen synthase in a Cre-specific manner. And also we recapitulated the data when we targeted PTG, so suggesting that these results are very robust. So now, the next question was a difficult one. So what are the effects of glycogen synthase deletion driven by loss of glycogen synthesis or any another function yet to be described of this enzyme? So to answer this question, we designed an experiment in which we generated several AAVs. We used the knockout background, the POMC GIS1 knockout background, in order to overexpress or express either the wild-type form of glycogen synthase or a mutated form, catalytically edited form, in which we replaced 2 residues of the catalytic domain to make them unable to generate any glycogen, right? So this mutation renders the enzyme unable to make glycogen. So we infected these mice with these AAVs. This is just to prove that indeed, when we express the wild-type form, we are able to recover the levels of glycogen in the POMC neuronal axons. And when we express the catalytically dead, so again, we see a reduction in glycogen, so suggesting that the system is working. And we conducted, again, fiber photometry on those animals. After an overnight fasting, they were submitted to the sensory perception paradigm. So basically what we saw is that as expected, and as I have shown you several times already, mutant animals showed a defect in the sensory perception of food in terms of POMC neuron activity. The POMC neuron activity was recovered when we expressed the wild-type form of the glycogen synthase, and again, this effect was lost when we expressed the catalytically dead form of glycogen synthase. Right, so the next question is, what are the physiological consequences of perturbing the sensory signaling in those neurons? So we conducted a comprehensive phenotyping of those animals, and basically what we saw is that under standard diet conditions, mutant animals show normal body weight, no changes in the glucose levels under fed or fasting conditions. However, we observe a slight but significant and very consistent insulin resistance, as you can see in this graph, in the face of no change in their glucose tolerance, because they were able to compensate by enhanced insulin release by the pancreas. Right? So these results together were resembling a pre-diabetic state. And we reasoned that perhaps by challenging the system, we would see a much stronger phenotype on those animals. So therefore, we fed those animals with high-fat diet. And as it can be seen here, so as soon as we provided the high-fat diet, so mutant animals start to gain more weight than control counterparts, as it can be seen in this graph here that summarizes the final body weight. So this increased body weight was due to increased fat content, no changes in the energy expenditure of these mice, and basically this overweight phenotype was caused by hyperphagia. So these animals also showed alterations in glucose metabolism, in particular increased or hyperglycemia under fasting conditions, hyperinsulinemia. The mice remain insulin resistant, and they also develop glucose intolerant, and still we were seeing this enhanced insulin secretion. So when we put those animals on high-fat diet, they developed over diabetes. So what happened during aging? So very similar results, actually. So aging is also a challenge for those animals. So actually what you see is that at old ages, so these mice show increased body weight, increased glucose under fasted conditions, insulin insensitivity, glucose intolerance, and again, so this recapitulates very well the phenotype that we see under high-fat diet. So suggesting that when these mice are challenged, so metabolic alterations appear. And this is my last slide, which is a sort of graphical abstract. So I have shown you that the sensory, under sensory perception conditions, so POMC neurons required a rapid but transient energy source that is provided by the degradation of glycogen, right? So this glycogen in POMC neurons is important for the feeding entrainment, for the anticipatory insulin release, and also for the liver preparation for the postprandial nutrient outpouring. And that when these animals are challenged either by a high-fat diet or by just aging, so then they develop overdiabetes and obesity. And with this, I would like to thank everyone that has been involved in this project, in particular the people from the lab, and more especially Alicia Garcia Gomez Valadez, who's a very talented scientist that has done an amazing job in this project. We have done this project side by side with Mark Schneeberger at Yale University. And I would also like to thank, of course, all of our collaborators, Matthew Gentry for the antibodies, Nicolas Renier, Giuseppe D'Agostino, Ruben Nogueras, Tomasz Horvath, and especially, of course, Jordi Duran and Joan Guinovart. So thank you very much for your attention, and I'll be happy to take questions.
Thank you.
I will try not to dominate all the time, so I just have 12 questions.
Well,
no, anyway.
Do we have time?
Only two.
The first question is, what's driving the accumulation of GYS1 and PTG and PYG and all these other proteins in the sensory neurons?
So in our transcriptomic analysis, we believe that this is a compensatory mechanism. So this is not what it drives our phenotype. So these changes in the expression of those genes related with glycogen metabolism, we believe is a reflection of a compensation to maintain the glycogen machinery, ongoing, and to make sure that when it's needed, the glycogen granules are there to be used for the sensory perception.
Are the genes upregulated in obese mice?
That's a good question. I couldn't say. I don't know. We haven't checked it in our hands, and I can't recall of any study in which they have checked it specifically in these neurons, whether they are highly expressed or not.
Is glucose transport in the sensory neuron required for the effect?
Yes, there is glucose transport.
So you require the glucose transport also. So have you tried to block glucose transport?
No, we didn't.
Fluorescein or anything like that to see?
No, we didn't do this experiment. No.
So you don't know if it's necessary?
No, we don't know. We don't know if it's necessary.
And just one more question. Have you tried to specifically block the turnover of glycogen in the brain as opposed to just the accumulation of glycogen? So I'm just wondering if there's a signal coming from the turnover. So like, you could use a phosphorylase inhibitor or knockout.
We were considering that, but we're discussing this, a long time with Jordi and Joan, and we thought that perhaps by inhibiting glycogen phosphorylase, what we would lead is to an accumulation of glycogen that perhaps would kill the neuron. And
or maybe you block the signal that you need.
Yeah, but this would require perhaps, a pharmacological intervention. So but then would not be specific, So the thing is that the hypothalamus is very, very heterogeneous. There are very different types of neurons. These neurons respond, for example, to the sensory perception of food and glycogen. We know it's important, but we have done the same experiments in the neighbor neurons called AGRP, and they don't need it. So these mice are normal, and their sensory perception is fine. So what I mean is that this is something very specific that perhaps using pharmacological approaches that are not targeted would have missed or interpreted.
Yeah. Just one more little question. I wonder if you ever saw GYS2 coming up. And the reason I'm asking that is because GYS2 normally it's one that's expressed in, it's a muscle form, but it's also expressed in other tissues. But in fat cells, when we activate fat cells with adrenergic agonists, GYS2 comes up for some reasons I don't really understand.
All right.
And GYS2 is less sensitive to glucose 6-phosphate. So I'm wondering if there's like a shift between 1 and 2.
We can have a look at our data set, but we didn't look specifically for GS2 because it was
And
not supposed to be expressed.
PYG
right?
too, it's also different.
Right, but in theory, it shouldn't be expressed, right, in the brain?
It shouldn't be.
But perhaps under sensory perception, there is a, an upregulation or, yeah.
Yeah, interesting.
Okay.
Thank you.
Thank you. I'll have to start to defend Mark. Is there any other question? Okay, if no, thank you very much. Thank you, Mark.
Thank you.
So now I'm a little bit nervous because I'm not sure if I will pronounce correctly the last name. But with us is Elton Zakai-Raj, and he will try to give us a little bit of insight in structural dimension of glycogen synthesis.
Is this, oh. All right. Thank you to the organizers and for organizing this fantastic meeting. And it's been a great reminder of Joan and the ways that he influenced all of us and our work. I'd like to give you sort of an update or one of the most up-to-date views on how we make glycogen. And as Philip mentioned this morning, this is still an area in which is surprising us and is always giving us new things to wonder. So we've seen throughout the day this sort of schematic of glycogen and with glycogenin in the middle and the branched glycogen in tiers. And this is a very simplistic sort of view of the glycogen structure, but typically there's about, theoretically at least 12 tiers and probably 50,000 glucose residues. So this is a massive macromolecule. But what we can be sure is that glycogenin operates as a dimer, and that's clearly established by many groups, including Anna and Peter Roach's early work, and from Wyatt Hughes' lab, who's now at Newcastle, we know that this transglucosylates and autoglycosylates, something that Philip and Bill Whelan discovered many years ago. So we came into this when we tried to understand how does the conserved C-terminus of glycogenin docks into the glycogen synthase. And we found that through a crystal structure of the C. elegans enzymes, which was the only one we could crystallize at the time, that these interact in a very particular docking site on the synthase. And this was our first sort of interaction through Kay and Joan, who really helped us to then translate these findings into the human enzyme. So with the help of Joan's and his collaboration, we could figure out that these binding interacting sites between GN and GS were also relevant in humans. And so we've come up with this sort of relatively simplistic model, which by and large is correct, although I will sort of towards the end of this talk, you will see that we've revised this somewhat, and we understand a little bit more. So we have a tetrameric GS and glycogenin, but the way glycogenin comes together is a little bit more complicated than this. I think it goes without saying that the first sort of understanding of how GS is regulated, or the glycogen synthase is regulated, comes from yeast structures that came from Bhaskaran and Anne and Peter Roach and Tom Hurley's early studies, where, sorry, this is supposed to be an animation, but I'm afraid it isn't working. But imagine a tetramer with 4 glycogen synthases, and when it's phosphorylated, this is in a relatively sort of tense, closed up conformation, and as the G6P binds, this extends, the 4 subunits sort of extends outwards, and while the tetramerization domain acts as a spring that basically is regulated by G6P, and we have higher activity with G6P, almost inactive when it's fully phosphorylated and sort of intermediate state. Marcello mentioned the Roseman fold, so there's 2 Roseman folds domains in GS and this tetramerization domain. But one of the key things that we've only started or beginning to understand the structural basis, at least, while we knew for a very long time that these phosphorylation sites were important at either end, both at the C and the N terminus. We've only recently have started to understand exactly the molecular basis for how this tense form is stabilized and is inactive. And I'll touch on that a little bit throughout my talk. So the key questions that I'll try to answer today is that, how does phosphorylation of each tail lead to inhibition, and how does glycogen synthesis initiate? So how do we start? And more importantly, how can we inhibit GS therapeutically? So we heard throughout the day, and also from Matthew earlier about the MZ101, this GS1 specific small molecule inhibitor. And I'll actually start with number 3, because using this compound has actually really helped us answer the first 2 questions. So, MZ101 was developed by Maze Therapeutics and is currently in clinical trials for Pompe disease, is selective for muscle versus liver. And this was one of the things that we were really intrigued by. How is this selective versus GS1 versus GS2? Because the 2 are really similar. And with the help of Kay, who you've heard a little bit earlier, we started working on this. And the first thing we did is that we incubated MZ101 with our purified glycogen synthase complexes, and we found that this has a really strong effect in stabilizing the complex. So this is about, if you try and heat up and unfold the protein, the stabilizing effect was about 7.5 degrees, and that's really much better than G6P, which is a known ligand. There's no additive effects between the 2, and no additive effect between UDP glucose and MZ101, whereas UDP glucose and G6P have a huge effect. They synergize, whereas when you add MZ101, you actually start bringing these back down. So that kind of hinted that MZ101 was somewhat different, so it didn't quite interact in the same in the known sort of pockets that we usually think about how these ligands bind. So we went on and we collected 4 cryo-EM data sets, all of them with MZ101, and then differing whether we put G6P or UDP glucose, and one with all 4. And to our surprise, but also satisfaction, we ended up with some really high resolution structures of both the glycogen synthase tetramer, but rather unexpectedly also the GN dimer. So the glycogenin, we could always see it, but it was always fairly low resolution. Whereas this one we could really see it pretty well. We could see all of our ligands at really good clarity, and we could see these 2 forms of glycogenin dimer, one horizontal in all of our data sets and one sort of vertical, and I'll come back to these towards the end of my talk. So to start with, and I really apologize for the quality of this, but you will see these pockets in more detail throughout the talk. But basically, we have again 4 subunits and all the ligands bound, including phosphorylation, and MZ101 binds in a completely new pocket, which seems to be a new and druggable regulatory pocket right at the hinge between the tetramerization helix and where the Roseman fold 2 domain. And we can see a really beautiful cryo-EM density for all the ligands, UDP glucose, MZ101, phosphorylation of the C terminus, which we had seen before as well, and UDP glucose and G6P as well. And I won't go through all, in the interest of time, go through the entire sort of structural basis, but just to highlight that MZ101 binds to this hinge pocket, and which sits precisely between the tetramerization helix and Rosenthal 2 domain, and that sort of makes tightens up the structure more towards the phosphorylated form and basically keeps it in an inhibited form. And to our satisfaction, we could also see that if you look at all the residues that interact, they're all conserved between GOS1 and GOS2, with the exception of one residue, and this is valine 491, which is a methionine in GS2, and we think that that's the that is the basis for the selectivity, and we're currently making mutants and testing this experimentally, but I would be surprised if that's not the case. And so just to summarize this part, so MZ101 really locks the GS hinge into this novel inhibitory form, and it's all to do with how this tetramerization helix and the Rosenthal domains come together, and how the tetramer, the structure of the tetramer, which is a lot more constrained when it has MZ101, and that looks more like the phosphorylated form versus the G6P form, which is much more extended and active. So the angle, the hinge angle in this case, represents a regulatory mechanism, which we previously didn't quite appreciate it. We thought that was from the yeast structures. We thought that was important, but this just adds to that notion. So I'll come back to how does phosphorylation of each tail lead to inhibition? And this was a real surprise, but a couple of years ago, together with we published, as well as YTU's group, showed that how serine 641, you've got all these phosphorylation sites, and one key thing was, why does GSK3 just keep going? And one of the reasons was that you really need that 641 residue so that could be coordinated by this arginine cradle, and that keeps the structure in its phosphorylated form. And so we've answered that, but we were also always wondering what happens. Why do we need also phosphorylation sites at the N-terminus? And many of you in the room have either discovered or blotted for these at some point. And I'm pleased to say that we now sort of understand the reason why this N-terminus leads to inhibition, and it has to do with a new lysine-arginine-rich pocket that is nearby, but not the same as the pocket in the that has to do with 641, and this is coordinated by both the same subunit. So this comes in, gets pretty close, but doesn't quite compete or with the arginine cradle, but then turns almost 180 degrees and binds to its own pocket, and that has residues that are coming from this subunit, but also from a nearby subunit that leads to the tetramer, and then you've got the same sort of thing happening at this end. And so this just gives you a little bit of an appreciation of this regulatory site. So you've got, it's all to do with this helix alpha 22, which Kay mentioned earlier, and Hannah and Peter Roach would know that by mutagenesis they showed that this was important. So this helix and this cluster of arginine residues is really coordinating both the G6P and the front and the back of this helix is basically keeping the whole thing. It's a proper sort of rheostat of activation. And so we now basically understand how both the N-terminal phosphorylation and the C-terminal phosphorylation inhibit the enzyme. So the next thing that we wanted to understand is how does glycogen synthesis initiate? And this is, again, we had some ideas, but we're yet to fully understand, and it's sort of still an unfinished business. And our best hypothesis right now is that in our new structures we can see two different forms of how glycogenin interacts with the tetramer. Glycogen synthase tetramer, and what we've termed the horizontal form. So this is a horizontal glycogenin dimer, and a vertical form, which was also seen by a structure by the Mays team in 2022. And the difference is obviously how glycogenin sits, but also one of the subunits of GS is sort of uncoupled from the tetramer. You can see that this is now not completely symmetrical. One of them just essentially pops out when you have the vertical form of glycogen. And that leads to the question, why do you have two? So why do we need two different forms of glycogenin? And our best thinking right now is that, so we don't have any sugars on the active site yet, so because this was a mutant that we used. But if you look at the horizontal GN dimer and you look at distances and line of sight from where the initiating, the first glucose residue would be and where it will need to get to, it's about 32 angstroms in the horizontal GN dimer and about 44 angstroms in a vertical GN dimer. And this is also sterically obstructed. So it's not it's not an easy way. It's longer, but also not an easy way to get through. So our best bet right now is that the horizontal dimer is better suited for glucose chain handover. So glycogenin will have already autoglycosylated, and it will be easier for the synthase to basically pick it up from this subunit in this conformation. Now that leads to the question is, why do you need the vertical dimer? And our hypothesis is that as you switch from horizontal to vertical, you now create the space with that subunit sort of popping out. You now create a pretty good space for glycogen branching enzyme to come in, and this is it's not a real structure, but it's great shape complementarity. And as you switch from vertical to horizontal, you can get this sort of extend cycle for glycogen synthesis. So we think that basically the vertical dimer is better suited to accommodate branching, whereas the horizontal dimer is basically better suited for extending, so handing over the chain and extending. And so with that, I'll leave you with this model, which is, again, it's still a model, and we may have to revise it, and I'm pretty sure we will revise it pretty soon. But our best bet at the moment is that as we have glycogenin as a dimer docking in with GN34, and this is where the ubiquitination sites that Philip mentioned this morning are, and glycogenin already has some sugars which can then be extended in the horizontal form. And once they are extended to a point that then glycogenin switches conformation, comes from a horizontal form to a vertical form, which basically can accommodate branching. And then you can get the branching, and then you can start again, extend, branch, extend cycle to get to this to continue doing the cycle and keep growing the particle. So just to finish off, so I've shown you that both phosphorylated N and C termini converge on this region, which we term the spike region, and that is a constrained movement by these two arginine and arginine lysine pockets. MZ11 binds to a new hinge pocket, which locks the enzyme into an inactive conformation, and there are multiple GN conformations which dictate chain handover and may regulate branching. And finally, I'll acknowledge Emma, who did pretty much all of the EM work and been helped by various people in the lab, our cryo-EM facility in Leeds. And I didn't show any of Kay's data in the interest of time, but we've collaborated with Kay for very long on this, and he's has been a great supporter and collaborator on this project. And thank you for your attention.
Thank you, Alton.
Now, if I understood correctly, you said that MC1 binds also to liver glycogen synthase. When you show at the beginning.
No,
GYS2.
I think it would not bind to GYS2 because of the V2M mutation.
Okay,
Yeah,
so
sorry,
I thought you said that it was binding because
no, it would not.
We can even administration of the compounds in mice. It decreases glycogen everywhere else, but GYS1, but never in liver.
Yeah, and that's consistent with what we see. Yeah. That's not the case. Sorry.
Okay. Thank you. Any other question? Yeah. Sorry.
I'm not sure. I presume that the glycogen synthase inhibitor does not inhibit the UDP glucose-dependent glycosylase activity of glycogenin?
No.
Because it's binding to this allosteric pocket that
Yep.
doesn't do that. So, okay, but do you see in the presence of this inhibitor, can you see the glucoses in your?
We cannot see the glucose because we are using a mutant that does not have any glucose. We're using a Y to F mutant that, yeah.
Okay.
We don't yet.
was
We're
good.
making those grids right now, but because of the heterogeneity of, it's just much easier to get structures without the glucose.
All right.
And
It would be interesting to know how many glucoses were on it at that juncture.
We're hopefully next year.
Yeah. Matthew, please. Any other questions? Okay. If not, I would like to thank you, Alton. And yeah, so although Jean was present in all those lectures and mentioned several times, I believe that for the closing this symposium, we would all like to hear the presentation of Anne Paoli Roach, who will bring us back to the beginnings and share with us some also personal thoughts about Joan. Thank you, Anne.
No. Yes. Yes. So how do I advance? This one? Yes. Yeah. So I would like to thank the organizers for giving me the opportunity to reminisce my 50 years of knowing Joan. So I thought it was this one, but how do I advance? Is it this one? So I met Joan in 1974, and I have known him, as I said, for 50 years, from 74 to 2024. And this is, not sure how does this pointer work? This is Joan when I met him. Probably you guys don't remember him, but he has a big afro hair. And this is the Joan that we all know. As I said, I met Joan in '74 when my group, consisting of a friend, Rino Masiello, and my boss, Ettore Bergamini, myself went to Charlottesville in the summer to conduct a small study on the effect of epinephrine on cyclic AMP-dependent protein kinase and glycogen phosphorylation. So Joan and Rosa took us to Shenandoah National Park, and this is a picture of us there. You probably don't recognize me. So Joan used to say to me, "La vida es duras para madre soltera." And I didn't know at the time what it meant. I didn't know if he was trying to warn me, be protective of me, because it was around that time that I met Peter. But Rosa told me recently that this is a say in the time when it was considered disgraceful to be an unmarried mother. And Joan likes to rehearse this old say. So, while I was there in the fall, actually let me say, Joan joined the Launders Lab in '74 after he has gotten his PhD degree with Manuel Rosel Perez. And Rosel Perez had worked with Lerner on frog and liver glycogen synthase. So when Joan got his PhD, it was an obvious choice for him to join the Lerner's lab. In the fall of '74, Peter joined the group he was at UCLA, and he joined the Joe Lerner's lab. And at that time, you can see here the three most important people who carried Joe Lerner's legacy on glycogen metabolism. John Lawrence, which at the time was a graduate student, and he worked on fat cell glycogen synthase and insulin regulation of fat cell glycogen synthase. You can see Peter here, just coming fresh from California with long hair and purple shirt. And Joan here. So Joan was working also on insulin and epinephrine control of glycogen synthase in fat cell. The ones of you who knew Joe, you know that Joe was convinced that insulin generated a chemical mediator who would control phosphorylation and dephosphorylation. And he pursued this goal for all his life, chasing this elusive chemical mediator. Unfortunately, we lost all of them. Lawrence at a very young age, Peter, John, and Joan. The only one left of the group is me. It's very sad. I'm going to show you a series of pictures. This is the Lerner's group in Joan and Rose's apartment in Charlottesville, Virginia in '75. And here is another one. Here we also have Rosa and Joan, again, Joan with afro hairs. And this is a pictures of Peter's birthday in Charlottesville, Virginia, and Rosa, you made a cake for him for his birthday. And here are the two cousins. Peter, Joan, and myself, we used to call ourselves cousins. And since '74, we established a long life friendship that lasted over the years. I was able to dig out some emails that Joan and Peter had exchanged, and you can see that they address each other's dear cousin, dear cousin. And in one of the emails, Peter wrote, "It is indeed an odd turn of fate that we have been in contact for so many years." At the time, in 2020, was 45 years. And Peter was in Joan's lab, and I'll tell you a bit more about that, in 1977. And he was in Barcelona when the first post-Franco election occurred. And Joan dragged him down to the Ramblas to look at the results of the elections. So, I want to tell you a couple of stories in Charlottesville. We and Rosa and Joan lived in the Copley Hill housing that was postdoc and students housing. And these were the housing, I believe this might have been the one where Rosa and Joan lived. And I recall one time when they invited us over their place to make paella. Now, In a small apartment, there was no room to make paella inside. They didn't have a coal grill to make the paella outside. And I remember that Joan and Rosa were arguing about the order in which to add all the various ingredients of the paella. One particular thing that struck to my mind, does chicken go first or green beans go first? But it doesn't matter. That was my first and best ever paella. Thank you, Rosa. Now, another story is, the ones of you that are old enough will know that in 74, 75, and even later, it was very expensive to buy P32 ATP. So we had to synthesize it and make it in the lab. So in Joe Lerner's lab, we took turns. And one time it was Joan's turn to make the P32 ATP.
Was it alpha or gamma?
It's gamma.
Gamma.
It's ATP. So, and it's P32. So Joan set up carefully, all boarded up to make this prep. And the prep usually was done in a hot lab that was away from the main lab. So after he finished the prep, somebody using a Geiger counter traced the radioactivity from the hot room to Joe's lab. So Joan was sent home to shower, change his clothes, and the next day, Rosa, you remember that? Rosa said that even his private parts were contaminated. So these are nice memories. So in 75, Joan returned to Barcelona. Peter joined me in Pisa. 76, Joan visited us in Pisa. And the summer of 77, while Peter was still was in Pisa with me, Peter joined Joan's lab for 3 months, where he was teaching Joan's students to purify and analyze glycogen synthase. And this work resulted in a paper that they published together in 1979. Actually, this was really emotional to me, going down to clear Peter's office. I found the notebook where Peter had the notes from the time that he worked in Joan's lab in 77. See, isoelectric focusing, GS, Barcelona, summer 2077, with Joan, and signed by Peter. These were the days where we were still writing our protocols by hand. And this is the paper that resulted from Peter's 3 years in Joan's lab. And actually, while Peter was in Joan's lab, he shared the desk with Joan Massaguet, who you all know. And that was at the time that Peter talked Joan Massaguet to join the lab of Mike Jack, which at the time was a young rising star in insulin signal. And the rest is history. Now, as I said, Joan, Peter, and myself were very close friends. And although we all worked on glycogen metabolism, we were never very strong competitors. Joan mostly worked on liver and brain. We stuck mostly with skeletal muscle. And so we really had the highest respect for each other. And as said, we have been friends for 50 years, exchanging visiting and interaction at meetings. And I'm going to show a series of pictures. This is 79 when we visited Rosa and Joan, walking down Las Ramblas. Joan took these pictures. This is with Rosa. Joan obviously was taking these pictures, and This is 2011, when we went to Charlottesville, Virginia, for Joe Lardner's 90th birthday. And I love these pictures of Joan and Joe, and this is all of us. Again, only one surviving here is me. This is also Charlottesville. You can see how happy we were together, always smiling. We never had a hard word for each other. And here are the two cousins, beautiful. And the ones of you that knew Peter, you know that Peter was the most gentle soul that you can imagine. And so was Joan, obviously. This, in 2017, the La Fora meeting was in Barcelona, and Joan and Rosa hosted a wonderful dinner at their house. And this is a picture at their house in 2017, and Joan, Peter, Mariana, the granddaughter, and Oriol. And this was a meeting in San Diego. Now, in 2019, Indiana University honored Peter and myself for 40 years of service at Indiana University. And these were the speakers, obviously centered on glycogen metabolism. Joan, Philip, and Matt. Three of the main people are there. This is Joan giving his talk. And actually, Jordi showed the same picture. This is the picture that you... I made that one. Oh, you made that? Sorry, it's possible. But this is the first time I saw that. And he presented, he started his talk with the introduction of good glycogen. That was his... And now, you all know that Joan liked to joke and had a very good sense of humor. And look at this. Isn't this beautiful? And he was entertaining people at the dinner. And note his notorious scarf. Joan always was afraid of catching a cold. And even in the summer, he always had his scarf. And this is the same time was a party at our house. Look, everybody else, it's a short sleeve. Look at Joan. His scarf, and you know, at the meetings, and the Fora meetings, he always wore his scarf. So now, this is his family now. This is Rosa. This is grandchildren, Oriol and Mariona, and they will carry on Joan's family legacy for the future. Joan, we love you, and we miss you. Thank you.
It's a fantastic presentation. And now I would like, first I would like to thank all of you for staying during the day. Also I would like to thank IUBMB, FEBS and IRB Barcelona to join the forces and to organize this fantastic meeting. I believe that Joan would be proud on all of us. I enjoyed, I'm not from a glycogen field, so I can imagine that for all of you was a great experience. So now I would like to ask Francisc Posas, the director of ERB, to address you, but at the end, please do not leave. We would like to make a joint photo before we leave. Thank you.
Okay, so thank you very much. Dear colleagues and friends, as we come to the end of this special day, I would like to thank you, all of you, for being in Barcelona. And also I would like to thank the people that has been joining the meeting online. That has been actually many people actually following you. Of course, I would like to thank the organizers, the people that help actually to put all this symposium online, and obviously to FEBS and IUBMB to make it possible. Okay? So today has been a remarkable day, I would say, especially for us. We heard a lot of very outstanding talks on glycogen, synthesis, degradation, regulation, physiology, disease, new therapeutic avenues, and so on. But for us, has been much more than just a scientific meeting. It has been a gathering of a community that actually grew around a specific subject, but also around a specific person that was Joan, right? So Joan actually had many faces, and we have seen actually comments about those all around the speeches that we had today. So Joan, the scientist, is the one that we have reflected the most today. He has working on glycogen metabolism, muscle, liver, but especially with a great impact on brain glycogen, and we have seen this in many of the talks today, and especially because this was related with something that he always thought that it was important with science, is to do something that actually tried to solve a problem that had society, and this is reflected by the approach of the Lafora disease that I think that this is actually advancing also thanks to the contributions of Joan. But Joan was never just a scientist, okay? So Joan was a mentor. Many people here has already reflected this aspect of Joan having him as a mentor, as a collaborator, or even that has been just influenced by his ideas. But Joan had this special ability to recognize potential and to encourage young scientists to do and to progress, right? And if you just allow me, I just remember this one moment that I was a kid, actually. I was finishing my first year of biology degree. I was in a microbiology lab, just preparing media to learn actually how to manage bacteria and so on. And suddenly someone enters into the room. That was Joan, obviously. I didn't have an idea who was Joan. And just sits there and says, "What are you doing, kid?" I said, "Oh, yes, I'm preparing media so I can learn how to manage this bacteria and so on." He looks at me this way he was looking at people, right? And he said, "Oh, yeah, keep doing this and you will get far," right? And these were just small words, a small comment, but that was Joan, that had this ability that with few words had the potential to encourage young people to do things and to advance. No? And I suspect that many of you had similar memories of this interaction with Joan, that at some point he always was so enthusiastic that made us to believe a little bit more on what we were actually doing. No? And Joan was also leader, right? Joan thought that science was a collective effort. It needed collaboration. It needed strong institutions, and I needed actually the right ecosystem. And that's why he was fighting and belonging to scientific societies like national societies like the FEB and the COSE, but also international societies like FEBS and IUBMB, actually to fight for science and to actually try to convince these policymakers that was always very difficult, but in constant advocacy for research and education. So one of the, I would say, clearest expressions of this vision was the creation of the IRB Barcelona, right? This was one of Joan's dreams, and I would say is one of the most important legacies that he left us. So he imagined a place in which we could bring together scientists of different disciplines to tackle really health problems from the basic research towards finding solutions to those diseases and to those problems, because he really wanted that science was translated to society. So today IRB is just a vibrant international research centers with this clear idea. But beyond the IRB and all these other institutions that actually Joan helped to build, Joan's most ambitious legacy would say is the way that he was living and he was thinking about science, right? So Joan was thinking that science should be ambitious, collaborative, and open to science. And I think that this is one of the big issues that actually people recognize on Joan's always activity. So for Joan, research institutions actually, So for Joan, research institutions were not just places to make discoveries, but also places to educate and to inspire the next generation of scientists. And he was very much involved in this task during all his life. So to finish this tribute, I would like to share with you a short video that reflects on Joan's life, on science, but also contains some words of people that has been involved on this trajectory of Joan's all along the way. And I think that you will enjoy this video.
Everyone who knew Dr. Joan Guinovart agrees on one thing. Joan was an extraordinary man. But why? What made him so exceptional? Joan was a man of action. He made outstanding contributions across many different areas. In the academic domain, Joan had a fruitful career as a biochemistry teacher and professor at the University of Barcelona.
I el doctor Guinovart fou una persona extraordinària perquè em meravellava sempre la seva capacitat de trobar els aspectes positius de qualsevol cosa. Mai li vaig sentir dir un no. Mai li vaig escoltar dir això no es pot fer. Mai li vaig escoltar dir això no ho farem. Quan jo li plantejava alguna cosa menor, ell em deia, xiquet, serà complicat però ens en sortirem. Jo li havia sentit dir moltes vegades que no es podia ser un bon professor de classe, sinó ser un bon investigador.
Recuerdo que era un catedrático que estaba súper liado, tenía el típico despacho de catedrático grande de la universidad, en la Facultad de Química, y él era el director del departamento, y si querías hablar con él durante el día, te lo encontrabas siempre hablando por teléfono.
Joan was a scientist, a man who sought to understand the world through facts, a man ready to face the unknown, a researcher who led his research group into a less travelled but fascinating scientific field.
No hi ha res més excitant que fer recerca, descobrir el que ningú ha vist abans. Explorar solubles de turistes, explorar espais en què cap altre bé humà hi ha entrat abans. Això és el que és fer recerca. No hi ha res més engrescador i més satisfactori. L'eureka d'avui entès per primera vegada és orgàsmic.
Yo creo que era un grupo líder brillante que combinaba su gran capacidad intelectual, que era capaz de llevar seres multitarea, para siempre abordar un objetivo único, que era trabajar en el metabolismo del glucógeno y trabajar en alteraciones que este metabolismo implicasen alteraciones patológicas en diferentes enfermedades, etc. Y al final acabó en una enfermedad neurodegenerativa que nunca él hubiera pensado que iba a acabar trabajando en una enfermedad neurodegenerativa y además rara.
Two genes, malin and laforin, monitor that glucose does not accumulate in neurons. When one of them fails, the glucose builds up and causes the death of the neuron. This is the beginning of Lafora disease, a rare form of neurodegenerative epilepsy that affects some 20 people in Spain. The disease manifests itself during adolescence with seizures and the progressive loss of brain functions. The degeneration of the nervous system causes the patient to stop moving, talking, or eating. There is no known cure.
Malalties rares afecten a molt poca gent, però des del punt de vista científic són enormement interessants. De fet, hem après molta bioquímica gràcies a la gent que té malalties rares. Aquestes han descobert mecanismes que eren desconeguts i que no els haguessin anat a buscar si no hagués sigut perquè fallaven en una sèrie de pacients.
What Joan did was ensure that the next generation of scientists were ready to take on and move the scientific knowledge, the scientific curiosity forward. And he wasn't only limited to Barcelona, as important as Barcelona was to him. He wanted to make sure that Spain, Europe, Latin America in particular, and the rest of the world were able to move forward.
Joan was an excellent mentor who was deeply committed to educating and guiding his students into becoming top-level scientists.
Él tenía muy claro todo lo que es el proceso científico, el espíritu crítico, el aprender, la humildad de saber que hay gente que sabe más e intentar aprender de ellos, el concepto súper importante de equipo, de colaboración. Me acuerdo que nos decía ir a los congresos, interaccionar con la gente, aprender, enseñar, y todo esto son pequeños puntos que hacen que Joan haya generado una escuela científica extraordinaria.
Joan Guinovart mentó diverses coses. Primera, un positivisme explosiu. Segona, donar ànim a l'estudiant, donar-li plena confiança. Tercera, creure absolutament en la possibilitat de l'avenç científic. Quarta, reconèixer el mèrit de l'estudiant i la idea de l'estudiant mai adjudicar-se a si mateix.
Joan, com a mestre, primer era generosa amb aquells que s'esforçaven, que ells veia compromesos i que ell veia que tenien ganes d'empènyer un projecte, una fita de la recerca. I era generós perquè era generós en facilitar un ambient, buscava sempre que els seus deixebles trobessin un entorn on desenvolupar-se, on maximitzar les seves capacitats. Jo també et diria que tenia una capacitat única, que és que fomentava i era capaç d'estimular la creativitat de la gent.
Un investigador mai no serà millor del que són els seus estudiants. Jo he tingut molta sort a la vida i he aconseguit que vinguin a treballar amb mi gent realment brillant. Crec que una de les meves poques virtuts és no haver-los espatllat. Realment, la primera cosa que s'espera que faci un cap de laboratori és estimular les capacitats dels seus estudiants i després permetre'ls singular i que ells puguin desenvolupar les seves idees i les puguin dur a terme.
Joan was a visionary leader who made his lifelong dream come true. He successfully faced an extremely complex challenge, creating a whole new biomedical research centre from scratch.
Si em preguntes a mi, jo diria que un dels llegats més importants que ens ha deixat el Joan va ser la creació de l'IRB Barcelona. I ara la nostra responsabilitat és cuidar de la seva criatura i que el seu llegat perduri.
Durant tot aquest procés, que va ser molt llarg, primer el part científic, aleshores el part científic com a hoste de l'IRB, i després tots els anys d'anar creixent a l'IRB van haver-hi moments molt difícils. El que va tenir és una gran resiliència, diguéssim.
En Joan Guinovart va ser un gran líder. Va aconseguir fundar, fer créixer i convertir l'IRB Barcelona en una institució molt potent, sens dubte, gràcies al seu coneixement científic, coneixement de les institucions, però també una forma de fer, de connectar aquesta ambició amb les persones, a transmetre un sentit col·legial a la institució sense deixar de ser ambiciós, alhora que exigia cuidar els investigadors perquè donessin el seu màxim a preocupar-se per les seves vides.
En Joan era un innovador, tant com a investigador com a director de l'IRB Barcelona. Va creure fermament en la transferència de tecnologia i en l'impacte de la recerca.
En Joan tenia un interès molt genuí i molt autèntic per les persones i per totes les persones. Ell anava a un acte i connectava amb la consellera
i també connectava amb el taxista que el portava a casa. D'aquesta manera d'actuar feia que tots ens sentíssim molt especials.
Joan was a gifted communicator who believed in the power of words. He was a pioneer in advocating that IRB Barcelona should not only develop the best basic research, but also communicate it to society and promote scientific vocations among future generations.
Què és això de la curiositat? La ciència és curiositat, la ciència... D'on surt la ciència? Com podem saber nosaltres si tenim un noi que nano que coneixem una nana que coneixem que vol ser científic?
Tots són científics quan són petits, perquè tots volen descobrir com és el món, tots volen entendre com és el món, tots tenen preguntes per fer-se sobre com funcionen les coses. El que massa sovint som els adults que els hi matem aquesta il·lusió.
Joan va ser un grandíssim comunicador científic. Sempre estava predisposat, convençut de la necessitat de connectar l'IRB, però en general els centres de recerca, la ciència en la societat, i en aquest sentit pioner, i fins i tot innovador. Programes com Bojos per la Biomedicina, que s'han exportat a altres disciplines, o iniciatives d'art i ciència. Jo li tinc una grandíssima estima personal, sens dubte, però també professional.
At Crazy About Biomedicine, I, of course, learned a lot of new protocols, new techniques, like theoretically and practically, but most importantly, I learned a lot about all of the scientists that I could talk to and about their research, and that expanded my knowledge way more than all of the theoretical stuff that I learned.
Joan was a man of great passion and joy, qualities he shared in every role he embraced. Teacher, scientist, mentor, leader, and communicator. Oh, and we almost forgot, as a dancer too.
Moltes gràcies, Joan.
Gràcies, Joan. Gràcies, Joan.
Gràcies, Joan.
Moltíssimes gràcies, Joan.
Gràcies, Joan. Gràcies, Joan.
Gràcies, Joan.
Gràcies, Joan.
Moltes gràcies per tot, Joan.
Thank you for everything that you did for the world.
So I think that with that, we will close the symposium, and I would like to thank you again for being here, and above all, thank you, Joan. Okay.
Very nice. Really lovely. Really